Queenie?
Bluetooth connected. Bluetooth disconnected. Bluetooth connected. Bluetooth disconnected.
Hello.
Hello, yeah, Shishir. Can you hear me?
Good morning, sir.
Good morning. Can you hear me now?
You are audible, sir.
Huh?
Yes, you are audible.
Okay. So once ready, I can share the screen. Okay.
Hello. Hello. Hello.
Or then, yeah, automatically.
Sir, can you see all the controls on your panel?
Yeah.
Hello.
Yeah, I do.
Hello.
Huh?
A very warm good morning to all esteemed dignitaries, distinguished guests, and participants. I hope you all are doing well and embracing the serenity of nature here in Ramnagar Corbett, surrounded by lush greenery and the soothing sound of the wilderness. This gathering is truly a perfect blend of knowledge and tranquility. So, we are going to start our second scientific session, for which I would like to invite Professor Dr. Y.K. Gupta sir and Dr. D.K. Dikshit sir to chair the session. Dr. D.K. Dikshit sir had been Honorable Chief Scientist of CDRI CSIR Lucknow. He worked on green chemistry, medical chemistry, and nature product Lucknow. Professor Dr. Y.K. Gupta sir is President of All India Institute of Medical Sciences, Jammu, and very potent medical doctor and scientist devoted for the mankind. He has more than 200 publications in international and national journals and several chapters in book to his credits. Dr. Gupta is recipient of several awards, including Young Scientist Medal from Indian National Science Academy, Shakuntala, Amirchand Prize, and many more to his account. Over to you, sir.
Hello, good morning to all of you. We have our first speaker, Dr. Basak. He is a well-known person in Dr. Natarajan. Dr. Natarajan completed his master's in chemistry from 1979 from St. Joseph's College and carried PhD in chemistry from Bharat Hassan University, Tiruchirappalli, Tamil Nadu in 95. From 2004 to 2007, he worked as a scientist to Dr. Subhash Basak Group at Natural Resource Research Institute, USA. Between 2010 and 2019, he served as a CFO of Polymer company in Karur, Tamil Nadu. During this period, he developed 3 vector control products, including chemically treated mosquitoes recommended by WHO for controlling malaria remission. His contributions helped his company generate an annual turnover of $40 million and created jobs for more than 3,000 people at Kattur district, Tamil Nadu. In 2020, he returned in academia and is currently the head of research and development at Sharinathan College of Engineering in Tiruchirappalli. Dr. Nagarajan has published more than 45 research papers, authored book chapters, and written review articles. He has also written more than 20 visited countries to present his research. I now invite Dr. Natarajan to deliver his talk.
Thank you, dear Chairman. Can you all hear me? Hello? Can you hear me? Am I audible there?
Yes, yes.
Okay, sure. Thanks, Chairman, for introducing me. And today I'll be presenting topic on generating new chirality descriptors for the prediction of pharmacological and toxicological activities of chiral molecules. And before I proceed, I would like to add to this. Recently, we organized the 9th Indo-US workshop on mathematical chemistry here at Basak, and many of his collaborators came to our college, and it was an event where Dr. Saxena was also one of the invited speakers on day 1. So with this note, I will try to proceed. So the outline of the presentation is what is the background, why we started generating developing new chirality descriptors, then from Kahn-Ingold product rule to chemical graphs, then 3D to 2D to chirality indices, why a diverse pool of chirality descriptors are used rather than a single set of descriptors, then quantitative chiral structure-active relationship modeling using the newly generated descriptors. So the beginning of the whole program, why we went into developing new descriptors for chiral molecules is in 2004, Dr. Basak had a USDA grant of about $1.4 million, and Professor Hawkins from University of Minnesota was a co-PA, and Dr. Balaban was a consultant. I joined the group in 2004 as a senior postdoc, and from there we started. Then how we got interested in chirality descriptor is the principal investigator, Dr. Bloom. He had a chiral molecule as a potent repellent, an alternative for DEET. This is a molecule which has got 2 chiral centers, therefore it has 4 diastereomers. Of the 4 diastereomers, we separated them individually, measured the binding proportion, and the SS molecule, or the one which is having the SS, is having the lowest binding proportion. In the binding proportion, the lowest, that is the most active chemical. So it is the most repelling chemical, and we were posed the problem: how are you going to model when we have chiral molecules? That is how we started into developing chirality descriptors. I'm going back to the early days of where chirality was the word defined by Lord Kelvin. So he said that I call any geometric figure or group of points chiral and say that it has chirally if its image in a plane mirror ideally relates. That is, it is a mirror image where the object in the image are not superimposable. And that when we have only one chiral center, then we have enantiomers which are non-superimposable mirror image relationship. And more than one chiral center, we have diastereomers, where diastereomers do not have the mirror image. And chiral drug manufacturing is taking a big role because everybody knows the thalidomide controversy. So from after that, everybody realized that though there are 2 molecules and we use often decimates, only one of the 2 isomers, enantiomer, is chirally is therapeutically active. That does not mean the other is inactive. It may be having a negative effect or it may be acting a synergistic effect. Therefore, this was realized after the thalidomide controversy in 1961, but it took US FDA to come up with a regulation in 1992. That is, it took nearly about 3 decades. So after that, the decimers can be sold, but the pharmacological and toxicology effects of both enantiomers must be characterized. That's why people are now switching over to enantiomer pure drugs. And in the last 5 years, 80% of the small molecules that are approved by FDA are all chiral. 75% or more are single enantiomer, that is enantiomer pure drugs. And every year, about 200 chiral molecules are coming into market. So that means that to design new chiral drugs, fews are approached as we are using another pharmacological or designing other drugs must be used. But here we have to use a special type of descriptor. That means chiral molecules have to be numerically characterized. Once you use numerical characterized chiral, that is indices, chirality indices, the structure activity relationship modeling becomes quantitative chiral structure activity relationship modeling. And this requires a mathematical calculation of molecular chirality, and it is very, very important. Why don't we use, there are empty number of normal topological indices. Can we just use the normal topological indices? Now, it is not possible because in case of topological indices, they have the same numerical value for both the enantiomers. So let us go back to our fundamental organic chemistry, isomers. In isomers, we have the structural isomers, stereoisomers. Among the structural isomers, we have chain position, functional, or in the case of structural isomers, unless or otherwise you break a bond and make a new bond, you will not be able to generate the isomers. But in the case of stereoisomers, especially enantiomers, they differ only in the 3-dimensional disposition around the chiral atom or the chiral center. Therefore, the structural isomers may be very easily differentiated because they differ in connectivity using the normal topological indices, whereas enantiomers cannot be differentiated by normal topological indices because they have the same molecular graph. So what are we going to do? So any chiral index, there are an empty number of atoms. There are several atoms in generating new chiral indices, and many of these atoms can be classified into 2 major types. Adding a chiral correction to the connectivity matrix. For example, if it is a R isomer, we will add +1 in the diagonal element. If it is a S isomer, we used to add a -1 in the diagonal element and compute the new descriptors. Otherwise, apply a chiral correction factor to already existing conventional topological indices. That means for using the second type of generation of indices, the existing topological indices have to be calculated as a first step. So any new chiral or any new index developed, first requirement must be they should be able to differentiate the enantiomers. They should be able to discriminate. That means they should have 2 different numerical values for the enantiomers, and they must be used in application in modeling the toxicological or biological properties of these chiral molecules. So in most of the cases, as I explained earlier, many of these cases, there is only one set of indices are derived. That means one value for the S isomer, one value for the R isomer, or if we have a sequence of molecules, we'll have only one set of descriptors. And that means one size does not fit all. For example, if you are studying 2 pharmacological properties, or if you are studying for the same molecules, 2 different biological properties, the receptors are not going to be the same. Therefore, the same chirality index may not be able to fit. Maybe if you're able to fit one property, it may not be able to fit the other property. So we need a diverse pool of descriptors so that we can effectively utilize and qualitative chiral structure activity relationship model. With this, we went into developing relative chirality indices. It is a diverse pool of new chirality descriptors. And here, everybody may be wondering, this is the normal question that comes to the chemist mind. Chirality is a 3D phenomenon, whereas your topological indices are based upon graph theory, and where all chemical graphs are 2D. Then how are you going to convert the 3D phenomenon into a 2D descriptor? That is normally remaining in everybody's mind. So before we proceed, I will tell you that how we assign the RS configuration based upon the Kahn-Ingold-Verlag rule. First, we assign priorities to the 4 substituents or the 4 ligands attached to the central atom, that's a asymmetric atom. Then assign the priority based upon the atomic number of the atoms as you go from cell. Otherwise, I would explain like this. So this is a chiral molecule where the center is this is the chiral atom, the asymmetric center. So we go for the first atom, second, third, fourth. That means in the first, that we call it as the first sphere. In the first sphere, we go by the atomic number. If the atomic number is higher, then that will be the priority higher, A. Therefore, if they are same, then we go to the second sphere, then we go to the third sphere. That is how we will be able to identify the priority, that is A, B, C, D, the four groups, the four ligands attached to chiral atom. That means, though chiral Kahn, Ingold, and Pollack, when I specified this sequence rule, they never used the word graph. They just developed like this, but it is nothing but a graph. It is a tree graph. Therefore, I would prefer to say that Kahn CIP rule implicitly has used the graph theory. It is this institution I normally, in analogy, I would say Gregory Mendel, who was working initially on he never used the word gene or DNA, but whatever, those days gene and DNA was not on, but implicitly he was meaning whatever he said was only genetic. So same way here, CIP, though it is not specifically or explicitly saying graph theory, it is implicitly, it is nothing but a graph theoretical approach. Let us explain this with an example. This is the structural formula of a chiral molecule. So we are representing the form of a hydrogen included chemical graph. In this graph, we are going to identify the four ligands that are attached to the central carbon atom or the asymmetric atom. Then these are the four, and these four ligands, the bond that is attached to the chiral center, I have added a dummy atom so that these are the four, and these four are nothing but four chemical subgraphs. And now we go from the X, that's the dummy atom, then the first atom is everywhere carbon, but in this case, the carbon is connected to two more carbons, whereas the other the four. This decides the priority. Then in the case here, bromine, here it is chlorine. That is how we go from that. And A, B, C, D, the four ligands are identified. Therefore, CIP rule, we have the chemical graph, which is the hydrogen chemical graph. Then to identify the priority of the four ligands attached to the central chiral center or asymmetric carbon, we divide them into subgraphs. From the subgraphs, we can get the priority. So once we know the priority rule, it's everybody is organic chemistry. So we place so that the least priority group is away from the observer, and then if A, B, C are in the clockwise direction, the R isomer, if it is there, the anti-clockwise direction is S isomer, then that is a 3D. Then using the wedge formula, we convert the 3D to 2D, and then we have the Fisher projection. So this is the Fisher projection where the least possible, which is away from the observer, is kept at the bottom, whereas kept at the bottom. So, and this is ABC, this is R isomer, this is the S isomer, this is the Fisher projection. Therefore, from 3D, we are able to reduce to 2D, and this is nothing but a graph theoretical approach. Therefore, once we have the Fisher projection, then we identify whether it is ABC, that is clockwise and anti-clockwise. But what I am trying to do here is from ABC, in this case, I am converting it into a direct graph. This is A, B, C. This is nothing but a direct graph. In this case of a S isomer, that is ACB. Therefore, it is a direct graph, which is an ACB. Therefore, we can generate a new topological indices based on the direct graph. And we have given this formulation. For an R isomer, the relative current indices, we take A, delta, that the weight, we assign weights to all the four ligands. So A plus AB, A plus AB plus ABC, then ABC. That's what I would say. In the case of S isomer, we say A plus AB, A plus AB, sorry, A plus AC, A plus AC, ABC. That means this is the term which is differing in both the cases. So this is going to bring the difference in the two values that we are going to calculate. Let me illustrate with an example of, otherwise before proceeding, what are the steps in the calculation of relative chirality indices? First, we have a chiral molecule, converted into a hydrogen included graph, then identify the four ligands, separate them and form the subgraphs. For the subgraphs, you develop the right the Smil's graph from that, we can develop any number of all the various topological indices using the commonly available softwares. And these topological indices for the 4 ligands are treated as the weights of the 4 groups A, delta B, delta C, and delta D. Then substituting them in the formulae, we can calculate the topological indices. Let me illustrate this with an example of tyrosine. In the case of tyrosine, so the 4 ligands are NH2COH and then the benzyl group, and then the hydrogen. Hydrogen is not, whenever you have hydrogen, there are not many connectivity, 0. So I am for them, I have written the SMILES code. Using the SMILES code, for example, I have given first are the Randish connectivity indices, Balaban's indexes, and then shape index 1 of the order 1. And from this, if I substitute in this formula, A plus AB, A plus AB, AC, ABC, I am able to get 2 different chiral indices. For R isomer, it is 18.204. For the S isomer, it is 17.82. When I use the Randish connectivity indices instead of Balaban indices, it is 13.96 and 19.1. That means what I want to emphasize here is for the same set of chiral molecules, I am able to generate a series or a large pool of chiral indices, which are one for the R isomer, another for the S isomer. So computation of weights are, once we have written the subgraph on the SMILES code, we can calculate for all the 4 ligands, simple connectivity indices, valence connectivity, information content, shape index, whatever index that is available to be calculated using the common software like Poly, Dragon, Triplet, or Molcons, we can calculate it. And then now the question is, if the molecule is having 2 chiral centers, what are you going to have? 3 chiral centers, what are you going to do? In such cases, we will calculate the relative chiral index for each center and then take the root mean square. Let me explain this with an example. But this is 2,3-dibromopentane. So, for example, if you are going to calculate it for the 2s, 3s, then take this molecule. This is the first chiral center. For that, this is the ligand. When you consider the second chiral center, this is the ligand. So, for using that ABCD, we can calculate the chiral indices and then take the root mean square of the chiral indices. So, advantages of the newly developed chiral indices is for every pair of enantiomers, we have a large pool of chiral indices. They are based on different basics, different fundamentals, they are namely connectivity or neighborhood symmetry or nature of the bonds or information content or even electrotopological state. That means a multi-dimensional space is available, and for a set of molecules, these chiral indices, they are not there. Some of them are mutually correlated, some of them are not at all correlated. That means the correlation varies from 0.99 to 0.01. That means we have from mutually correlated to mutually uncorrelated descriptors, which can be used for the general pool. And this is always superior to having only one single set of indices. Therefore, we can use this in modeling the pharmacological or toxicology aspects of chiral molecules that are mutually uncorrelated. I'm trying to explain using 2 examples. This is the first example where we have taken the 2 biological property for the molecule, but this is the same set of molecules. This is 3-hydroxyphenylpiperidine. For the 3-hydroxyphenylpiperidine, I have this set was published by Waterbeam et al. In the year 1987, and we have taken it from the Medical Journal of Medical Chemistry. The 2 properties, they are D2 and sigma affinities, dopamine D2 receptor and sigma affinities of the 14 molecules are molecules. There is 7 pairs of enantiomers are given, and the 2 biological activities are uncorrelated. That means the correlation between the sigma D2 receptor activity and the sigma affinity is only 0.165. That means if we have only one set of descriptors, it may fit D2, but may fail miserably in fitting the sigma affinity. That is why when we have a large pool of descriptors as we have, we are able to model them, and we have 140 descriptors for the 14 molecules. Therefore, we have 14 by 140 matrix, and instead we did the principal compound analysis. After principal compound analysis, we are able to fit both the sigma. We are able to model the sigma affinity as well as the dopamine D2 receptor affinity. That means this example illustrates that even if we have mutually uncorrelated uncorrelated biological activities, and we can use related correlative indices because it's a large pool of descriptors. We can use them to model even mutually uncorrelated biological property or pharmacological. And the second example I have taken is where we have a diverse set of compounds. These are hemaggrenin receptor type 2 antagonists. These 14 for these 20 molecules, we have the antagonic property. It is reported in 2008, and Nair et al. Used it using the hologram descriptors to model it, and we used the relative correlative indices to model the same set of compounds. For that, we calculated 190 chirality descriptors. From the 190, we extracted 5 principal components, and we are able to model this using the principal components, and we are able to get reasonably good R square as well as the adjusted R square. That means the second example also where we have a diverse set of chemicals, but for that we have calculated that again a diverse pool of descriptors. That means diversity captures diversity, and from that we are able to model this. And this is the predictor versus we have used the benchmark as a Nair et al. Is the benchmark. We use the relative correlations as well as the principal components. Okay. So in conclusion, I would say relative correlative indices are able to distinguish enantiomers and diastereomers. In the case of isoisomers, they have the same value. Predictive models for quantitative structure activity, chiral structure activation modeling can be built. And in case of quite large pool of descriptors are that we can use data reduction if necessary. So the new chiral descriptors would be extremely useful in computational method precisely direct asymmetric synthesis. So chiral CAP may be considered as a two-way discrimination, and in CAP rule, it is graph theory is implicitly used, whereas we use graph theory explicitly. And pure quantitative chiral structure accumulation modeling needs not a single set of descriptors, but a large set of descriptors. Like they put them on a relativity scale so that it is like a large P scale, so that we can use it where the chirality is on a scale. As a different properties of one chiral set of molecules may recognize different aspects of the chiral structure. A family of mutually different indices are always needed. As we normally say that one size does not fit all. So one set of descriptors cannot be suitable to model all the biological properties of the same set of molecules. So as a relative correlative indices that we have proposed is able to give a diverse set of descriptors, and that will be very useful. And we recently published these results in the molecules paper journal, and I acknowledge first the organizers, especially Shishir Nandi, who was in touch with me to, but for him, I would not even attend this conference. And I thank Dr. Saxena. And my thanks are due to Dr. Jerry Clone, who was the principal investigator in the mosquito project, and he invited me to join the Minnesota group, the authorized Dr. Basser group. Greg was always useful in calculating the poly and triplet indices, and my student Nanbaragan who developed me the calculator for these indices. And this is a great thanks to everyone, and thanks for listening. Thanks for all your opportunities. I think I'm on time. Yeah. Thank you, sir.
Rajan,
Yes, sir.
I hope you can take some questions, if any.
Yeah, sure, sir.
Any questions, please?
Is there anybody asking a question? I'm not able to hear.
Hello.
Yeah, please.
Yeah, my question is that you try that if you take enantiomers in any quantity QSAR, and you
Yeah.
can weight +1, suppose for R configuration, -1 for S configuration, and 0 for racemic RS compound.
Okay.
How does the results which come after using these descriptors with this kind of analysis?
When you use the plus and the minus, for as I mentioned, okay, that may fit for a particular biological property, but the index that you develop may fit for a particular biological property. But when for the same set of molecules, we have a different biological property, which is I'm working on different for example, receptor. The receptor will be looking for something else, right? It may not be looking for the same property of the molecule. In that case, this using a +1 and -1, which is applicable for situation A, may not be useful in situation B or situation C. That is why it may not work in all the situations. It may work for any, for example, I would say it's a chance correlation. It may work for one type. For us here, what we propose is a large pool of descriptors which is encoding different aspects of the molecule. Therefore, if it may fit, one of these descriptors may fit for this property, another set of descriptors may fit for another property. That's why using +1 and -1, otherwise, enantiomerism is not like treated like Boolean, like a +1 and -1. That means black and white. That's that's the idea with which you developed this type of descriptors.
But you have not done a comparison. It would have been better if you could have taken a comparison between them, so that I think what you are saying it may be would have been put to be as it is.
Yeah, sure. Yeah.
Hello.
Yeah.
Thank you very much, sir.
Thank you.
my-
Thanks everybody. Thank you very much. And this.
Myself, Srishi Nandi, and
Yeah,
thank
sure.
you for your support. We are missing Dr. Bosak today.
Yeah,
He is my
like,
great teacher and
yeah.
great, yeah,
I happened to work
long
with him
association.
for 2 years, and from 2000.
He is very ill, physically ill.
Please.
Yeah.
Yeah, 2005, I worked with him, and still our collaboration continues. And
Yeah.
I happen to be in his group for all these years. Yeah, thank you. And Dr.
Yeah,
Saxena-
sir, my question is,
Yeah,
this chiral kinds of chiral drugs, after getting bind with the receptor inside
sure.
in vivo system, in biological system.
Yeah,
So after reaching into the receptor, after binding to the receptor means when drug receptor complex is formed,
yeah.
in complex form, does it still exist as chiral molecule? Am I clear, sir? Question is clear?
You are clear. So once it binds to that, once after the recognition is taken, for chiral molecule, for example, if the receptor is capable of recognizing chirality, it takes only the plus isomer or the minus isomer, or R or S will be go on fit. And I don't think it is no more a chiral molecule. Once it fit into the chiral receptor, the receptor, the molecule loses its identity, it starts behaving, it has to undergo the metabolism. So I don't think so.
Okay.
Okay. Thank you very much, Dr. Natarajan, for giving a talk.
Sure.
Whereas
Thank you,
you
sir.
can use chiral descriptors in QSAR. Thank you
Thank
very
you, sir.
much.
Thank you. Thanks, everybody. Thank you.
Our next speaker is a person who is well known to all of you and to everybody in the audience, Dr. Anil Kumar Saxena. Please do come. Dr. Anil Kumar Saxena, I have known him for almost whole my scientific career. He was my senior, and I will not go by the written description here. He started QSAR when this was not known in the country. He worked on those calculations using a pure scientific calculator when no computers were accessible. With that passion and the training he got furthermore, he came back and established a QSAR school in India. He's one of the prominent researchers in that. And not only QSAR, he's an important medicinal chemistry person also. His one drug was approved by Rajiv Gandhi, released by Rajiv Gandhi a long time back, who did not come to the market. He has several more compounds running into various stages of activity description. He has published more than 270 publications, I believe so, and so many review articles, enormous number of invited lectures in India and abroad. He traveled virtually throughout the world giving lectures, and he established couple of scientific institutions, of which you are witness to that. Now I invite Dr. Saxena to give his output talk.
So now I am taking you the field of drug discovery. My voice is pointer then. Point toward me. So the topic innovations in drug design, I'm put some case study. But first I will give you a kind of an overview. It is very difficult, very wide subject. At least you will be able to know a view. I would say that suppose you are going to a city and I'm taking you by a helicopter with a slow speed.
Nobody can
stop. Let's go off. Let's go off. Now it is okay. Now you can listen. So next slide. You know, invention of a drug is a very lengthy process. It takes about 15, 16 years to come.
Kahin pe echo hai, please.
I think everybody else can off the other. Or I shall be from here, I think. If this mic is working and this is not.
Follow mic nahi lagta. Ye तेज भी कर दिया.
Yeah, point is. going to not absorb. Okay, it is activated. So those kind of things can be also improved. Building of active substructure into molecule. Yeah. And then how it is done? It is done by conformational restriction on the molecules. If suppose they are next. The substructure approach is simple, that if you see a structure present in different molecule, then you know in how many molecules it is shown the activity which you wanted, and how many molecules it is not shown. So if you say it's mathematically, I will say substructure activity frequency. That means the total number of active compounds which have been tested, how many of those compounds had that structure. So suppose you have tested, say, 100 compounds, and in 100, only 50 have shown activity, then its substructure activity value is 50 upon 100.5. So in that way, if you have different substructures, you can add them, it becomes a minimum substructure activity frequency. Next. Now to this age, computer-aided drug design come, and I think it was early 73 year or 74 when I think in India I introduced this, and starting with the kind of a indirect design. There's two kinds of designs, direct design and indirect design. The direct design is you know the target, you know the structure where it is just to go. On the other one, you don't know the structure, but you know the molecules which are showing some activity. It is just like that you go to a tailor and tailor is blind. So one time that if he's not blind, he sees you, takes your measurement and makes it. Other way is he had a lot of shirts or one shirt, he fits you, and then he sees kahan se chila hai, kahan se tez hai, usko he can monitor. And so that kind of it is indirect design. So the ligand molecules, we use QSAR, structure activity relationship into this, while on the direct design, you know the structure and you can fit into molecules. Next. Now, there are advantages and disadvantages of these. I will not go in very much detail, but the direct design, it's a requirement of nowadays, you have the structure of the target. Indirect design, you don't know, but you know the ligands. The other thing is, it is highly complex, very high computational power, but I think they are going now faster. And then this is an abstract, while here minimum and sample, which when you don't have, you just have the biological data, because it is very expensive sometimes to get the biological data at the receptor or at the enzyme level. And then reliability, it is more reliable when screening against that diverse molecules on that target has been done. But it is as good as thing when you have a training set, then you have test set, and then use training set, test set means you know the structures, you know the activity, you develop these relationship. The test set is when this relationship is also good for those molecules which you not have tested. Means you predict their activity. So this all models are for prediction in one way. Next. Then also the other thing is, people were doing at one time STS, high throughput screening, which is also done, and use this word also in the computational screening as well. But the thing was, when it was STS, particularly for structure wise, then the thing was that you needed a physical amount of the molecule to be tested. It means you have to synthesize that. But nowadays, with this computer-aided design, the virtual screening means you can design, synthesize a lot of, in one go, you can have 1,000 or 2,000, even millions structures generated. While otherwise, it was very difficult to synthesize. So this whole approach, which came at one time in late 90s, as a combinatorial, this STS approach, then it went down, and then this is a virtual screening gives good results. Next slide. Now I'm coming to you to tell you the ligand-based approaches, how you develop the molecule, what is the basic about it. The molecular structure of molecule is related to biological activity. You can't correlate very directly. So not available when you have not target structure, where it is going. When you take a body, some drug, it goes into body, it can go to brain, it can go to anywhere. So if you don't know where it is going, where is the site, then you cannot draw. But if you describe this structure or this drug in terms of structures, which can be 0D, 1D, or 3D structure, then you can design structural descriptors. You have just seen an example just before my talk where they were designing the descriptors. So descriptors can design and define the molecule. And then from there, you can select some of them, and then you can correlate biological activity. Next. And the other way is, of course, it is a drug. The basic is drug reacts with the substrate or enzyme or receptor in the body, forms a complex. Then biological response comes after a lot of events. And this has been correlated. Actually, this energy, free energy, which is changed, is correlated to the hydrophobic descriptors, electronic effect, steric effect. So they are the thing. And then finally, and one old time, this was there that you don't know any descriptors. You just put the values, what the substituents are, and then show which are showing good activity. And sometimes it can be a combined approach where you take the physicochemical properties into consideration, and you take these one, which I was also asking a question to him. How does implying these indicator variables, which are structural descriptors, which you can't define, and then you can correlate. Next. So the steps and the model development is you have a structure, molecular modeling you do, your descriptors generate, then feature selection, then you construct a model, and then it becomes a predictive model when you validate it. Next. And in this, basic methodology is bioactive conformation, you should know. You can design a pharmacophore. What is a pharmacophore? Pharmacophore name has given which part of the substructure or a structure which is there in the whole molecule is responsible for particular activity. So it is called pharmacophore. And this pharmacophore, if you have, you can ligand receptor drug docking you do once you have pharmacophore, or you can have a bioactive conformation alignment, 3D equation. They're all interconnected approaches, and you can develop a model which can be a predictive model. Next slide. So what one does now, virtual screening, it means you have the candidate, lot of ligands, but you put forward through these models, advent model, drug has to be absorbed, excreted, all these parameters you want to improve, that the polygonal दवाई हम खा रहे हैं वो ठीक से यूज हो रही है, निकल तो नहीं जा रही, एक्सक्रीट तो नहीं हो जा रही, तो एब्जॉर्प्शन हो रहा है कि नहीं हो रहा है, ये सब भी मॉडल से हो सकता है, कंप्यूटर एडेड मॉडल्स जो है, we can put, then you can select out of say 5,000, you can come out with 200 or 300, 400. Then you can have similarity searching, कौन-कौन similars हैं, then you test them on the model which you have done for pharmacophore, and then you can optimize and act for activity. Finally, when you have it, you can use structure-based design when you know the target structure. Then you can select the molecules, and here you can validate. So in that way, this whole 10,000 molecules or so, you can come down to even 50 or 30 or 40 molecules. And then you can select, and that compound you can make and test. So it reduces a lot of money and time in this which you would have spent otherwise. Next slide. Now I'll take you to some examples. नहीं उसमें है ही नहीं, Shichiran. No, no, this is not the next slide. तो बहुत बाद में. No, this is you're coming backward. So I think before they collect it, I will give you some examples, real examples from my work. And the first example which I will show you will be the one when I joined CTRI in 69 or 70. How does that molecule, which was conceptualized using the pattern recognition and substructure approach, how that molecule showed activity and how did it further came up? That molecule which we selected and made, that coming out from the substructure means we saw it was thinking of making a neuroleptic drug. In the market, you have haloperidol at that time, chloropromazine, which were at that time used for this. And we had some leads of our molecules which were there with antihypertensive activity. So what we did, we found out at that time there was no computer here. And it was thought that, okay, this substructure, take one substructure from haloperidol, and you then incorporate all the three. Then that design molecules we synthesized. And that design molecules, in starting with only 30, 40 compounds, showed activity. And then it was developed, and finally that compound came or introduced from CTRA, was the earliest drug in the market, and was released for market by the Prime Minister Rajiv Gandhi in 1987. So it took also almost 16 to 17 years, because it was in 1986 while it was really conceptualized. Its number was 70 by 343 in CDRA, code number, means 1970 compound. The other compounds was through medicinal chemical hybridization. And medicinal chemical hybridization is that if you have one substructure responsible for one activity, other substructure for other activity, and other properties which you want to introduce, तो ये एक तरह से जो वैद्य लोग बनाते हैं औषधि, ये इसके लिए मिला के खरल में पीस दिया, तो उस तरह का मॉलिक्यूल आपने मेडिसिनल केमिस्ट्री में आपने सिंथेसाइज कर लिया, and उसको you will find that it shows those properties which you wanted, but you have to analyze. Again, this medicinal chemical hybridization can also be said that it goes with the cooperation of the substructure, and then it showed activity, and that compound. And the other, which nowadays you are listening of, artificial intelligence. You know, I was interested in artificial intelligence somewhere in 1976, 75, when I found a book in some place and I read it. And then I thought, what is to be done? Which problems which you cannot solve in the normal way, and particularly the complex biological activities. Yeah. So it is the first slide, which that slide that I told you, haloperidol, chloropromazine, oxypartine was also one of the compounds which showed active. CDRI 69183. These substructures, we took these together from these structures and incorporated this molecule. And this molecule is a drug. And then we did QSR, of course, afterwards for the molecules. And we found that this substructure, where one phenyl ring is separated by the neuroleptic nitrogen. When I say neuroleptic nitrogen, you have to find out what is the electron density for that neuroleptic nitrogen. So that was done in detail. And then we made this substructure. Now a question comes, intuitiveness, that if suppose this substructure you incorporate should show very high activity. So next slide. So I thought, this is the substructure. You are having a phenyl ring substituted with distances from this way. So I thought, why not make this? Here you have a repetition of this, because I told you mean substructure activity frequency. And then we synthesized this compound in 1986. And coincidentally, some neurology something, it was 86 by 373 again. That was 70 by 34. Sorry, that was 343, but it is 373. And there we found that this molecule was better than butanol. So that is called the intuition in a researcher. You think something, and then you make it. It's not easy to make this molecule. We made it, and these are all biological activity data on the receptor, because we were knowing the senputindol, which we prepared first, that was going to D2 receptor, dopaminergic D2 receptor, and that's why it is having some extrapyramidal symptoms, which is a side effect in one way of this. And this compound was better than that. We have of course patent, but this compound we did not pursue for a drug later on, because a lot of money is needed for development phase So we thought already we have one drug, we may not take that further. Next slide. This is the case I was talking about, antihypertensive, medicinal chemical hybridization. You have a slide before that? इससे पहले वाला पहली slide इससे पहले. No, okay. So there it was showing how this. So this compound is centaquin, which we had prepared for that. And this is the compound which I made through molecular modeling and this. And exactly this compound also showed the same activity, which it was showing. Fortunately, this centaquin, later on from one of the CDRA colleague, Dr. Anil Gulati, it has been now introduced in the market just 2 years back, and not for that antihypertensive action, but for other activity, dissertation activity for this. I think it is the reference given. Its name is Lifequin, used in desensitization agent in the treatment of hypovolemic shock, which is very common now, particularly for those persons who are having heart problems. Next slide. Yeah, this was the comparison you see. That compound and our compound. So this is 86 by our compound, and this is centaquin. And you see the values in animal model and so. And not only this, with receptors also. Next slide. Now, I was taking to you to this artificial intelligence. It is basically artificial neural net. And this is the approach, ANN approach, artificial neural net. So the problem was, I had a drug when I was working on antihypertensive. तो हमारे यहाँ एक फार्माकोलॉजी में डॉक्टर श्रीमल थे, हेड होते थे. उन्होंने कहा, अनिल तुमने बनाया, you have made this compound 86 by 3, which I have shown you. It is better because the blood pressure lower था उस compound centaquin का, वो था केवल 45 mm. इस compound का था 78. मैंने कहा मैंने इससे अच्छा बना के दे दिया आपको अपनी कैलकुलेशंस. उन्होंने कहा ये नहीं, पर ये चलेगा नहीं. हमने कहा क्यों नहीं चलेगा? उन्होंने कहा भाई इसका ड्यूरेशन देखो. जो पहला कंपाउंड है उसमें जो बिल्ली के ऊपर हार्ट पे देखते हैं तो उसका ड्यूरेशन आता है, कितनी देर तक इफेक्ट है. वो तो रहता है करीब 50-60 मिनट्स तक, और तुम्हारा तो बस 10 मिनट में खत्म हो रहा है. इमीडिएटली मैंने सोचा ये कैसे ऑप्टिमाइज करें. तो फिर हमने सोचा भाई इसको टाइम को करो, कोरिलेशन कुछ नहीं आया, कोरिलेशन आता ही नहीं था, कोई मॉडल डेवलप नहीं हो रहा था. तो मैंने सोचा आर्टिफिशियल इंटेलिजेंस लगानी चाहिए. इन द एट दैट टाइम यूज्ड ANN, and this we developed. Next slide. ये सिंपल थ्योरी है ANN की, बैक प्रोपोगेशन हमने यूज किया, और बहुत डिटेल्स में मैं नहीं जाऊंगा. जर्मनी में मेरे कोलीग थे डॉक्टर शापर, इज नो मोर नाउ. वो इसमें काफी शाह थे, तो मैं वहाँ 3 महीने के उसमें वैसे ही गया था. तो हमने फिर सोचा इसको मैं डेवलप करते. तो हमने इसमें डेवलप किया. तो इसमें 2 प्रॉपर्टीज आ जाती. एक तो आती है फार्माकोडायनामिक, जो आप उससे कर लेते हैं, मीन्स उस पर एक्टिव हो रहा है. दूसरा काइनेटिक्स. काइनेटिक्स होता है जो हमारा वहां कैसे पहुंच भी रहा है, मॉलिक्यूल नहीं पहुंच रहा है. वो पार्ट करके हमने क्या किया, इसको प्रॉपर्टीज दे दी, और प्रॉपर्टीज देके उसको बार बार कंप्यूटर से जनरेट करते हैं कि ये जो प्रॉपर्टी है ये कितनी है. जैसे आप किसी की बैल कभी पहचानते हैं जब आप वो बैल सुनते हैं कितनी बार, वो आपके न्यूरॉन्स में जाता है. वही थ्योरी करके नटशेल में देखा कि पाई और सिग्मा ये कर्व्स हैं जो उस समय, अब तो और भी बढ़िया आने लगे. This was in 2000 or so, 2002. हमने डिजाइन किया. Finally, next slide. We could simulate that. जो कर्व्स आते थे उनको हमने simulate करके prediction भी कर दिया. Next slide. And some of the compounds, and then we put it that you can use, and one has to use these kind of approaches when it is very complex, because it's very complex. आपने ड्रग दिया और हम उसको होल एनिमल में एनालाइज कर रहे हैं, रिसेप्टर पे नहीं कर रहे. नेक्स्ट स्लाइड. नाउ आई विल गिव यू एग्जांपल आल्सो वेरी फॉर एंटी डिमेंशिया, अल्जाइमर. आजकल ये बहुत कॉमन डिजीज है, आदमी भूलने लगते हैं. इसके अंदर एक एंजाइम है एसिटाइलकोलिन. तो हम एसिटाइलकोलिन स्ट्रे जो उसकी हाइड्रोलिसिस हो जाती है उसको रिमूव करने एसिटाइलकोलिनेस इनहिबिटरी. इसमें हमने क्या किया, पहले बहुत से मॉलिक्यूल्स लेके मॉडल डेवलप किया. उसके बाद में कुछ अपने मॉलिक्यूल्स डिजाइन किया और फार्माकोफोर एनालिसिस की. उसके थ्रू हम इस पे आ गए स्ट्रक्चर. और इसका ऑप्टिमाइज करके कैंडिडेट ड्रग बनाया, पेटेंट भी है, उसके सबके भी पेटेंट्स है। नेक्स्ट स्लाइड। ये मॉलिक्यूल्स थे, और जो ड्रग है मार्केट में डोनाजेपिल, उससे हमने इसको correlate किया, we found, and this compound was very good, and इसकी एक्टिविटी नेक्स्ट, ये पैसिव अवॉइडेंस रिस्पॉन्स है जो स्कोपोलामाइन से इंड्यूस किया जाता है, उसमें दिस कंपाउंड शोड हाई activity as compared to donazepam. Next slide. And we correlated these with our model, and it was very well fitted. So this is where the computer-aided design was fitted, and we could get the molecules for Alzheimer's disease. Next. Now, last say when I joined after my superannuation, I was given a responsibility just 1 year before for tuberculosis, which I had never worked. So there on tuberculosis also, which is also a very big challenge, we have developed, next slide. You see the data there. You see the green here, it is very much common still, the disease. In India, it is there. Then these are what are the biological characteristics we should have? It is mainly yesterday you have seen Dr. Y.K. Gupta, who got this microbial resistance, which is a major problem with TB. So MDR resistant drugs. For this, we have also wanted this की frequency का. Other important aspect is that इसको बड़ा regularly लेना पड़ता है. Tuberculosis को 9 months का treatment है डॉट से, तो आपको बहुत रेगुलर लेना है. डोजेस कम हो जाए, ड्यूरेशन कम हो जाए लेने का, ये सब चीजें हैं इम्प्रूवमेंट के लिए. That's why this whole area was taken. Next slide. And you know, these are the different molecules जिसकी combinations में ये हम लोग इसको लेते हैं. And then this one compound is there, betaquilene, which was introduced, और ये एक receptor system ATPase synthase में करता है, उसको लेके next slide. ये XTR है, extra resistance. एक तो ये multi drug resistance, उसके ऊपर भी हो जाता है फिर extra resistance, that is there XTR. Next slide. And this is the enzyme F0 ATPase synthase, which is in the system. This has been isolated, and one can have the ATPase synthesis activity. So we optimize through structure-based part. Next slide. And also made bioavailable. Again, the thing is that we played these molecules, we synthesized very few molecules, and we found that, next slide, one of the compounds are very good, and that's why we had to file patent for this. Next slide. Yeah. So one of these compounds was very good, which has been here. And you see that next, we developed the models. And you know, I think you were listening about stereochemical descriptors. So here the problem was also the same thing. We had a chiral center. So we had R configuration and S configuration, which we tackled with this molecule, not by having a substructure, but putting them at 1, 0, -1. And then this was quite successful. And we published it also, and it could make this model. Next slide. And not only this, later on with this receptor, what we have done last year in the same conference, I put a new novel where the drug receptor interaction, which have never been quantified, never been described, that we did. And I think using that approach and the topological parameters, which you were just listening from him in the previous lecture, we made this kind of a model which is all interconnected. And this novel approach, which is called quantitative structure interaction activity relations, this is QSIAR. Next slide. We could make this model, and that model has worked very well in the prediction and validation. Next slide. This was a model, and we finally came out with this model of just, which is very good in terms of correlation and predictability results. Next. And finally, I think these are the other papers which have been recently published. I mean to say in you will see here 20 onwards, 23, 24 even, which we are continuing still. Next slide. And this takes into account the interactions. Next. And automatically when interactions come, the problem of if you have the data, it is fitting, and how the Shishin Nandi's question was whether the receptor, what kind of conformation it is taking or not taking. that come together. And you know, this is the QS equation, which is describing 74% of the data. Next slide. And this has been done on diverse set. A paper appeared in GTHTM 2017, and Dr. Mr. Sarfraz is there, who has used it, and it predicted very well this model, the activity. Next slide. Very good prediction. Next slide. Published in also very recently. So this model validation, again, we thought that is it possible? What should we do? So we have predicted the 3 molecules or 4 molecules and got them tested at CDRA. Next slide. This was done very recently. This is PhD thesis, these 3 compounds. And here are the activity which are predicted and which have been really observed and show good correlation, except one count. So what the conclusion is that drug design is crucial in drug discovery, as you have understood. The process involves 2 main stages: identifying the target lead molecule or optimizing the lead to develop the drug. Traditionally, lead identification relied on natural products, disease, random screening, or human pattern recognition, which I have given example. And CAD helps in drug discovery challenges. CAD methods include ligand and structure-based approaches, which I have just told you an overview. It allows efficient virtual screening of vast molecular databases, including focus libraries. CAD also promises repositioning of drugs. I think one slide I missed, which is not there. Nowadays, if you have a model and you already have drugs in the market for something or the other, so their toxicity, their availability, that is proven. Now, if you can find out that this particular drug can be used for other disease through these approaches, it is called repurposing of the drug. And many molecules are being taken, and basically the dealing of COVID pandemic, this was a great success. Very great success. And I will just give you, tell you that we had a CTPMS just before Corona. And one of the Ronald, he came from Denmark. He was sitting behind me and he said, "What do you think, Dr. Saxena, chloroquine?" I said, "It should be," because that was my pattern recognition. And later on, I got a letter from him, after 2 months, that Dr. Saxena, can you tell on what basis you predicted that? So because there is one companies, I told this, and they are asking, what is the, so there was no, there's simply my brain worked. That is the pattern recognition. And then I was thinking how to justify. And then I told why I thought it over. So this is called the neurons. And then that compound, of course, people were giving chloroquine in the pandemic. So repurposing of the drugs is of very important high use. And I think this example, which I have neuroleptic drug, then we have this lifoquine, which is xanthine, Alzheimer's disease molecules, ATPase synthase inhibitors, I think it's very well listed. Thank you very much for listening, and I shall be happy to give answers. Okay, take this. Yeah. Yeah. That's why it is comparatively easy to work on infectious diseases, and particularly parasitic disease also, because the mechanism of simple, you have to kill the bacteria, parasite or something. But when it comes to central nervous system, I think in which I had one of the expertise in CDRI, CNS, CBS. Nobody was coming. Dikshit knows that nobody was working in that area. Here, the reason is, here we have so many receptors. Even simple take dopamine receptor. At the time I joined in 70s, it was only two receptors, one receptor dopamine. Then it came in D1, D2 receptors. Then it came to D1, D2, D3, D4, D5 receptors. Similarly, you have 5-ST receptor, 5-hydroxytryptamine receptor. So we used to think that, okay, some very edge, the compounds which are active on D2 receptor, they are basically classical neuroleptic, sclerotonin. But they have got extrapyramidal catalepsy, means if somebody takes, stiffness comes and these, and tongue twists, many things happen. And this particular effect is D2. I think time will permit or not. I can tell you an example, when we were having a phase 1 trials, one of my student, not my colleague, MN Rao, he developed at the higher dose. Tongue started twisting, going. And people got registered. We were all friends who were volunteering ourselves. And I was the first one to come, and I think Dikshit must have also been there as volunteers. We were working. So when this came, we rushed to the clinician, Mrs. Nityanand, and she said, she jumped on this. She said, oh, then it is a neuroleptic. Because it is the side effect neuroleptic. So when she observed this, they became, instead of becoming panicky, which we were, she said, oh, it means it is. And really it was. Now, if you act on 5-ST2 receptor, then that removes this thing. And which I later worked. And which was introduced in 86373. That means that your extrapyramidal symptoms can go out or can be lessened. Simultaneously, there is two kinds of negative symptoms and positive symptoms of schizophrenia. There are two kinds of schizophrenia. The positive symptoms are normally people take, and then I think they are cured. But negative symptoms is no drug. Now risperidone has come. Negative symptoms is when the person who is suffering, she or he will not try to go to a society. आप उससे कहेंगे घर में है, वो सोचेगा मैं घर से जाऊंगा नहीं, पार्टी में नहीं जाएगा, यहाँ जाएगा. वो नेगेटिव उसको नहीं, लेकिन हमारा ड्रग 5-ST2 की प्रॉपर्टीज मैंने इंट्रोड्यूस की 86373 में through my medicinal chemical habit, and that compound was there. And stereoisomers of sandpitendol में भी है, एक में है, एक में नहीं. So I mean, say इतना जो receptors है, इतने ज़्यादा है, इतनी complicated है. That's why what your question is कि neuron में क्यों नहीं हमारा उतनी जल्दी आता. That is the reason.
The drug research has been a journey where you don't know anything from the beginning, now to know you many things. Like when Dr. Saxena started this work, and many in the country and globally, receptor was known there is a receptor, but they don't know what it is. It was like a mystery woman. But slowly it was delineated. Now it was genetic sequences, receptor subclassification occurred not because of the functional changes, more so because of the genetic changes in the receptors. And now they are more amenable to USAR and multiple modeling. Even in that time frame, when Dr. Saxena was starting, it was a black box. And I think Dr. Saxena must be given an award for Lifetime Achievement and Passion Award. With the passion he started this work, with the passion he is doing it now, even after retiring in 2012, now it's 2025, and he had the same passion. I salute him for that. Thank you, Dr. Saxena. Our next speaker is Dr. Diwan Singh Rawat. He is again a person who you must appreciate. He did his master's in 1993, and now he's vice chancellor of the same university in Nainital. When he came to CDRI with the open eye researcher, he did work, did a postdoc in Lafayette, came back and served for a brief period in Naipur Mohali. Since then, he moved to Delhi University, where he had taught. He has given so many PhD students. He published more than 170 publications. But that's not important. The important point is he has started working on a couple of molecules. One of his anti-Parkinsonian compound is got permission as a drug. Now phase 1 has been completed. I think phase 2 just about to start. And not in India. It is basically in USA and Korea. So I salute you for that also. Please, Dr. Rawat, describe your journey to phase 1 program of the same drug.
Uh, so today what I'm going to talk, some of the work we did at University of Delhi. You can see my affiliations, there are two logos. One is University of Delhi and another is Puma University. And University of Delhi, actually, I started my journey way back in 2003. Let me just show you a couple of slides because nowadays, wherever I go, I try to expose the audience about the university where I am working with. And then after that, I will come to my talk. So, okay. So this is University of Delhi, the top part. This is called Viceroy Regal Lodge. So till 1933, Viceroy of British India used to operate from this building. And then they moved to Raisina Hill, and it was given to University of Delhi in 1933. And this building has huge historical importance because Gandhi-Irwin Pact was signed in one of the room in this building, number one. Number two, you must have seen Bhagat Singh sitting on a cot, right? निवाड़ की चारपाई में बैठे. तो वो जो पिक्चर ली गई थी वो इस बिल्डिंग के अंडरग्राउंड में एक रूम है छोटा सा, उसमें उनको रखा गया था. एंड जो आयरन ग्रिल्स हैं वो इंग्लैंड से आए थे, आज भी वो वैसे ही प्रिजर्व किया गया. एंड this is the central block of chemistry department. Actually, CSR was started from this place. And जो chemistry department and physics department है वो kitchen हुआ करता था वाइस. And this is the central block, and my office is located to this side. My research laboratories to that side. And since July 23, I am with Kuma University now. That's the university from where I studied actually. And this is the administrative block of Kuma University. We have 10 faculty, 33 departments. Our sanction posts are 205, 83 colleges. Student strength is about 70,000, and we have 2 campuses. And my accommodations to this side about 100 meters from here. And that's the accommodation. And I strictly warned not to come out because we have a very good companion. We have a couple of leopards to this area. So in night, I cannot come out my own. We have with somebody with me. And that's the lake. And this part is the campus, what we call DSB campus. And this is the another view of the lake. It's a beautiful city. If those who have not visited, it's worth to visit. And this is the DSB campus, and this is the administrative block. This is where I sit. But if I have to come, I come there. It's about 2 kilometers. And another campus is in Bhimtal, another beautiful small town. And another campus is here. We have 4 departments in this campus. It's a very cold place. At times it snows heavily, and nights are mesmerizing. This is how it looks, the city. And when I joined university, actually, I always keep thinking whenever I was in Delhi University or so, that why Vice Chancellor doesn't go to the classroom and teach. But after I joined, I found that it's not that easy because you have so much of administrative responsibility. But I still try to find out to spare 2 hours per week to go to the classroom to teach. And this is the class, the picture I took just 3 days back when I took the first class of this semester. And this is the same classroom where I studied way back in 1993. So I was a student in this class. So a couple of things about the initiative which we took at the university. We got 100 crores from an under Meru program from Government of India. We got 26.4 acre land very recently. We started internal research funding. We are giving 2 lakh rupees to the faculty member to do research. We established Biofloc Fish Technology Center, Hydroponics Research Center. We are giving SSB training to the students free of cost. We have a talent hunt program for UG and PG students with an annual budget of 30 lakhs. Orphan students are getting free education now at university. We have visiting professorship directorate. You name any top Indian academician, They are visiting professor with Kumaon University now. We save about 5.5 crores in printing job. And that money utilize what the schemes I'm telling. We are giving the best awards to the teacher. We have a patent cell, honor the Kargil material. I'm taking classes. Open tender for confidential printing. Kumaon University became the first university in the country to go for a tender for question paper printing. So we are funding to master student, PhD student faculty for conferences. We have introduced this internship program which government of India has introduced last year. Before that, we introduced that. We have hostel innovation 50 years, first time the hostels were renovated. Fellowship to best PhD student based on their publication. We are giving 5,000 rupees scholarship for 1 year. And we have a Vidya Setu program to connect our colleges with the university. And we have recently purchased the e-books which are made available to college students also. And we have a library championship award also. With this brief note, let me come today what I wanted to talk to you. As I said yesterday, organic chemistry starts from Euler's urea synthesis in 1828. And Euler is known for his urea synthesis and many more chemical processes he developed. But he was not a chemist actually. He was a medical doctor. He was taking the subsidiary classes in the lab of Gamlin, Professor Gamlin. Gamlin noticed that this person has a very good knack on chemistry. He asked him to join the lab of J.J. Berzelius. J.J. Berzelius' hypothesis was organic compounds cannot be synthesized in the lab. At the age of 28, he made urea in the lab, proved his boss that you are wrong. So this is what it happens. Then the oldest, okay, oh, sorry, I think I went to the next slide. Anyway, so the oldest written evidence of medicinal plants uses as a drug has been found in a Sumerian clay slab from Nagpur about 5,000 years back. That's the oldest written evidence. It comprises 12 recipes of the drug preparation, referring 250 various type of plants. Some of them are alkaloids and all that. So this is what I was talking actually. Sorry, okay. So in 1900, the average life at birth in India was 23.5. Now it is more than 66 years. So chemistry played a pivotal role on this. So Dr. Saxena made my talk a bit easier because he has explained it so well. So I'll not go with this talk. It's a very risky and time-consuming process. So what I will talk today is one of the approach that is called hybrid drug discovery. So I will concentrate on that. That's a problem. The chemtrail structures are not working, I guess. Look, why can't you use my laptop? I could sense this one Dr. Saxena was making. No? No, why can't I use my laptop? Leave it. Streaming is okay, I think. If it cannot stream, that's okay because I can use at least my laptop. I bought that. नहीं वो नहीं हो पाएगा शायद कर कोई बात नहीं होता तो छोड़ो उसको कम से कम ऑडियंस तो देखेगा Yeah, sorry for this inconvenience. Actually, I realized this when Dr. Saxena was making a presentation. I rushed to the hotel and brought my laptop. These people said, no, it will work. But anyway, so let me just show you this slide. How many of you have seen this plant? कितने लोगों ने सभी ने देखा होगा इसको सदाबहार बोलते हैं and this has been source of 70 alkaloids. 70 alkaloids have been isolated from this. And structure of two of these alkaloids is mentioned here. Both of these alkaloids showed poor anti-cancer activity. But when these two alkaloids, 1 and 2, are joined by a covalent bond like this, these are the two different compounds called vincristine and vinblastine. In one case, you have a methyl group, another case, you have a formyl group. Both of these molecules are blockbuster drug for the treatment of leukemia and licensed by US FDA in 1963. But synthetic medicinal chemists utilize this concept very late, and I will concentrate on that today. But before going to my presentation, let me show you what kind of training I had, because I am PhD from CDRI. So wherever I go, people feel that I'm a trained medicinal chemist. So Dr. Saxena and Dr. Dixit Saab knew that what kind of work we did. During my PhD stay at CDRI, I didn't work on medicinal chemistry. I work on non-covalent interaction, pi-pi stacking, these kind of molecules. So you can see this. Afterwards, because at that time, the job market and postdoc are very difficult to get, I landed up to a pharma industry in Lupin and Panchil Orni Limited at Indore. I work on a process chemistry. At that time, actually, I got fascinated towards medicinal chemistry and process chemistry because in lab, whatever you do, as small changes can make a huge difference in commercial scale. This is what I learned at industry. Then I went for a postdoc at Indiana University. There I worked 2 years on activation of this hexene diene So if you hit this, it forms a cyclohexene diene radical, and that radical cleaves the DNA. So it's a more kind of a experimental work. At Purdue 1 year stay, I worked on typical medicinal chemistry, that is farnesyl transferase inhibitors. But when I joined Naipur Mohali in 2002, one day my director called me, he said that he should work on malaria. Since I was PhD from CDRI, I knew about malaria. Dr. Chandan Singh groups and other groups used to work there. So there are two molecules if you want to work on malaria, that is chloroquine or artemisinin. So as a beginner, if you want to write a project on chloroquine, nobody will support you because they will say the molecule was discovered in 1941. What you will do in this now, right? In artemisinin, if I want to work on artemisinin, I have to compete with a group which are so established and they have everything, all the kind of facilities. So I started looking at the literature and I found this structure called tetraoxane. You can see the center part. You have 6 atoms, cyclohexene, 4 oxygens. So 4 oxygens are fixed, right? And only this carbon and this carbon is available for medicinal chemistry purpose. And this paper was published by Bunstrom in 1999, and I'm talking about in 2002. And in 3-4 years, only less than 100 molecules were reported. So the reason is it's very difficult to functionalize. So I thought, okay, let's start on this molecule because nobody is working, at least in India, and abroad also only one group was working. So we designed and submitted a proposal to CSIR, made this compound, first crystal structure, first paper on tetraoxane from India. But afterwards, I have to stop this because someone from my own department started working on this. I said, okay, stop it. But at that point of time, I thought, let's do work on hybrid molecules. In that, I thought take this part of chloroquine and these yellow part of cycloguanine and pyrimethamine, and make a hybrid molecule, something like this. This is a DHFR inhibitor. This is the hemojoin force. Different mechanism, make the hybrid. We did that. The initial studies, this is the triazine, and this is the aminoquinolate. This is the linker. What we proved here is this. This is the IC50 value for chloroquine, and this is the IC50 value for our compounds. Better than chloroquine. I mean, we started in 2008. At that time, Delhi University even do not have the NMR. Okay, and you have to collaborate for each and everything. But 2009 end, two research papers were published, one from Dr. P.M.S. Chauhan, another from a group from Argentina, wherein they prove, yes, this concept works. Luckily, the compounds were a bit different, so we could also publish a paper. As a medicinal chemist, this is what the criticism the medicinal chemists get from hardcore organic chemists. People say, तुम लोग chemistry तो तुमको कुछ आती नहीं है, और ये chemistry है क्या? आपने nitrogen को CH से replace कर दिया तो क्या होगा? So this is what we did. Small thing, change this nitrogen with CH and make this kind of molecules. So I will not go to chemistry. If you have a 2 Cls there, you have an opportunity to get these 2 regioisomers. We characterize this by X-ray, and the major isomer was used for further study. We introduced the carbocyclic amines. So in vitro anti-malarial activity data tells us you have a major, you have a minor. What is the difference? The attachment here, look at this position and this position. Activity is almost same. So that's a good thing actually. So we use this major isomer, make these kind of compounds. From 0.21 micromolar, we got to 7 nanomolar. So when you have a nanomolar activity, people will always talk to you about toxicity and in vivo activity data. So in vivo, it was very good, sorry. And without any tox, we published in 2012. This is where actually we started collaborating with Professor Kim at Harvard Medical School. When he read our paper, actually, he sent me an email one day. He said that he's interested to screen these compounds in the Parkinson model, what he has done. The motivation of this collaboration was this paper published in Sanders in 1987 in Nature, which concludes that chloroquine and related molecule can retard the progression of Parkinson's disease. And nobody followed that. And Professor Kim screened 960 FDA-approved drugs, found 3 hits. All these 3 hits has 4 aminoquinolone pharmacophore. So that was the perfect rationale to enter the collaboration. And in fact, in one of the meetings, he said, because Harvard Medical School and the Harvard Chemistry Department are about 3 miles away. So he said that the easiest thing to go to chemistry department, and he requested some of the people there. They agreed, but they said, we need $10 million as a research grant to make those compounds. So we have already in our kitty. So we said, okay, no problem. We don't need anything. We'll send compounds to you. So for the students' purpose, what is Parkinson? Parkinson is a neurodegenerative disease, number 1. Number 2, human brain has more than 60 billion neurons. Okay. And none of the neurons are connected to each other. They pass on a message through this space called synapse. So you can imagine how many synapses are there in our brain. One of the neuron is a dopamine neuron. What it does, it releases the dopamine in your brain and you are happy. And if somehow the dopamine production decreases, you feel all these kind of problems. That is called Parkinson's disease. And there is no drug for its treatment till now. And this is the graphical representation. This is a normal, healthy human being. You have a dopamine neuron. It generates the dopamine. And this is the protein called alpha-synuclein. This is how it looks like. But if you have a Parkinson's disease, the dopamine neuron dies. You don't have a dopamine neuron. You have a movement disorder. And during the same period, this alpha-synuclein misfolding takes place, forms lumps like this. So if you want to work on this project, you can think two things. One, stop the death of the dopamine neuron, or stop the aggregation of this. But what could be the target? Till 2015, people felt that there is one receptor called NER1 receptor. People were talking about it, and they said it's an orphan receptor. But in 2015, the crystal structure were determined, wherein the people feel that it's a bulky binding pocket, but there is a mystery molecule. When it binds with this NER1 activation takes place. As a result, the death of dopamine neuron is arrested. So if you can do that, it will be a drug for Parkinson. And this NER1 is essential for development and maintenance and protection of dopamine neuron, which are progressively lost in Parkinson patient. It boosts the activity of the genes involved in the dopamine production. So this is what it meant. And more than 10 million people across the globe are affected with Parkinson. And we do not have data here, but there is a recent study which says about 10% of these affected people belongs to our country. And in next decade, by the next decade, India will be on the top on the list of the Parkinson's disease patient. So with this background, we started collaborating with him, with Professor Kim. And this is the one set of molecules. This is the other set of molecules. These are totally inactive. And these 4 molecules showed little bit of activation, not too much. But we started functionalizing this molecule. So this was our hit molecule. So we worked around this with NH modification, this methyl, and this portion. And we synthesized about 700 molecules. And these are the regioisomers. The regioisomer purification is really, really challenging. And this was funded by MJ Foundation 2014 and 16. The initial studies, the chloroquine EC50 value is 50 micromolar. And our compound EC50 value is 2 to 3 nanomolar. In one case, it's a 0.5 nanomolar. I will concentrate on 2 compounds today. This is one, SBB94. And this is another one, 8ES399A. So actually what happens, we published this anti-malarial activity of these series of compounds in 2015 in Royal Society Advances, and my collaborator from University of Mississippi asked me to send a little bit more quantity. So I asked one of the students to make it. One day he came to me in the evening, he said in the 100 milligram scale reaction, he got a very polar compound and he isolated it, and he told me also. How much quantity? 8 milligram. No PhD student will come to you कि 8 मिलीग्राम मिला है तो उसको भी दिखा दें. अगर मान लीजिए वो बॉस ने कहा इसी को बनाना तो जिंदगी तो आपकी खराब होनी है, राइट? तो इसलिए इसको फेंक दो. That he was honest actually. He bought. So I said, well, let's characterize. We characterized that molecule. We found this is what the structure is. Then activity 0.5 nanomolar, the best compound among all the series by that time. And we got nature communication on that. Then the next modification was the nitrogen modification here. And we have many compounds less than 0.1 nanomolar. And from that series, we have more than 40 compounds, which EC50 value is in nanomolar range. Let me just show you some biological data. I'm not a biologist. थोड़ा सा जितना पढ़ा, जितना समझाया, उसी को समझाऊंगा. So this is luciferase enzyme assay using NER1 ligand bind domain. Okay, जो उसका binding domain है, उसी के साथ ऐसे किया गया. What we found, the chloroquine activates it at 100 micromolar concentration. Our compound 5 times better than chloroquine. This is the full length data. In full length, the same result. These red dots are the genes responsible for the dopamine neuron, red one. And when this is treated with this compound called MPP+, you can see here red dots depletes. कम हो गए रेड डॉट्स। उसी तरीके से एलपीएस से जब ट्रीट किया रेड डॉट कम हो गए, इसका मतलब डोपामिन न्यूरॉन की डेथ हो गई और पार्किंसन हो गया। Now, when this is treated with chloroquine, you can see little bit more red dots appears in both the cases. But when this is treated with our compound, you can see much more red dots. So what we observed there, more than 60% of the neurons were rescued by this. It is an in vitro data. Then the in vivo, this is the compound called MPP plus, which causes the Parkinson disease. This is the serotonin part of healthy mice, this one. When this is treated with this compound, Parkinson took place. And this is how the brain looks like. When this is treated with L-DOPA, no change. But when this is treated with our compound, you can compare the first and the last figure looks same. So what we proved here? We proved, yes, Parkinson can be cured and treated. This is one. Second one is, the mice was injected with alpha-synuclein. Alpha-synuclein, other agent which causes the Parkinson disease. And what we found, 5 milligram per kg body weight per day oral dose for 1 month rescued the mice. This is what it is. So this is the figure for alpha-synuclein induced mice brain. This is with chloroquine. This is with our compound. Look at the first and the fourth. Looks same. So it activates neuron receptor, stops the death of dopamine neuron. It stops the aggregation of alpha-synuclein. Third one, you have yellow dots and red dots. Okay. So under starvation condition, autophagy takes place, more red. When this is treated with this compound, it stops the autophagy, all the red dots are gone. But when this is treated with chloroquine, the dots are reduced to 22%, this one. But when this is treated with our compound, we found that 64% autophagy was introduced. Chloroquine is known to retard the autophagy. Autophagy सुना बच्चों ने? Autophagy word सुना आप लोगों ने? Autophagy में 2015 का नोबेल प्राइज मिला जापानीज साइंटिस्ट को. Autophagy is simple layman language. If you fast, क्या होता है? भूख लगती है ना, तो your cells will be in stress. तो कोई भी मरना नहीं चाहता, सेल भी नहीं मरना चाहता. तो सेल क्या करता है? सेल जो गार्बेज सेल है, जो डिजीज कॉजिंग सेल है, उसको खा करके जिंदा रहता है. So that's the process called autophagy. And what is the difference? The structure difference. This is the beauty of chemistry. Look at it here. This N-dimethyl group is substituted by this. This stops the autophagy. This induces the autophagy. And this molecule was approved by US FDA for phase 1 clinical trial in 2023. Professor Govardhan Mehta told me in a meeting that this is the first molecule from Indian academia which got the US FDA approval. Though I don't know whether it's correct or not. So these are the patents. And very recently, on November 25 to 2024, when all biopharma, Devon Pharmaceutical, Nervan Pharmaceutical, the Nervan was established based on our molecule by our collaborator in the United States. They announced that this molecule cleared the phase 1 human clinical trial. It says HL192, that's the code given by this company. The ATS399A is the code which we have given. It says was well tolerated with no major safety issues with any patient in 76 participants. No serious treatment adverse events were reported, and such findings were proportional between placebo and dose groups, and PK results supports once a day dosing. So that's the structure. So this result was published in 2023 in Nature Communication. And this is now we are working on a second generation of molecule right now. And this is the group of students who made this story success actually. Rohit Kholia is assistant professor at Deshbandhu now, and he is general manager at SRF. He is in Bangalore in the industry. Sunny Manohar is with the Deen Dayal Upadhyay College. He is in BSF. Gagandeep is Hansraj College. He is in Kirolimal College, and she is working right now. Not on one molecule actually. Now this molecule has cleared the phase 1 clinical trial. These two molecules are the preclinical development. One is for autoimmune disease. Another one is for dementia. And this is the Hanall Biopharma website where they reported about our compound has cleared the phase 1 clinical trial, and phase 2 clinical trial will start very soon. As Dr. Mehta, the Saxena has explained you what these trials are. The story was covered by Bloomberg, Wall Street Journal, other electronic and print media abroad, and one is in a Korean biomedical review also. So this is important for the younger generation people. How smooth journey is, क्योंकि हम जब आते हैं तो बड़ी अच्छी अच्छी बातें करते हैं, फेलियर क्या था तो कोई बताया नहीं, मेन तो फेलियर है. So in 2012 I wrote a project. I will not name the funding agency, and till now luckily कोई भी project reject नहीं हुआ, but मेरे को एक HPLC भी नहीं मिली अभी तक. जब भी मैं HPLC भी लिखा उसको काट करके पैसा दिया. And sanction letter के 32 lakhs maximum grant I got from Government of India till now. It says actually the PI must change the routine area of work. Sanction letter का part बनाया है. जितना मैं समझता हूँ उसके हिसाब से आप फंड कर ही नहीं सकते थे, but this is there. का HPLC अभी तक नहीं हमारे पास. Delhi University took 4 months to give me permission to file Indian patent in 2012. With a rider, you make payment from your own pocket. Project progress report. Excellent, very good, good, satisfactory. I got good in this project when I had one US patent at that time. Delhi University took 9 months to sign MOU. MOU था, all patent filing cost will be borne out by them. Royalty अगर होगी 50-50% होगा, 9 months. Technology transfer, 16 months. Now, 86 lakh grant I got from this company now, just before moving to Nainital. They said, no, अब तो आप छुट्टी पे जा रहे हैं, आप नहीं कर सकते. तो I said, should I return it? So I was given permission, okay, तुम use कर सकते हो, but you can't take PhD student to a person जिसका molecule second phase clinical trial. Moral of the story is, keep yourself motivated. Let me just show you 2 slides more. Not this one. Okay, this one. ये जब नैनीताल गया उसके बाद का काम. So reaction of this, making this cis alkenes using diphosphine oxide. Diphosphine oxide अभी तक known है alkene and alkynes में addition reaction. हमने प्रूव किया ये हैलोजन एटम ट्रांसफर करके आप इस तरीके से alkenes को cis बना सकते हैं. We submitted this JAX actually. As happens, we know that JAX ने editor के level पे ही वापस कर दिया. Published in Orne Letters. Look at this paper now. Compare these two. What is the difference? This part and this part. And this is well known for halogen atom transfer. 7-8 months later, this paper was published by somebody in JAX. Okay, the last one. And this is the fundamental. This excites me much more actually. Class 2 की chemistry सभी ने पढ़ी होगी ना? अगर इस मॉलिक्यूल का resonating structure लिखने के लिए कहा जाए, हर कोई ये structure लिखेगा. Okay. With this deuteronining form structure, this becomes a nucleophilic center. तो इलेक्ट्रोफाइल यहाँ अटैक करेगा. इलेक्ट्रोफाइल अटैक करके बहुत सारे साइक्लेशन के कितने पेपर पब्लिश हुए? More than 1550 research papers have been published on this. One day, when my student came to me, he showed me one of the papers. He said, "Sir, ये ऐसा है, इसको हम ऐसा करके ये कर सकते हैं." I said, "Okay, why not this structure? इसका resonating structure तो ये भी बन सकता था. तो why not?" मेडिसिनल केमिस्ट्री में काम करते हुए एक्चुअली 2 चीजों में बहुत डर लगा. एक स्टीरियोकेमिस्ट्री से, तो हम स्टीरियोकेमिस्ट्री बहुत दूर रहते हैं ताकि कॉल को कोई प्रॉब्लम हो. दूसरा रेडिकल पता नहीं कहाँ जाके अटक जाएगा, कोई पता नहीं. तो भी नेवर वर्क. But when I talk to him, then I realize it is a diphenyl methyl radical. Diphenyl methyl radical are not room temperature stable as per textbooks and literature. So जब room temperature stable का छोड़ो, मैंने उसको इतना बोला कि ये ऐसा हो सकता है. But this boy was very clever. Actually. He went to lab, put up a control reaction, heat it up, he got dimer. इस इंटरमीडिएट से डायमर बन नहीं सकता. डायमर तभी बनेगा जब आपका रेडिकल. was very happy. He showed me, sir, ये देखिए मैंने डायमर बना दिया. एक्सरे स्ट्रक्चर. सर तो रेडिकल केमिस्ट्री में सिंपलेस्ट एक्सपेरिमेंट इज टेम्पो. We did reaction with tempo. We confirmed. Yes, it is. Then I said, यार मैकेनिज्म प्रूव करने के लिए क्या ये हो सकता है हम ईपीआर करें, किसी तरीके से लो टेम्परेचर ईपीआर करें. He took that idea. To our utter surprise, this is room temperature stable free radical. First report. Now, with this, what we can do, we can reverse the reactivity. यहाँ न्यूक्लियोफाइल अटैक करा था, हम इलेक्ट्रोफाइल कर सकते हैं, जो आप तक एक्सप्लोर नहीं हुआ. इस पोजीशन पर आप बीआर किसी भी मेथड से नहीं लगा सकते. We put BR here. Not only that, we could make triphenyl or diphenyl methanes. Not only that, triphenyl ethanes. And this one molecule is in sigma aldris. 50 milligram cost 18,000 rupees. We can make it in single step. इसको रिएक्शन करने के लिए इलेक्ट्रॉन रिच एल्कीन्स और एल्काइन्स चाहिए होते हैं. We said no, we don't need to that. We can react with electron deficient alkyne now. We can make jet alkenes. This jet alkenes can be converted to this kind of molecule. एक कार्बन में 2 phenyl ring in one step. It's a gold mine. Not only that, you can make a tetra substrate jet alkenes. Jet alkene बनाना सबसे difficult काम. Cis trans तो बहुत ज़्यादा बनता है, cis बनाना बहुत मुश्किल है. We can do that. We can make a disulfide bond with cis stereochemistry. So with this, I thank each one of you. In between, 2012 से स्टोरी शुरू की थी, अभी तक खत्म नहीं हुई है. तो PhD स्टूडेंट कैसे रुकेंगे? So we thought, let's work on some nanocatalysis, which gives motivation to the student in the form of the publication. We work on this, published many papers on that area also. So the funding came from all these funding agencies, and malaria work was collaborated with Dr. Sabana Khan, and the Parkinson work was with McLean Hospital with Kong Sho Kim. And these are the bunch of students who made this journey possible. All of them are well settled at their respective places now. And this is the another view of Nainital. Thank you very much for your kind attention.
Any questions?
क्वेश्चन नहीं पूछने का मतलब दो होते हैं एक्चुअली में. एक, सब कुछ समझ में आ गया, और सब कुछ. सर प्लीज. Yeah. Yeah. Yes, for cure actually. Yes. Mm. No, this is what we showed actually experimentally. It regenerates up to 60%. Up to 6 months. This is what we have to do. Yes. Yes. Mhm. Okay, no. In this portion, it has nothing to do with biology. It is a fundamental chemistry problem. Okay, and one of the molecules, there are many molecules actually, but one of them is in the sigma aldrich, which is sold as a fine chemical. Okay, so it's a chemistry part. No biology, the last. Yeah. Yes. No, no, no. No. The triphenyl one is the last slide, right? So last slide, the quinine methide one, that is more of a chemistry problem. Till now, people thought कि ये nucleophilic ही reaction दे सकता. Now we proved कि electrophilic भी देता है. तो उसमें biology की बात नहीं कर रहे last वाले. जो हमारा main work है वो aminoquinoline and pyrimidine through link. तो उसमें वो problem. Yeah. Yes. 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 Yeah. 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 So to answer your question on anti-cancer, I've not shown because I don't have this much of time. When we made the intermediate, जहाँ पर आपको कार्बोसाइक्लिक अमीन नहीं लगाया, that molecule induced the cancer. Okay, this is what the beauty of chemistry, right? And जैसे ही Cl को substitute किया कार्बोसाइक्लिक अमीन से, it kills. Okay, then the second कार्बोसाइक्लिक अमीन लगाया, that inhibits. We have that figure also. We have not published that data.
Okay, thank you,
Yes,
Dr.
please.
Rawat. Okay, go ahead. Aruni.
Yeah, your question is well taken. Yes. Let me answer this question. जो ऑर्गेनिक कंपाउंड्स जो होते हैं वो वाटर सॉल्युबल होगा ही नहीं. 99% आपके वाटर इंसॉल्युबल होगा. हमारे सिस्टम में वाटर के अलावा कुछ दूसरा नहीं, राइट? So this is why when the in vitro assays are done, these are done in a diluted DMSO solution. Okay, diluted. But if you have a better activity, if you want to go to animal models, at that time the chemist role starts. To that selected molecules, we make a salt. These molecules are highly insoluble in water. Water में डालेंगे कुछ नहीं होना इनका. But we made a salt. The salt बनाते वक्त भी आपको बहुत सारी चीजें ध्यान रखना होगा as a chemist. Okay, so when we make a salt, then we make them water soluble. So the molecule now which is being developed, it's a water soluble as a salt. Yeah. Okay, no. In phase one, no. Okay, in phase one, these are the healthy volunteers, age group 18 to 78 or so. Okay, it is the old age group. And in this case, the Parkinson disease patient of different age groups, not one. This is what they have planned.
Okay,
We have in our kitty maybe 3,000, more than 3,000 molecules there. But real जिसको आंखों से देख सकते हो. And we characterize them. Okay, but yes, most of the molecules are published. But जो पार्किंसन वाला है उसमें क्या हुआ कि जब एक्टिविटी अच्छी आई, मलेरिया वाला पब्लिश किया क्योंकि बच्चा कम से कम रहेगा, रुका रहेगा, एंड पार्किंसन की जो एक्टिविटी अच्छी थी उनको हमने असाइड कर दिया. So when we publish this nature communication, it has 500 compound system. It's 500.
okay. Dr. Rawat, thank you very much.
Thank you.
After this wonderful presentation by Dr. Rawat, the next presentation is by Dr. Prakhar Saraswat. And may I request to Dr. Saraswat? Prakhar Saraswat holds MBA, MTech, and BTech degree and is a dynamic, result-driven professional with 13 years experience. So can I request him to come here from IT industry? He has excelled in delivery and stakeholder management, leadership roles. They're very important. So, you see. So, Dr. Prakhar.
Am I audible? Okay, good morning, everyone. I hope you all are energized and not feeling sleepy after the breakfast and after some, intense lectures. First of all, thanks to the chairpersons and the young minds, I will say. Their minds are more sharper than us for most of us and very energized. So thanks to them. Before we move forward, let me first say thank you to the organizing committee. They have put a lot of efforts. That's the reason we are sitting here, and it's been organized with we have some technical challenge that's ifs and buts and part and parcel for any conference and any sessions. But kudos to you guys. They have worked well under the guidance of Dr. Anil Kumar Saxena and Dr. Aruni Saxena. And I wish them congratulations and success for upcoming years to make this at a very larger scale, what we are doing currently, right? And another set of, thanks, which I want to say is to my parents, because they have made sure that I have been taught the importance of education. And another set of parents are here in form of my in-laws here. So thanks to them as well. Okay, so today, a lot of you, after listening to my introduction, will be thinking that what this kind of person is doing on the stage in this conference, right? So on the lighter note, let me answer you. I'm just here with all due respect. I am here to save you from some molecular structure for the next 15-20 minutes, but after that, I can't confirm. Okay. So, and also, if I fail in that, the committee people have made sure that you guys have a break after this, my session, so that you guys can have one dosage of tea or coffee, which might cure you. And if it is severe, then you guys can take 2 of them. Right? Okay, jumping into the topic. So I am basically coming from IT service-based company, and being an operations head and working in SAP from last 13 to 14 years. So SAP is, so I'm assuming that, okay, let's, for the benefit of doubt, let me tell you that what SAP is at a very high level. It's a ERP system which helps the large and medium organization in terms of resource planning. Okay, when I say resource planning, it is enterprise resource planning, and it is not subjected to any domain, any industry. It is widely used in many industry. And since it is a conference which is related to pharma and medical sciences, I chose that how SCP helps in that industry, right? So we'll be talking about, if you can you please put the second slide? So we'll be talking, next slide, please. Yeah. Okay. So yes, we will be talking about the counterfeit threat in pharmaceutical. We have, I mean, a lot of people, anyways, you guys are from that industry, will be knowing it. That's a big problem. It has been identified. And I will be talking about how SCP has few solutions which can help in making sure that we don't have, that substandard or falsified medicines which are available in market. So just wanted to make sure that how SCP will be having some solutions, and they have been implemented. It's not only the solutions which we are talking about on the paper. They have been implemented in many companies, and they're taking the benefit of it. So if you see, 1 in 10 medical products, okay, medical products in low and medical economic countries are substandard or falsified. So that's alarming number, right? In every 10, one is either substandard or it has been falsified. And this is according to the WHO, right? And the counterfeit drug market is, if you see the number, it is mind-boggling. It is 200 to 431 billion annually. It's a huge number, if you see. And this is not only, anyways, it's a worldwide number, but it's growing. It is not stopping at that number. So it's alarming as well, right? And the scale of the problem, see, this is the scale. This is a large scale. And if you see that over 50% of the FDA warning letters are also cited, saying that the falsification is there. And the biggest example is Ranbaxy case. In, I think 2013, it was identified. And this is the biggest amount of penalty which was issued to such a company during this and in the industry of almost 500 million. Fine was asked for Ranbaxy to pay, and they agreed and they paid for it. Next slide. So that was the problem. Okay, that problem statement is there. And now in SAP, we talk about 3 solutions which are interlinked. Also, before we jump into this, I think yesterday, respected sir was talking about it, that it's a problem which humankind is facing, and it is not one single industry can help. It's an ecosystem. And if you fit all the pieces, different pieces, all the industry should join their hands, and then only it can be solved. Okay. So how this technology or how IT service play a role, we are just trying to say that, okay, we can also help in this. So there is one of the solutions which we talk about which says that it's an advanced track and trace for pharmaceuticals. It's a ATP, which we call it as acronym. And please, when we're talking, there's a lot of things which will be new for you. Don't get overwhelmed. It is no rocket science. Okay. I'm just trying to explain it very layman language as much as possible. And I will make sure that my contact details are given for you guys. So if you guys have questions and you want to learn more or understand more, I'm available, and I will make sure that I will answer those after the session as well. Okay. Okay. So coming to the first one. So this is, so the problem has been addressed is that the serialization, the numbering is the problem, right? So in this system, what we do is that end-to-end serialization has been there. It is possible in the system, and track and trace compliance is also there with the global regulations. So it has been from the FDA. It has been global regularized also. So that serialization is one issue which you can solve it. We also make sure that when unique serial numbers are given, and to prevent the counterfeit products from entering the supply chain. So if it doesn't enter the supply chain, it will actually not be as an end product to the customers or the consumers. So that's the idea, that if we solve it at the first level from point A, it should not reach the point B. So that's the idea behind it. Okay. And it also and it is not beyond the regulations or the standards. We make sure that the standards and everything has been followed and agreed upon, and then only the process has been taken into consideration. Can you go to the next slide? Okay, another one solution which, and all the three solutions are interlinked. I will also explain step by step how at what stage what solution comes into picture in upcoming slides. But as of now, I'm just trying to explain that what is the individual solution brings to the table. Another one is ICH, SAP ICH, and this is again all cloud-based in network for securing the serialization data exchange. Once we have it, we also make sure that it is not temporized, right? If you are tampering it, and if the loophole is found, again the problem is not solved. And it also verifies the authenticity at every stage of the supply chain. It is very important because from point A to point B, there will be many other points as well, and where you guys can fail, and then still you will not met the outcome of it. As I also told that it is also in compliance with the FDA, EMA, and WHO also, and other regulatory agencies as well. Next slide, please. Okay. So this is the slide which I was talking about. If you see at stage 1, it is the manufacturers. So how system can help us is that it assign a unique serial number, and the data has been uploaded to ICH, which was presented in the previous slide. Next is when it goes to the wholesale and distributor. You can scan it, which validates the serial number which was assigned, a unique serial number, before distributing the products. So after that only it will be going to the distributors. Now, once it reaches the pharmacy or retailer, it will check the serial numbers. Again, the unique serial numbers will be there upon receiving the stocks and before selling to the customers. Now, if you wonder that how this unique serial numbers can be traced, it's all can be done and can be authenticated from the mobile apps, or there are QR codes which you can scan it and it will tell you whether it is authentic or not. And this is no rocket science, because this is not only the problem which medical industry is facing. Even if you go to cloth industry or everything, you will have a lot of fake clothes and everything where people say, okay, there is a scanner. If you go to any branded retail shops for the clothing as well, you will have scanners. If you scan it, it will take you their own website. That's how the authenticating idea works into it. You will have a unique serial number, the barcoding is there. But having said that, I mean, there are loopholes in that also. I mean, to be very, very honest, there are people who will copy the serial numbers and still put it on different products also. That is also there. But to counter that, there is only one solution as of today available in the market, that is blockchain. Using the blockchain is the only way you can actually fix this problem. Because let's say that I'm sure that few of you will knowing there is a very peculiar fish called tuna fish. Okay, and it is so peculiar that there has been a bidding also in Japan also. That for every year that a bidding starts when the first fish is bidded, and you will be baffled to know that the first fish at auction sold for 40 crores. Okay, first fish, because they believe there is some sentiments behind it. But for that also, you will know there is a serial code, there is a coding for behind it. You will know from which ocean, when it was picked, when it was refrigerated, and the process goes on. So you can actually find out from where your fish is coming, right? So, but since it is peculiar, you can't identify for all the local fishes, but the logic remains the same behind all of it. Now, after this pharmacy retailer, the regular authorities are there. They can also access SAP ICH to audit and verify the compliance are met or not. So this is very important. And sir was also telling that one way is that we have our regulations and everything is in place. But the other thing is we should implement, and second thing is to control that, and to also validate it, whether these guys are, and to audit also whether every regulations and every compliance has been followed. Now, and at the end, the last stage is hospitals and patients. When the medicine is coming to hospitals and the patients and hospitals, even like it is not only for us, I mean, for the bodies to, authenticate that. As a patient also, you can also scan these codes and serial numbers to check whether it has been a falsified drug or is it an authentic one. Okay. Next slide. Okay. Now, another problem statement is track and trace. So another solution which is very important is which uses your real-time shipment. Now, there are a lot of medicines which has to come from through a transport. Transport mode can be your road, your railways, and your air, right? And with medicines and for others industries also, one of the thing is that temperature control is very important. Few medicines are coming, they have to be in certain temperature and all, which can be also controlled from IoT. Not even controlled, that even let's say that there is a malfunction in the software and the temperature is not set right, it will actually authenticate and send a reminder or an email to the person. Let's say that some manager is there involved in this, who should know whether something has been wrong or right, right? It will say, sense it that, okay, temperature is falling due to this. It will actually send a reminder and an email in real time saying that something has been wrong and we have to fix this. So you will know that after it. So because if it has been not notified and it is coming to your end product and it is not been in the right temperature, there is no point of it. You actually have not solved the problem. So IoT helps in that. And even as I said, blockchain also helps in the authentication and the serial number of it. And the second line, anyways, I have talked about it, that it detects the temperature and exertion and tampering of the ensuring the drug efficacy is met. Yeah. Next slide. The impact. So, I mean, bringing all these solutions together, what is the impact we are trying to create, right? So, one is that up to 90% reduction in the counterfeit drugs in the high risk market, because it's a, kind of a mafia which is running. I'm not sure if I mean, we can call that, but everyone knows behind the scene that this is happening, and either it is fatal for this humankind, and second is either the fake medicines are there, which will actually eventually not help you. I mean, you're taking the medicine, but you're not getting cured. Then what is the point of it, right? Other thing, impact which we can bring it after all this compliance and audit is that 40% fewer FDA warnings letters will be issued. Now, this is important. I mean, as I said, the Ranbaxy case, right? There is 50 crores or 40 crores were implemented on them for this letter, and they have to pay the fine. So that reduction can also happen from this. Then 30% lower regulatory reporting cost due to the automation. If you automate a lot of process in terms of transport, instead of real-time tracking, it will eventually help in automated. Okay. Now 35% in reduction in supply chain distribution, companies now using the SAP solutions. Next. Okay, so this is one of the app which we have implemented in our company using one of the APIs, and it is integrated with your transport management, and it helps the track and trace. We have named it as Navika. I'll just take quickly 1 minute just to explain that what exactly we are trying to do here. You guys would have noticed one is track and trace, which is very okay, that it will send the reminders and where is your truck and everything. That is very basic. One thing is optimization. Now, let's say the truck has to go and pick certain supplies from a vendor, but this app helps to optimize this. It will tell you which route to take, okay, and also will tell you that which truck should go. There might be multiple routes to reach from point A to point B, but this will eventually help you optimize it and tell you which truck should take it and which route to take it, which will save you money even in terms of your manpower, also in your fuel. So it has to be also considered when we're talking about large organization, pharma industries are also there, right? So eventually it will save cost in implementing such applications. Next. So I think I have saved you time, and I'm open for questions. Thank you for your patience. Thank you very much. Thank you very much for wonderful.
And I think today automation, AI, digital platform, they have become the very important tool for any industry, and particularly for pharma industry, where we have to take care not only the transport of the medicine, but also the transport which is as per SOP. And for the information of the house, it was estimated that in India, about 15 to 16% of the drug is lost because of the poor transport arrangement and storage arrangement. We have now it is mandatory that when you transport from one warehouse to another warehouse the drug, you have to have data logger for temperature, humidity, and the variation in the temperature. So that is very critical also. And also it is also important, particularly for vaccine and biologics and also other drugs, that we have very rigid temperature control in a storage. So far, so much so that if you remember, during COVID time, when we have amphotericin B, liposomal amphotericin very heat labile. And if liposomal amphotericin B is not stored properly in temperature controlled manner, then not only that it will be ineffective, it will get precipitated, it will become toxic. Particularly, the entire liposomal activity will be lost. So this is something which is very important and often neglected, but a lot of effort is now being put into this. So this type of automation will be very useful, I think. Awareness is very important. And in India, I do not know whether the people sitting on the young, he said there's a young, though he is also very young, we also feel young, but he said the younger generation sitting outside, whether you are attentively listening to this or not, I see some of them going and coming, such an important talk. Just let me know, how many of you know what do you mean by cool temperature, cold temperature, room temperature? Are you aware of this? Often it is written in the, please keep in the cool temperature, keep in the humid. कहीं लिखा होता है इसमें चार लगा होता है, ये मतलब क्या है इसका? पता ही नहीं है लोगों को, 90% लोग, आपको भी शायद ही पता हो. What do you mean by cool temperature? What do you mean by cold temperature? What do you mean by refrigerated? What do you mean by deep refrigerated? So storage condition, that's why we have added one chapter, how to storage condition in the national formulary. तो ये तो बड़ा जरूरी है. यहाँ पे आप लोग बैठ के इतनी इम्पोर्टेंट टॉक्स को सुन के अगर आप ये ज्ञान प्राप्त नहीं कर सकते, आपके दिमाग में स्टिमुलेशन नहीं रहेगा, तो फिर फायदा क्या है? Just listen how what is the so important. See, just COVID में वैक्सीन ट्रांसपोर्टेशन के लिए जो ट्रक्स इस्तेमाल हुए थे, उनके छत के ऊपर there was a specific type of coating was done so that the temperature can be reduced to 3 degrees effectively. And 3-degree reduction in temperature was so effective that then the refrigeration was easier. And you know that when the vaccines are given, and अगर वैक्सीन हमारा वहाँ पे आधे रास्ते में खराब हो जाए, फ्रिज खराब हो जाए, then what is the backup? And अगर वैक्सीन का डेटा लॉगिंग सिस्टम से टेम्परेचर हमारा 5 घंटे तक लाइट नहीं है, तो कितनी वैक्सीन का डैमेज होगा? There are many times we say that keep this in a cool temperature, room temperature, when this drug is kept in open temperature, where the temperature is 45 degrees centigrade, what is the meaning of that? So ये चीजें बड़ी important है. I think which he has highlighted how much the importance of digitalization and locking system, and that's what the entire system is. Good transportation system practices, good transportation, good storage practices, good transportation practices. This is something very important. Otherwise, you may lose the license of your pharmacy. So any comment which you would like to have? Comment or question? I would like to have a comment or question from the people sitting after 4th bench. या तो सो रहे हो, अगर सो रहे हो तो इतना नाश्ता क्यों किया था? और अगर सो नहीं रहे तो कुछ तो दिमाग में से सवाल पूछो ना. तो ये वर्कशॉप क्यों अटेंड करने आए? एक तो सवाल पूछो, कम से कम कमेंट करो यार, व्हाट इनोवेशन यू कैन डू। अरे इनोवेशन तो उसी एज से ग्रुप से आता है ना ज्यादा, जहाँ फर्टाइल दिमाग होता है। यस।
So SAP is a company which helps the medium and large organization, okay, for enterprise resource planning. It's a company, but the acronym says as System, Application and Products. So it's a German-based company, okay, IT company. And as a respected sir is saying, I mean, you should ask the question. See, the question is not right or wrong. So if you're feeling that, okay, if it is a right question or not, you should just ask it. Okay, if you have any thoughts about it. And sir was also telling that, I mean, it's very important for you guys to step forward. And if you're not asking that, I mean, then you're, not learning, right? बिल्कुल.
आपने कहा कि सिर्फ है कि आप वगैरह एक पर्सन एक तक को उसमें 1000
Absolutely. Yes. That's what I'm telling you. So, I mean, even if you see, and this has been used maximum times or majority in fashion industry. आप कोई भी क्लोथिंग ले लें, हाँ, नहीं, इसमें आपको ज्यादा मिलेगा। आपको मतलब एक फेकनेस होती है जिसमें नाम, मतलब जिसमें अकल में, मतलब नकल में अकल की जरूरत होती है ना, तो वो प्यूमा को कुछ भी लिख देंगे, उपमा लिख देंगे, समथिंग दैट इज डिफरेंट। बट अगर आपको बार कोडिंग से लेके सीरियल नंबर तक एग्जैक्टली मैच मिलेगा, एंड ऑफ कोर्स if once you copy it, eventually it will, when you track it, it will take you to the website, nothing wrong in it. तो उसको करने के लिए से ब्लॉकचेन यूज कर सकते हैं, क्योंकि वो रियल टाइम ऑथेंटिकेट करता है। इट यू विल, उसको आप कॉपी नहीं कर पाओगे, क्योंकि वो एक तरीका है जिसको डायनेमिक रख सकते हो आप। वंस यू मेक इट डायनेमिक, इट कीप्स चेंजिंग, तो इट विल बी वेरी इजी, एंड ओनली सर्टेन पीपल एंड सर्टेन रेगुलेशन बॉडी विल नो कंट्रोल ओवर इट, राइट? तो यू, दे विल नॉट नो कि कब चेंज हो रहा है, तो यू कैन एक्चुअली हेल्प विद दैट। तो एज आई सेड कि एज ऑफ नाउ सिर्फ ब्लॉकचेन एक सोल्यूशन है कि जिसको ये हेल्प कर सकता है। अदरवाइज दैट इज द इजीएस्ट थिंग। एक दवाई ओरिजिनली, उसका बारकोड सीरियल नंबर कॉपी किया, सबके चिपका दिया, तो यू विल नेवर कम टू नो।
प्राइम मिनिस्टर साहब ने कहा था ब्लॉकचेन लोग इस्तेमाल करें, पर
जो इंटीग्रेटेड है, यस, बट अगर सभी से कर देंगे, देन द प्रॉब्लम इज अगेन द सेम कि कंट्रोल नहीं रहेगा, राइट? तो उसको ऑडिट नहीं कर पाएंगे, एंड देन द, इफ दैट डेटा इज द क्रस्ट ऑफ इट, सो इफ इट इज अपलोडेड एट क्लाउड, then only you can control. If it is going beyond your ecosystem, you don't have any control.
there will be no tea break, there will be one more lecture after that, please. अच्छा, अब इसके बाद टी ब्रेक होने वाला था, लेकिन टी ब्रेक पोस्टपोन हो गया। थैंक यू वेरी मच, and my suggestion to those sitting on the back side, at least आप लोगों को 2-3 चीजें तो देखो मैसेज आज प्रैक्टिकल में करना चाहिए। अब मुझे ये बताओ कि काउंटरफीट मेडिसिन, स्पूरियस मेडिसिन, सब स्टैंडर्ड मेडिसिन, NSP medicine, not a standard quality medicine, is a major problem in India. So what can be the digital method for this exercise for you? So now we go to the next presentation by Professor Valdemir Verokko, and his talk is on machine learning application for detection of potential hero protector in the chemical or pharmaceutical universe. So I think this is something very important when we talk about learning and AI and learning. And may I request you to talk and listen to talk carefully. I will ask on his behalf questions from those sitting in the last 6 bench. You can take the opportunity to come front
And if I ask question and if you are not able to answer, I will presume that you have not written a single word of this talk. Then I will request the organizer to withhold your certificate of attendance. Sir, over to you. Dr. Valdemir,
Yes.
please, stage is yours.
Okay.
Is the system working? Can he start his
Do
talk?
you see my screen? Sorry?
Yes, you can start. We can see you.
Yes,
You
and,
can start.
and what about my screen? Full screen, do you see it? Sorry?
Please share your screen.
Sorry, I cannot see my screen. Okay,
Okay.
now you
Now
see
we
it.
have it.
Good. Thank you very much. Yeah, first of all, I would like to thank the organizer to giving me the opportunity to present some results of our studies at this famous conference. Second, I would like to say some greetings from Dmitry Druzhilovsky, who took part in this conference in the past year. Unfortunately, in this year, I could not come personally, so I just presented online. And the title of my talk is Machine Learning Application for Detection of Potential geroprotectors in the chemical pharmacological universe. The problem of the aging population is a big deal for the whole society, big issue. You can see from this slide that all countries and all regions of the world have aging population which is growing constantly. And of course, with the aging, the increase of age-associated diseases is observed. And you can see on this slide that cardiovascular diseases, oncological disorders or cancer, type 2 diabetes, dementia, musculoskeletal disorders, etc. increase with the aging. And people aged over 65 years old have at least one chronic disease, 54%, have frailty, 66%, rate their health as good only 25%, and have a disability primarily due to the age-associated diseases about 20%. And when we see the country ranking by the age of one set of age-related disease burden, you can see that India and Russia are approximately at the same situation. The age 60 is the threshold for increase of age-related disease burden. And World Health Organizations emphasized on the key facts that all countries face major challenges to ensure that their health and social systems are ready to make the most of this demographic shift. In 2050, 80% of all the people will be living in low and middle income countries. The pace of population aging is much faster than in the past. In 2020, the number of people aged 60 years and older outnumbered children younger than 5 years. And between 2015 and 2050, the proportion of the world's population over 60 years will nearly double from 12 to 22%. And of course, we are living to achieve the healthy aging, which will provide a significant impact of the population. And United Nations announced that currently we are in decade of healthy aging and create the appropriate platform, which you could see here. And the major determinants of aging and well are considered as diet, as physical activity, community engagement, etc. And these are the most important factors which are influence on the healthy aging. And of course, very important is financial security as well. But certainly, the people are looking for some magic pills, for magic medicine, for healthy aging. And you can see many papers in the internet and many different drug-like compounds like resveratrol, vitamin D, and currently Ozempic as considered as a magic pill for aging. And some people like Dr. Nir Barzilai are promoting the ways to use higher health span increase by geroprotectors. And metformin, rapamycin, quercetin, zatinib, and others are yet unidentified anti-aging drugs that could all be part of the picture. And of course, since we are in the Indian conference, we can see the impact of Ayurvedic medicine in healthy longevity, for instance, in improvement the skin conditions and some other issues which could be overcome by application of complex pharmaceutical substances provided by Ayurvedic medicine. When we are looking to PubMed and try to find how many papers are published just dealing with geroprotectors of anti-aging medicine, you can see the significant increase and permanent increase in the years. And of course, what is geroprotectors? It's not so simple definition for this subject. Dr. Moskalyov with colleagues actually formulated the criteria for geroprotectors, so-called primary and secondary criteria. Primary criteria include extension of life in experiments with wild-type animal models. The geroprotector should prolong the life of the model beyond the intact maximum lifespan by protecting it from one or more mechanisms of aging. Improvement in molecular, cellular, and physiological biomarkers of a younger state or a reduction in the progression of age-related changes in humans. Most potential geroprotectors are preventive only when applied at relatively high concentrations. The lifespan extended dose should be several orders of magnitude less than the toxic dose. Minimal side effects at the therapeutic dose during chronic application. The potential benefit of taking a geroprotector may come after a long period. Thus, potential geroprotectors should initially improve some parameters of health-related quality of life, such as physical, mental, emotional, or social functioning of the person. The secondary criteria include the target of mechanism of action of the geroprotector that extends the lifespan of the model organism should be evolutionarily conserved. Reproducibility of geroprotective effects on different model organisms increases the possibility that effects will also be discovered in humans, even in the absence of a known conserved target. Candidate geroprotectors should be able to delay the progression of one of several age-associated diseases in human, and of course, potential geroprotectors should increase organismal resistance to unfavorable environmental factors. And we are understanding that to study geroprotectors, we should have at least some reference drugs which are considered as potential geroprotectors because they do not do not correspond to all the criteria that were discussed above. And some list of 16 reference drugs which can be used as potential geroprotectors are given on this slide. They include metformin, rapamycin, resveratrol, curcumin, etc. In the current age, machine learning and artificial intelligence methods are used for finding of geroprotectors. Some actually publications are presented in this slide. And when we are talking about machine learning or artificial intelligence, you can understand and keep in mind that it's something like the QSAR, quantitative structure activity relationships. And to apply machine learning methods, you should have some structure descriptions, activity presentation, either in active as inactive or by real values, so it's present either in qualitative or quantitative mode. Of course, you should have some mathematical methods for classification or regression or some other qualitative models. And other essential issues here are training set preparation, model validation, and applicability domain determination. And in my team, for more than 30 years, we are developing the computer program PAS, prediction of activity spectra for substances, and started in 92. We moved from 9 kinds of biologically active compounds in the training set and about 100 predicted activities to more than 1,008,000 activities and have many web services which can be predicted now by PAS. And you can see that PAS is based on strong theoretical foundations, including local correspondence concept, novel structural descriptors, original classifiers based on Bayes approach, and high-quality training set, and of course, knowledge-based structure mechanism effects. And also you can see some statistics of use of our web services. And the most is Indian users which actually performed more than 400,000 predictions. The second are Russian and Chinese and Brazil, etc. This is just statistics for the BRICS countries. Also, to be able to use PAS for prediction geroprotectors, we have to investigate and create the training set. There are several databases which aggregated information about geroprotectors via internet. One is geroprotectors database, which is the earliest one, which includes more than 2,000 potential geroprotectors. And actually, this and the second database, DrugH, reflects the interactions of potential geroprotectors with model organisms like C. elegans, Drosophila melanogaster, mice, etc. And DrugH actually includes almost all drugs from geroprotectors database. The other databases that I would like to mention here is anti-aging peptides database, which were created by Indian colleagues. Also, Aging Atlas database created by Chinese colleagues. And of course, there is also a database on pharmacological trial targeting aging. And all this data could be used for creation of the training set. However, before you create the training set, you should curate both chemical and biological data. And when we look onto the drug age database, you can see that there is not so many small molecules, and a lot of them are just others. And also in the molecules which are presented in this database, there are items which really found in the pharmaceutical substances, including something like lithium, beryllium, aluminium, hydrargium, etc. And as a result of our study, we were able to reconstruct about 800 small molecule structures by the name. When we are looking to the distribution of lifespan values changing for model organism, you can see that it's not normal distribution, and this significantly differ between different model organisms. So actually, in preparation of the training set based on the drug age database, we recovered chemical structures and included only those compounds in the training set for which it was possible to unambiguously identify the chemical component associated with activity. And of course, changes in lifespan values were considered in the training set for each model organism separately. Lifespan change values were averaged if they related to the same chemical and were measured for the same model organism. Lifespan change values were categorized as having geroprotective activity or not. A lifespan change value of 5% was adopted as a classification threshold for actives and inactives because of the high standard deviations of the experimental values. And if compound was classified as geroprotector for at least one model organism, it was assigned the generalized activity category geroprotect. And with PASS, we created the appropriate models, and one could see from this slide that invariant accuracy of prediction EAP and 20-fold predictivity assessment EAP are quite good in this situation for model organisms. You can see that average invariant accuracy of predictions is 0.903. But you can see also that we are able to make prediction for some reference drugs like metformin and physitine. Some examples you can see for different model organisms, and generalized geroprotective activity is predicted as well. In summary, for 15 of 16 reference drugs, at least one biological activity is predicted, and for 14 drugs, generalized geroprotective activity is predicted as well. However, when we consider the action of potential geroprotectors of model organism and clinical trials, which actually are performed for different potential geroprotectors by specifying a particular age-related disease, you can see that there is very small correlation between the results obtained in model organism and selection of appropriate compounds to the clinical trials. Therefore, when you are able to make predictions for model organism, it may be not enough to be sure that the appropriate geroprotective effect will be observed in humans. That's why we actually tried to apply an alternative approach for creation of machine learning models to predict potential geroprotectors. We observed the agent hallmarks for the potential geroprotectors, which includes altered intercellular communication, stem cell deflation, cellular senescence, mitochondrial dysfunction, etc. They actually were also overviewed by Moskalev et al. In trans-endocrinological metabolism. And since PAS predicts over 8,000 types of activities, it's possible to select those which are intersected with the action on the aging hallmarks. So you can see here, for instance, if mechanism is prevention of free radical oxidation, PAS actually have free radical scavengers, antioxidant, adenosine deaminase inhibitors, etc. If it corresponds to the maintenance of mitochondrial functions, in PAS we have mitochondrial electron transport inhibitors, peroxisome proliferated activated receptor gamma agonist. For maintenance of proteostasis, we have RAGE receptor antagonist, anti-amyloidogenic, autophagy inducer, etc. So finally, we selected 115 activities in PAS which can be associated with anti-aging effects and mechanisms. And when we train PAS based on these activities, we have the invariant accuracy of prediction 0.96. And you can see on this slide for different activities. Prediction for reference drugs also showed that we are able to predict some pharmacological effects and mechanism of action which are known for these drugs. For instance, here for quercetin, some antimutagenic, antioxidant, etc. properties are predicted with high probability. And of course, taking into account very popular topic for drug repositioning, we actually try to apply our PAS-GIRO program to worldwide approved drug database, which were created by my team just recently. It's actually 4 years efforts to create such database. And of course, most of you know that there are drug database like Drug Bank, like Drug Central, but actually most of these databases include the information about drugs that were approved by FDA, by European Medicinal Agency, by Canadian regulators, etc. But when we are looking for the statistics, there are much more compounds or comparable amount of compounds which are approved as drugs in other countries by other regulatory agency. And sometimes compounds that are approved in other countries are approved by FDA in many years after the first approval. For instance, for taurolidine, it was approved in Austria in 1987 and by FDA only in 2023. And for instance, neostigmine was approved in USSR in 1964, but in US only in 2013. So we created this database which have more than 4,500 pharmaceutical substances approved by many registering agencies. And when we are looking which geroprotective activities are predicted by PAS-GIRO for drugs from a worldwide approved database, drugs, you can see that most of them are transcription factor and F-kappa B inhibitor, autophagy inducer, etc., etc., etc. And when we try to select such compounds, for instance, combination by transcription factor and F-kappa B inhibitor and antioxidant among the compounds from what database, You can see, for instance, that propofol, thiotic acid, alpha-tocopherol are candidates for the experimental studies which could combine these activities. And since PAS and PAS-GRO has 100 activities, one could combine the particular effects and particular mechanism of action, keeping in mind the individualized application of potential geroprotectors. And another database that also was created recently by my team is Phyto4Health, database of phytocomponents from Rush Pharmacopoeia medicinal plants. And we actually applied PAS-GRO, and the statistics of 12 top predicted geroprotective activities for these phytocomponents is given here. And also at the top you can see autophagy inducer, transcription factor, NF-kappa B inhibitor, antioxidant, etc. And we are trying to search by pharma expert. You can see some examples of compounds that are predicted, like geranium, sesamolene, alpha-tocotrienol, astilibin, etc., with the combination of the requested pharmacological effects and mechanisms of action. Therefore, you can actually apply this for search for some combination of a mechanism and effects in identification of potential geroprotectors. And of course, taking into account our experience in large ways virtual screening, for instance, during the COVID-19 time, we screening over 1 billion structures for 4 targets: MPRO, PLPRO, RDRP, TMPRSS2, and identify some hits, and some of them were confirmed by experiment. Therefore, we can provide screening in silico for new potential geroprotectors among billions of structures. But I would like to stress that before the high throughput virtual screening, one should clearly identify the aim, which combination of pharmacological geroprotective effects and mechanism of action is desirable for particular age-related disease and for reducing the biological age of the person. Therefore, to summarize, I would like to stress that aging of population and increase of age-related diseases is a significant challenge for the society, particularly in India and Russia. And geroprotectors may slow down the aging process and prevent age-related disease. But definition of geroprotectors is still somewhat ambiguous, because even pure water and antibiotics may increase the healthy lifespan. And of course, using our software PAS and Pharma Expert, it's possible to perform in silico screening aimed to discovery of new potential geroprotectors. And despite of very high complexity of aging processes, investigation in this field would shed light on the promising directions of pharmacological interventions. The further efforts in this field will certainly improve our understanding of the problem, and their results will lead to the healthy longevity in the population. And with this, I would like to acknowledge my colleagues from the Institute of Biomedical Chemistry who are taking part in this project, and the colleagues from Russian Clinical and Research Center of Gerontology, and also Alexei Moskalov from Longevity Institute with a clinical or preventive medicine and rehabilitation. And this study was supported by the Ministry of Science and Higher Education of the Russian Federation, and it's very important that we have such support. Thank you for your kind attention. Thank you very much. And I, of course, open for questions and discussion.
Concurrent diseases, age-related process in human.
Sorry?
What would be the appropriate clinical endpoint for clinical trial of these geroprotectors, particularly in view of several confounding factors and concurrent disorders?
Actually, when you are looking on the definition of geroprotectors, you can see that currently there is no some terms approved by FDA or European Medicinal Agency. Therefore, most of clinical trials are performed for some age-related disease, like, for instance, cognitive disorders for Alzheimer's disease, for some musculoskeletal disease, for fibrosis, etc. But when you are try to search in clinical trials org, you can see that agent could be associated by the selection of appropriate cohort of patients, because these clinical trials are performed in people older than 60 or maybe even 65.
So would you recommend these geroprotectors to be routinely taken by age more than 60, 65, 70, or 75 or 80, or this should be taken only those who are vulnerable, more vulnerable, suffering from one or other age-related disease?
It's not very clear. Sorry. Could you please repeat?
If I am 70 years or 65 years or 80 years, should all age of this age group should take geroprotector as a routine, or you would recommend only those who are suffering from one or two age-related disorders?
Okay, I think that currently all understand that healthy style of living, including diet, physical exercise, it is very important. And of course, if you have some particular age-related disease, you should apply the appropriate drugs which could actually cure this disease or at least reduce the symptoms. And currently there is no universal recommendation. This application of geroprotectors actually and rehabilitation of the patients could be very individual, taking into account all genotypic and phenotypic peculiarities of the person.
And taking time for this lecture. And it's very interesting, but why my
Sorry?
which should be taken particularly say which disease? There are so many diseases, no enteroprotectors. So is there some priority or sometime how you are you going to assess for this zero protection, or how you are assessing?
I heard only part of your question because the connection is not so good. Yeah, so please repeat.
Yeah, my point is, yes. No, I hear. Yeah, I am Anil. I think you are seeing me also?
No, not
Okay.
see you.
I am Anil
Anil,
Saxena.
Anil, I understand. Yeah, thank you.
Yeah. So,
No, nothing. I don't hear. Communication is not well. I don't hear you at all.
Hello.
now you are listening?
Yes.
And my question is that there are so many diseases which come in the old age, like Alzheimer's, dementia, diabetes, and so many. So is this zero protected? Which of these, or there will be overall effect? So how are you assessing that? Which it is what parameter you are taking that is a good zero protect for which disease, or how this thing is being parameterized in your analysis?
I do believe that when you have the complex situation like many age-associated disease, first problem is the diagnosis, which particular disorders has the certain patient. And then, of course, it's necessary to apply the combination of pharmacological interventions, which took into account these peculiarities of the situation. And maybe in this case, Ayurvedic medicine or some plant extracts will be more promising than the separate pharmaceutical substances we actually were indicated for a particular conditions. That's my current understanding. But I should stress that we are still in the beginning of studies, and therefore I do believe that we will have more clear recommendations maybe in 1-2 years, because we started the studies only in the middle of 2024.
Okay, thank you very much.
Thank
Thank
you,
you.
Anil. Okay.
For 40, and one observation to all of you, particularly the people sitting here, before this lecture started, I saw about 27 young people sitting in gossiping in the last few benches. हमने सिर्फ एक सेंटेंस में कहा कि आप ध्यान से सुनिए. Maybe I can ask 1 or 2 questions. If you don't answer, your attendance may be cancelled. अब आप एक भी स्टूडेंट नहीं है यहाँ. Shame. जो अंग्रेजी में कहावत है ना, उसका हिंदी में बोल रहा हूँ क्योंकि सुन रहे हैं. घोड़े को आप तालाब के पास तक खींच के ले जा सकते, लेकिन उसको पानी पीने के लिए फोर्स नहीं कर सकते. अब डॉक्टर सक्सेना ने इतना अरेंजमेंट किया, इतने एक्सपर्ट्स को यहाँ पे लेके आ गए, लेकिन हमें अफसोस है यहाँ के जो सुनने के लिए आए हैं, न तो उन्होंने ध्यान से सुना. जब उनसे कहा गया ध्यान से सुनो तो उठ के चले गए.
I think this generation need to be hammered. या तो उनसे कहो अटेंड न करें, अटेंड करें तो they should bring some seriousness. I think we have to be, we are failing somewhere in telling our young generation the ethics, the etiquette, and the sincerity. I think we have to put this. अगर अभी हम नहीं सिखाएंगे तो कब सिखाएंगे उन्हें? Thank you very much.
Thank you.
Thank you, sir. Thank you, sir.
Thank you very much, sir. Wonderful presentation.
Now, I would like to request Honorable Chairpersons, Professor Dr. Y.K. Gupta sir and Dr. D.K. Dixit sir for presenting a memento to the speakers of the session. I would like to invite Professor Dr. Anil Kumar Saxena sir, Professor Dr. D.S. Rawat sir, and Mr. Prakhar Shaswat sir. Please come on the dais. And I would also like to request Rajan sir to show the e-memento of Professor Dr. R. Natarajan sir and Professor Dr. Valamudi sir. Now, I would like to request Professor Dr. Anil Kumar Saxena sir and Professor Dr. D.S. Rawat sir to present a memento to the chairpersons of the session. Thank you, sir. We have organized a short tea break, so I request all the dignitaries and participants to join us for the tea, and we will start our third scientific session after the tea break. So I request everyone to join back at the conference hall at 12:45.
After 10 minutes.
15 minutes.
Yeah.
15 minutes.
And now for the next session, scientific session 3, the honorable chairpersons are Dr. Alok Saxena and Dr. Aruni Saxena. And the speakers for this session are Professor Dr. Subhra Ghosh Dosidar and Ms. Anusri Sinha. So please come back to the seminar hall after 15 minutes.
Hello. Hello. Check. Hello. 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 Check. Hello. 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 Check. Hello. Hello. Check. Hello. 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 Check. Hello. Hello. Hello.
Hey, हेलो ठीक वो हेलो ठीक वो नहीं नहीं आप इसमें लिखिए आपको ऑडियो आ रहा हो आपको ऑडियो आ रहा है ना फाइनल होगा मैं कंट्रोल कर है ना हेलो ठीक वो हे हे नहीं नहीं आवाज आएगी आपको ऑडियो दे आपको ऑडियो सिग्नल दे रखा है ना ये वाला? नहीं आ रहा? ठीक ठीक ठीक
equal A. Equal A. B T equal A and 1. A equal A J A 1 J A
Welcome back. Now we are going to commence the third scientific session, and I would like to invite Professor Dr. Alok Saxena sir and Dr. Aruni Saxena sir to chair the session. Dr. Alok Saxena sir, superannuated as Managing Director of Selzer GTS India Private Limited and is currently the proprietor of AA Consultant. He is a chemical engineer with a doctorate from IIT Delhi and a postdoctorate from the University of Oldenburg, Germany. Dr. Aruni Saxena sir is a clinician with an interest in the management and development of health plans and policies related to global health security. And he has completed his medical graduation and doctoral studies in medicine from the University of Rostock and Heinrich-Heine University, Düsseldorf, Germany. Over to you, sir. Hello. Working. Hello. Yes, the session is over to you, sir.
Hello? Am I audible? Yeah, this is the second part of the session 3. The first lecture is by Professor Dr. Subra Ghosh Dastidar. Dr. Subra Ghosh Dastidar earned his PhD in chemistry in 2005 from University of Calcutta. From 2005 to 2010, he worked as a postdoctoral research in UC Davis and University of Texas, Galveston, and A-Star Singapore, collaborating with renowned scientists like Yang Duan and Chandra Verma. In 2011, he joined Bose Institute as an assistant professor and now an assistant professor in the Department of Biological Sciences. His research focuses on understanding the molecular mechanisms and biomolecules and their complexes using computational methods. As a theoretical chemist, he studies structural dynamics through large-scale high-performance computing. Some of his key research areas include tubulins and BC12 family of proteins and kinases. He is the coordinator of DBT-funded Bioinformatics Center project by Bose Institute and also leads a multi-institutional national project. So far, he has published 46 papers, all in internationally recognized journals. He is currently associate editor of Frontier in Molecular Biosciences. To Dr. Saurabh Ghosh Dastidar, please.
Hello, hello. No, let's go. Going. Look at when you do the other one.
Oh, this is better. Thank you for your kind introduction, and I thank the organizers for giving me this opportunity to talk here. I like to come to these conferences. I have been in these meetings for several times. So I'm going to talk about allostery in proteins, protein structure, dynamics, and some small molecules as well. And from my experience, as I guess that there will be students predominantly in this audience. So I have designed my talks to start with some basics, okay? So don't feel shy to ask questions at the end of the talk if you want. Okay, so let's start with scratch. So anything we see or around has some molecular basis. So that is the first point I want to make. Suppose someone has broken his hand or leg, so you ask the reason. So if the answer is, so I wanted to walk on ice and then I fell down. So then you ask this question, why? Then you ask another question, why? This is how the scientific discoveries or research progresses. You ask why? Because if you ask walk on ice, then it can only happen, but not on concrete. So what's the difference? So once you ask this difference, you go into the molecular basis. So then you go to the ice structure, and what is the difference of the ice structure from water structure? So gradually, so anything around could be narrowed down to some molecular events. So you can even go further to ask in the electronic level reason. So everything is connected to each other, and it is up to you at what level you would set your investigation, and you would be satisfied with that kind of answers that you get. So similarly, let me make another point with an analogy that you all know, that Darwin, you know about the Darwin's theory, but that time it was not known what is the molecular basis of this evolution, right? So there were some molecular reasons, definitely, but he had this hypothesis without the understanding or without the details of the molecular basis. So around the same time when Darwin was doing his, was publishing his theories and discussing it, around the same time, Mendel also did another experiment, almost 5-6 years difference, but unfortunately, they did not know each other. So even the Mendel, started this quantitative way of understanding genomics, but he also didn't have the understanding of what molecules are responsible for this. So then, almost after, say, 78 or 90 years, that people came to know there is something called DNA. Okay, structure of DNA and DNA as a molecule of inheritance are two separate things, because they ask two different questions on same molecule. So people came to know what is the molecular basis, that DNA is the responsible for taking the information from one generation to another generation. So ultimately, from Darwin, we reached to a level where we do quantitative genomics, there's molecules. So ultimately, we deal with molecules, and then again, when you come to the molecules, we talk about conformation structures. So then the Watson-Crick model will come. So in this way, anything that we see around, for example, any experiment that we do in our laboratory, it is connected. Any observation that we get in the, supra level is connected to the molecular level. An event like this, where two proteins coming to each other and binding, maybe the experimental result that you are getting, maybe from test tube, from cell line or from animal or even from human trial, actually some level of molecular interactions and binding are there that we need to focus, narrow down and understand so that we can gradually tune the actual effect that we get. So how to do that thing at the molecular level, because you cannot see the molecules, right? So suppose you do an experiment, you can only see a conical flask like that or something equivalent to it and a solution, but you cannot see the molecule. So suppose you have a protein solution and doing some experiment on a protein solution, then how do you interpret that what is going on in the molecular level is connected to your actual result? So let me give an example with a thought experiment. Suppose I have an imaginary camera which can take the pictures of the molecules in this small portion of this solvent. We'll see that for the same protein, a small peptide is shown here. Small peptide is existing in all possible conformations. So the billions of copies are there of the same protein, same molecule, but they are in a different proportion in different conformational states. So whatever measurement that we do on this system, like you do a series experiment to understand the conformations, whatever, thermodynamic measurements on the protein ligand interaction, actually we measure the average picture of this entire thing. So this important point is that there are billions of copies of molecules, not all are in the same conformations, but still whatever we are measuring, actually a reflection of the average situation there. So how to represent this in computer? Because when we do a modeling, we model a protein or whatever, nucleic acid, we deal with one copy of the molecule usually, but not dealing with billions of copies with different situations. So we need to mimic something that is going on in the real situation. So this is how we do it. That theory says if you allow this molecule in computer through some theoretical methods to evolve with time, and you allow sufficient time, sufficient is something philosophical that needs more time to elaborate. But let's assume that we allow sufficient time to explore various conformations, that eventually it will represent an ensemble of conformation that is equivalent to this experimental setup. So there are theories which will connect to this. It's called statistical mechanics or theoretical chemistry, whatever you say. So these are the basis for understanding how a protein unfolds. So this is simulation. So you haven't seen this from experiment, but at least you can simulate and find out what is going on in the molecular level so that you are getting a change in experiment reflecting the protein folding or protein unfolding. So there are theoretical bases behind this. I'm not going into the detail of theoretical basis, that these are nothing but some very special type of calculations called molecular mechanical force field methods that takes into account of interaction between atoms and then calculates forces of interactions and uses simple Newton's law of motion to tell, using a supercomputer like this, that how things are evolving with time. So this is called molecular dynamic simulation, which you can use. There are standard software methods available as a black box, but still you need human interruptions, interventions from the level of your scientific knowledge. So with this background, let me go into a question, scientific question, where we can utilize this kind of methods and understanding, particularly that so far I have discussed. That kinase allostery. Allostery is something that you, suppose I give a pressure here and it opens up, means it's a remote sensing, that you do something at one end of the protein and you distort something on the other side that relates to the function of the protein, that helps the protein to function or it inhibits to function. It's called allostery. So protein kinases have wide variety in terms of sequence and structure, but one thing is common, is that they have a common kinase domain which has an architecture like this, N lobe, C lobe, and ATP binding site. You know, the general mechanism of the function of the kinase is to transfer the phosphate group, one phosphate group from the ATP to its substrate. So this is general truth. And so although this kinase, as you are students of the life science stream, I assume that you understand active, inactive states, etc., and protein structure, native state. So although the kinase mostly existing in a native state, but even around the native state, two distinctly different functionally different conformations exist. Those are called active and inactive. Means one group can do this transfer of phosphate group from ATP to the substrate, the others cannot. So this is called active state. So usually the kinases are inactive, and the active and inactive could be distinguished by very small changes of side chain like this. So whether kinase can do it themselves or they need some help from outside, from other molecules. So there are different kinases which have different kind of requirement. So we are going to dealing with some color problem, just ignore it. So we are going to dealing with some kinase that requires help with another molecule, activator molecule, another protein. So we are dealing with TAC1 kinase, which gets activated in this region, and only when another molecule, TAB1, comes to it, then only it can get activated. It goes to the active site. So the question is how this gets remotely activated, how this remote communication happens. So if I go into the theoretical chemistry language, then it will be difficult to understand that this is a very challenging one, because active inactive transition requires some special kind of simulations. It's not so easy. So I just keep this and give you this analogy. Suppose we are trying to understand the rule of such a game. If I don't know the rule, I'm watching the two players are playing, and I'm trying to understand the rule of this game, which is simulation actually. That a complex event you really don't know, you're allowing the computer to simulate the event using some fundamental principles, and you see how complex things are occurring in time. But for kinases like this, when the conformational transitions are required, you need a special kind of sampling called enhanced sampling. So where you, in a very short time, you have many different types of events. So using these methods, what we have done something, we have computed something very fundamental that is called free energy landscape, or in more scientific term, PMF landscape, potential of mean force. So more precise language is this. So we have calculated what happens for this active inactive transition. So when the activator molecule is not present. So if you cannot read this plot, read it in this way, as if these two colored regions are like two lakes and they're disconnected. And the molecule one has to go from inactive to another active state is just like a fish who wants to go from one pond to another pond has to jump now, means requires activation energy. So this is difficult for the fish. It's not trivial. So what is facilitated by this activator molecule is that it lowers the activation barrier, means it now connects these two states. So you need lesser energy to go from one state to another state. So this is the thermodynamic basis of this allosteric activation. Now this is a thermodynamic basis. So now once we have streamlined these methods, one can try many different types of mutations to decide that can we design many different inhibitors will go and bind in that site and still will not activate. So this is one site that we are currently exploring. But another point is that we want to know that which residues are responsible for taking this information from one side to another side. So this is something very interesting. And for this, instead of relying on our own eyes through visual inspection or some standard analysis in MD simulation, we tried why not to use these new methods, that buzzwords like AI, machine learning. So we tried with some kind of latest machine learning methods, and it's a simple representation. Maybe these details are not relevant to describe for you at this moment. That MD simulation, as I in the introduction mentioned, that you generate many conformations. Actually, this entire set of conformations are converted into a distance matrix. So that matrix is taken as an input by some machine learning methods, which can analyze that which distances or distance pairs of residues, which we call as features, are actually responsible for to distinguish between ensembles of active state and inactive state. So I just keep these details and tell you this result, that yes, finally, it has mapped that which residues are actually connecting this information, and what happens, how their fluctuations are actually taking the impact of this activator molecule binding and taking it on the other side. So if I summarize this entire story, okay, thermodynamics is one thing. So how thermodynamics is, utilized by this molecule is this molecule tab activator comes and it interacts with some residues that we have found from machine learning. And gradually, as the things progresses, it interacts and then it transmits the information on the other side. So this one, maybe I just skip this one. I have shortage of time. I'm just, we have also applied this on another system, tubulin, but I'm not going into about tubulin. But just to talk about the advantage of using machine learning, not machine learning, MD simulations in drug design. The induced fit. What is induced fit? What is the basic principle of designing a drug? That there are many principles, as Dr. Saxena introduced in his talk, that suppose I know the structure of the receptor and I know a peptide or protein binds like this, I have to design another molecule which will mimic those interactions and will displace this. In this competition, the peptide, peptide will get defeated by the small molecule in terms of binding affinity. So here, so far, we have taken this receptor, green color protein, as a rigid object. So any screening software who will try to find out few good binders from the list of millions of compounds will mostly utilize this receptor as a rigid object or slightly flexible object. But what can happen that you see these proteins are actually soft objects. So they continuously change their shape. So what happens, instead of using one conformation which we see from crystallographic structure, if you can generate an ensemble of representation of the structure, and then we screen the molecules. So that we tried on MPRO of SARS-CoV-2, where we generated many different conformations. And then if we expose them, expose the ligands to some of these receptors, then we find if one molecule is not binding to one receptor, maybe it is also a potential binder because it is binding to another conformation. So instead of doing some simulations on the compound after binding, which will have taken many times to complete all the simulations, we have first identified all possible conformations and clustered them and have taken 10, 20, or 30, whatever, it could be anything. And then in summary, it looks like this. See, many different conformations are generated, and different conformations will bind different ligands. So otherwise, if we have used one conformation, many of them would have got, excluded. So we have identified, this is in principle, this is a proof in principle. It's not something revolutionary, just a proof in principle that you can complete this entire thing within 7 days if you have some computers. And you see, we have identified several molecules as potential binder for SARS-CoV-2. Among them, we also got this one, which got FDA approval after that. I mean, it's not that we are claiming that this, as I said, it's not something revolutionary, but at least we say that this compound would have got missed out if in a prediction, if we have done with a rigid object. So I think maybe I can just one small story that how simulation can also help you, just take 2 minutes. So how can also help in doing some fundamental chemistry, like, okay. So one of my collaborators, Professor Pallab, came with a question that, okay, I am working on a plant protein which is active only at a low pH. The question is very precise, that this is functional only at low pH, not at neutral pH. Can you tell me that why it is active only at low pH, not, and what is the source of this problem? I mean, source of this pH sensitivity. So here we have run some calculations, and what we have found, the summary of the story, that we have found several glutamic acid residues which are forming the catalytic site of this particular protein. And they need to come very close to each other in order to bind the substrate. So I'm cutting the entire story short just to give you the feel. So if they have to come close to each other, now that's, the knowledge of chemistry comes into play, that at neutral pH, all these side chains of glutamic acids will remain ionized. So they cannot come close to each other. There will be repulsions. So then if we use low pH due to protonation or due to common ion effect, some of them will be partially neutralized. So then they come close to each other and they function. So if this is true, you see, then it's just a gaze. Then I said you have to go to the sequence alignment of evolutionary, from evolutionary aspect to look that if there is any protein present in the evolution in other species which has that some of these residues mutated to glutamine or any other neutral residue, it will have pH sensitivity. It will not have pH sensitivity. Like sequence alignment, I prepared this slide for non-biology people like physicists, that what is sequence alignment? So when the sequences are very close, we say they're functionally similar. Like these two words have similar meaning. If you know this was the longest word in English dictionary. So they're very, they have close meaning, but sometimes one small change can have a huge difference in the meaning, right? So sometimes one little change could be crucial. So we identified one residue, one glutamic acid residue, and asked the collaborator to mutate. And we really found that that particular glutamic acid has glutamine substitution in many species which are not really pH sensitivity. So then they mutated it one shot. This is how simulation can help you to narrow down the question, and ultimately you can do some few experiments and get it confirmed. So it was confirmed, and you also proposed another mutation which should not change the function, and that was also confirmed. So with this, I stop, and these two collaborators are only for those works I have mentioned here. Professor Bhavatara Bhattacharya is no more, but I started to work with him on tubulin funding, and thank you.
Thank you, Dr.
Shubhra Ghosh, Rasida. It was really nice and good insight about the insights from the molecular interaction and machine learning is something good. And thermodynamics, I suppose, also plays a role in this simulation package, what you are using to. Are different options available for thermodynamic models, or it's only. Hmm. Hmm. Yeah, actually, this question is from my background in chemical engineering, where also we have phase equilibrium thermodynamic model. Maybe there may be some similarity. Is there any other question?
Thank you very much. It was a very interesting topic, and hope many questions would be there, which we can also handle in the break. Now, I would like to introduce a very young expert, I should say, a learning expert in machine learning. She is Anushri Sinha. She has working currently with Google, and I think everybody in this room knows Google. I don't need to give any introductions to it. She did her engineering from Birla Institute of Technology, and then did pursued masters in machine learning from Georgia Tech Universities in US. I have been very fascinated with use of machine learning in my while I treat patients, and potentially I would give you some examples how I use it. But then it's a very interesting topic on machine learning technique for breast cancer diagnosis. And now over to Anushri.
Thank you. Hi, everyone. My name is Anushri. Before I start, I'd like to thank the organizing committee for inviting me here and giving me a chance to speak to you all. I would like to extend a special thank you to Dr. A.K. Saxena and Dr. Arni Saxena for this invitation, and my family for always supporting me, many of whom are present here today. Before I begin, I would just like to say that I'm not an expert in the topics and the fields that we are discussing today. So I might misuse a lot of, chemical, medical terms in this presentation. So please forgive me for that. I've started this presentation with something about machine learning, some basics, because I know that a lot of you probably would not have a lot of computer science background. So please, if you have any questions, feel free to ask. I will link some of the resources and code that I've used here. You can refer to it later, but please feel free to ask. I am as novice to your field as or probably more than you are to mine. So yeah, with that, let's get started. Oh, it's not. Okay, no worries. So I'm going to be talking about using machine learning techniques in breast cancer diagnosis today. These methods are in no way limited to this analysis, any medical analysis, or for that matter, any analysis in the world. I don't use it particularly day to day in, obviously, in these fields, but I use it in, very different fields. Okay, so we'll talk about what machine learning is just briefly. I'll try to explain it in very layman terms. And then we'll go into looking at the data set, the exploration, preparation, model building, and analysis. Next slide, please. Okay, so machine learning has been around for a really long time, 30, 40 years, and so has artificial intelligence. But as we all know, it's the latest fad in the last couple of years. I'm sure all of you have used some version of generative AI, whether it's ChatGPT, Gemini, Perplexity AI, whatnot. Without overwhelming everyone, I want to actually explain it in very layman terms and something that's how I understood it, 15 years ago when I began my journey, as a computer science student. So all of computer science is about understanding and predicting every single outcome of any possible problem. For example, if I gave you the equation of a straight line, there are no set of coordinates in the universe that you can't look at and say whether this fits in this line or not. But if I ask you, if X number of people attended the Mahakumbh this year, can you tell me the exact number of people that will attend Mahakumbh 4 years from now? And the best you can do is come up with a very, very, very reasonable estimate of how many people will be there. There is no exact number that you can come up with. And the ability to be able to predict that very, very, very reasonable estimate is what machine learning is all about. Okay, next slide, please. Okay, so this data set I actually picked up from a research paper that was actually published for very different reasons. It's about polynomial distributions back a long time ago by two professors from University of Wisconsin-Madison. The data actually is about, it was created from like digitized images of like the breast mass nuclei and okay, so I'm just going to talk about the data a little bit so you understand what it's all about. It has about 600 rows and about 9 characteristics of cell nuclei, and it tells whether this particular cell nuclei is malignant or benign. So that's what the data set looks like. It's a very clean data set, so we won't spend a lot of time understanding how data is cleaned. I'll talk about it a little bit. Next slide, please. Next slide. Okay, so I've done all of this exploration in a Python collab. Again, it's very technical, I don't know, CS thing, but I will link the collab here if someone wants to run the code. They can do it at leisure. And I've added some code snippets in the slides. Feel free to ignore it if it doesn't make a lot of sense. So this was one of the first analysis I did. The data actually contains about 300 instances of benign cell nuclei and about 200 of malignant. And that's just a histogram that I plotted. Next slide, please. Okay, now this is slightly small, but okay, that's fine. Okay, so then what I did was I basically created a histogram of each of the characteristics of the cell nuclei and for malignant and benign cases. And it's not visible here, but if as you see that, for example, the first one, that's the radius mean actually. And the one below is perimeter mean. The green ones are all benign and the orange ones are all malignant. Yeah, okay. So as you see that the radius mean, the perimeter mean, in those, it's very, very clear that if the nuclei is larger in size, it's going to be a benign nuclei, and if it's smaller, it's malignant. Similarly, if you look at the third one, the smoothness mean, okay. The third, okay. think it's oh long. No. It's okay. I'm talking about the first column, third histogram. So if you look at the third one, that is the smoothness mean of the cell nuclei. And as you see, there is no correlation there. You know, the two histograms almost overlap with each other. And similarly, if you see the last, okay. Oh, okay. Similarly, if you see the last one here, again, you see that there is no correlation between the malignant and the benign cell nuclei. This is just a data analysis. This is just histograms. There's no machine learning that I've used yet. But typically for any kind of analysis, we do these kind of explorations just to be able to get a sense of what is relevant. This is a very small data set that I've used for this example, but in real life, we are dealing with data sets that range up to like millions or billions of rows. And one of the harder challenges of machine learning have always been being able to procure the data, being able to clean the data, being able to analyze the data. And there are several challenges that you face along the way, especially in the health industry. If we talk about it, it is very difficult to actually get data because of, in the US, there are HIPAA regulations. In other countries, there are similar regulations. The data needs to be de-anonymized at the level that you're not able to identify the patient. The data has to be cleaned. The data has to be, you know, the patient has to give some kind of like permissions to be able to get that data. And then once you get that data, the data is not always clean, you know, because how many of us actually go for our annual reports or annual blood work every year? So it's extremely difficult to get that data, clean that data, and use that data. So I just did a little bit of analysis here so that you understand that these kind of exploratory steps are necessary before you jump into any kind of modeling onto the data. Next slide, please. Okay, next slide. Okay, so that was the data exploration, just to get a feel of what the problem that you are looking at is going to look like. Okay, the next is preparing the data. So whenever we do any kind of ML analysis, for you to be able to create a model, you don't know, you know, what is the case that is going to come to you tomorrow. So how do you test your data? And what we typically do for this is you already have this data set and you have the answers for each of the, you know, rows. So what we do is we split this data in training and test. We only use a certain percentage of the data for training and then test our models on the test data. So here what I did was I basically split the data into 70-30 because here each row is like individual, so you don't need to do anything complicated. After this, I don't touch the test data at all. You move it aside, keep it somewhere else, don't look at it at all. It is almost like a new patient walking into your hospital asking you to answer their questions after you've done the analysis. Next slide, please. Okay. This is a slightly, I don't know, not usual, but I'm just adding this sort of concept here because I've used it in my notebook. What this does is not, like you see, I'm not touching the test data at all. Here, I've not touched it at all. What we do is, it's called K-fold cross-validation. The K value can be anything. Usually we use 5 or 10. You can make it 100 or 1,000 depending on how big your data is. And what we do is for each of the splits, we actually take one of the 5. And what I mean by the split is like you had your data, you divide it into 5 equal parts. And now 4 parts is training and 1 part is test. And this is all happening within the training data. And then basically each of those 5 parts for each of the iterations, you make one of them the test and 4 of them the training data. So what this does is, and I'm sure all of you are familiar with that, the concept of mean or expectation in probability and statistics, what it does is it evens out your model. It removes any biases, overfitting, underfitting, and it makes your model a lot more robust to any kind of outliers. Yeah, so I've used this here and I'm going to, that's why I wanted to explain. Next slide, please. Okay. No, just can you stay on that slide? Okay. Um, there's another concept in machine learning, which is classification versus regression. This particular data set is a classification data set. Why? Because all your outputs are either going to be 0 or 1. And these are discrete numbers. So for example, if I said, I don't know, what color is your child going to choose, if you go take them to buy a bike or something? That is going to be a discrete number. It can be red or blue or yellow or green. There are only so many number of colors. But if you're asking someone what is the average weight of a woman who's 35 years old, that's not a classification model. That's a regression model because, as you know, it can be anything, 70, 70.1, 70.2, and it goes on forever. So because this data set has discrete output values, we here use classification model. The kind of model that you can run on your data set changes with the kind of data set. So the only models we will use here are classification models. Next slide, please. Okay. So you'll all hear about ML and AI so much. And then when you get into it, you realize that so much of it has existed for so long and has been used in such versatile ways, you almost didn't even realize you were at the foundational levels of ML. For example, logistic regression, there's nothing new about it. It's one of the core, concepts that you study in college when you're doing probability statistics of any kind. And it's one of the first models that we learn, we use, we run on almost all classification models. So anyway, so logistic regression is used for classification models. And I started with this. I ran the model on the data and it gave me an accuracy of 88.42%. Okay, not great. And then for each of the cross-validation scores that I spoke about, where I've created smaller test and training from the training itself, it was in the 80s. So, okay, good starting point. And I wanted to, can you go to the next slide? Okay, so the next model I ran is called a decision tree model. I know all of you are life sciences students, but I'm sure all of you have studied some kind of computer science at some levels, 9th grade, 10th grade. Decision tree is nothing but a binary tree or an n-ary tree. And I just kept an example here is like, the question they're trying to ask here is, should I have coffee right now? And then it's like, what time of the day is it? Oh, it's morning. And then you're like, but are you tired? Are you tired? Yes. If not, don't. So it's like that. It goes into a yes and no, very easy, but it can have like any number of outputs. So I started with the decision tree and the accuracy here was 100%. You must be like, oh, I found the answer to like all breast cancer analysis, but that's not what this is about. And the cross-validation scores were low. So what does this mean? Why is this happening? This is because of something called overfitting. Again, it's a very common concept in probability statistics where something relates very highly to the data. It doesn't mean that it's the answer. It just means that if you remove everything else, you have basically conformed the model to this particular parameter, and it's overfitting it. It's almost always right. But the moment you bring in something from outside, it will fail. So if something is too accurate, it is actually a cause for alarm rather than, cause for like joy. Okay, so this was with all the predictors. And okay, can we go to the next slide? So what I did was I just tried to try the decision tree again with a single predictor. And you see it's already changed. It's 97 now. So then you'll be like, why? What is this happening? So like I said, it was because of overfitting. The moment you change something, answers will start to change. Okay, next slide, please. So why I spoke about decision trees is because of the final model that I used on this data, and it's called the random forest model. A random forest model is just a collection of decision trees. Going back to, our core concepts in probability statistics, the expectation, the mean. What having a large number of trees does it evens out, normalizes your models. And a very good example of random forest is kind of like you're sitting with a group of friends and, you ask each of your friend who's going to win the Cricket World Cup, and you will get a different answer. Someone is going to be like India, and someone will be like South Africa, and Australia, and everyone will have their own reasons. It doesn't come out of nowhere. There is grounding there. And what it does is it opens up your mind to different possibilities. It evens out your opinion. And that's the same thing that happens in this kind of model. And this is a very common concept in ML. We almost always do this. I don't have the time to explain, but it's because we want to avoid overfitting, underfitting. We want to avoid any kind of false positives or true negatives. Okay, next slide, please. Okay, so an advantage of the random forest model is that it also lets you figure out feature importance. What I mean by that is, of course, all features are contributing to your prediction of whether a nuclei is malignant or benign. But it's very likely that some features are more correlated to the answer than other features. How we figure out what feature is more important is, for example, you took any two random rows, and I'm taking the radius as an example, radius of the nuclei. And supposing if the radius was, say, 1, or let me say 2, the cell nuclei was benign. But if the radius was 0.5, the cell nuclei was malignant. Just switch them, make it 0 and 1. Now you've broken the connection between the characteristic and the output. Run the model before you switched them and after you switched them. If before you switch the model, before you switch the values, the model was like at 95% accuracy, and after you switched it, it was a 65% accuracy. And I just took 2 rows. I meant you can take any number of rows and switch them around. You'll realize how important that particular characteristic is for your analysis. And that's what I did here. And I basically found the importance of each of these features. Again, there are some pieces of code here. Please feel free to ignore them. Yeah, it was just to help me out and you if you have any questions of how I'm doing this stuff. Next slide, please. Okay. So what I did was I ran it with the top 5 features, and you see now that the accuracy is about 94.2, and the cross-validation scores are very similar to the accuracy, which means that your model is actually performing well. There are less chances of you overfitting or underfitting the curve. Can we slide down, please? Oh, I feel like I missed a slide. Can you go up a little bit? Slightly up, please. The one above that. Oh, okay. So I had run the random forest model before I did the feature importance as well. And if you see here, the accuracy was 94.72. The difference between this and the 5 features that I picked is not too much in this case. It can be substantial in some cases if the values are like very, very skewed. Again, it's a cause for concern. If it's too much, please look at the data again, because one feature might be overfitting your curve. You can go back to the last slide. Okay, finally, we go back to whichever dark room you left your testing data in. Scroll down. Okay, and we ran the random forest with it, and you see that the accuracy is 96.49, which is actually pretty good for any kind of like ML model testing. Please note that these kind of accuracies happen a lot when you're in the classroom or doing something which is more theoretical. In real life, it's harder to achieve these numbers. But yeah, nevertheless, it sort of tells us how to go about these things. Yeah, okay, that was all from me. Does anyone have any questions?
Okay. Thank you. Very interesting presentation. I think for the next generation sitting on the back, machine learning should be something that you know as you are using your WhatsApp. So for you to get more job opportunities, what you can offer to a company when they come on the campus is the thing. So I did P-Farm, but I also know what applications of machine learning could be in the my domain or the company's product development, right? And this sort of a presentation sets the ground for it. We already have had some presentations early on which were using very high advanced use of machine learning and statistical analysis. So I would encourage any of the people who are on the back, we also say Gen Z. Do you have any questions about what machine learning is all about? Don't hesitate. It's a new technology. You might ask some very basic question, but would encourage to know how do you feel that you can use machine learning in your area? Well, I have one question from the clinical perspective. I had been using AI for long now, for like almost 10 years. The thing is, of course, you did forest model, and that is one of the things which is very time consuming for us if we do classical logistic regression. You think that using a forest model for getting and removing all the co-founders or outliers would improve potentially the practical implication of the model equation would be developed in sense of the real world? Like we do remove outliers, and of course you might say it's not very fair, you should have the old data points in it, but of course for getting better R square, it's a very common technique which I think most of the PhDs and researchers have been using. But do you think what is a better perspective, whether we should use forest model on a regular basis, or is it some sort of minimum data requirements to be?
So, like I said, this is a very foundational presentation. And I think in one of the presentations that Dr. A.K. Saxena gave, I think he mentioned using neural networks and with backpropagation, I think back in the 80s or something. The models are so sophisticated now that almost no one uses like logistic regression. In fact, the neural networks are also so sophisticated at this moment that the researchers using the models themselves after a point have no way to predict what goes into the black box and what comes out. So I would say that, and this is for everybody who's in a non-technical background, I don't think there is a lot of ROI in learning. It's better to be using generative AI. The large language models are very, very sophisticated at this point, which is why we are having all this the AI is going to take over. Without really causing an alarm, it's actually pretty true. People who are developing those large language models, and I'm one of them, we also don't know what happens after a while when it goes into the box. So no, I don't think people use decision tree models very much. Almost everyone uses ANNs and like really huge.
Oh, thank you. Very, very good source point for us to learn.
Thank you very much, Dominic, for this beautiful presentation.
Yeah, so basically you're talking about the area under the curve or the R square, which is what I was talking about. In one of the slides, I mentioned that if you take one feature, it gives almost 100% accuracy. And that's overfitting the curve way too much. So I don't have the numbers here. I will share my collab later, but you can take a look at that. Yeah. So this data set actually contained something called the tenderness of the nuclei, but that was the only, I would say that's the only physical attribute. I mean, radius and perimeter, all of those are also physical, but there is no of the cell nuclei. Yeah. Also, I might not be using all the terms correctly. Like I said, I'm not a life sciences expert. So, because the data set that I used actually was picked up from a research paper that was already published by the University of Wisconsin-Madison, the probability of it being biased is low. But you're right. And this is a big challenge. So I can give an example. For example, if you, and we can take any disease for that matter, if you get the data set of a particular country and you do your analysis on that, there can be like a high bias of some characteristics, whether it can also be up to the level of the drinking water or, the weather conditions. And that is a big challenge in the health industry because it is extremely difficult to obtain, especially in the US, it is extremely difficult to obtain data for any kind of analysis, especially when it comes to like medical. But it does give us some insights into it because a lot of health analysis changes with race. And what I mean by that is different for Caucasians, different for Asians, different for South Asians. So sometimes it is a blessing in disguise, but in general, I think it is, and also not all countries have a central body of data. So that is also a huge challenge, even within a country. Yeah. Hmm. Hmm. Yes, so that has been a challenge, but I feel like in the last couple of years, ever since Microsoft broke open with ChatGPT, it seems to be changing because a lot of Gen AI is getting incorporated into enterprise level products and end level products, whether it is the Office suite, whether it is Google Drive, whether it is, and there are more and more such products coming out. Before ChatGPT, there was a lot of research, but the research had not been translated into products. And that's the key change that we're seeing now. In fact, even for enterprise industries like Oracle, we have, and Google Cloud, there are a lot of new products that are coming. For example, there's Vertex AI with Google Cloud that is solving these challenges where you don't have to know every line of code to be able to like solve the real problem there. Yeah. Of course. Okay, I'll answer the first question. Okay, I'll answer the first question. I think for the second question, I will give it to Dr. Arni Saxena. So as an engineer, I can tell you that I can predict anything as long as you give me the data. So if you give me the data that has gene characteristics, you will get a model that will give you answers on gene characteristics. If you don't give me the data on gene characteristics, the model will not give you answers on gene characteristics. That said, I don't know if gene characteristics affect diseases, affect cancerous diseases. And for that, I will give it to Dr. Arni Saxena.
Yeah, no, I think I would be able to answer you this question specifically in my presentation, where you would see a real world case how it is done, and potentially I would like. Yes, please go ahead. Yeah.
Yes, definitely. Correct. Okay. Definitely. Correct. 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 Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes. So I'll answer your question. I'll answer your question twofold. Your first class in classical mechanics, when you're taught F is equal to mg, you don't account for air pressure. You don't account for that's because if you don't learn F is equal to mg, there's no way you're going to learn F is equal to mg plus c. Computer science has actually grown exponentially, even from the time that I was in college, and that was like a little over a decade. Um, One challenge that we face now, and you're right, ANNs have also not been that successful. One challenge that we have faced, and we face it even today, as we grow the model, it becomes extremely, extremely difficult for us to procure resources for those models to run. Um, I don't know if you heard about the new TPUs and GPUs. And the models have gotten very, very, very good now. Like I said, I think even the engineers don't understand some of the models beyond a point. The second thing to your point about uh, using random forest or using ANNs, I think it's at least from an engineer's perspective, I don't have the knowledge and the experience that you do. It's more of a hit and trial for someone like me because I don't have that kind of background. And I think this is where your experience and other dignitaries' experience counts. And yeah, I mean, I'm still learning, so definitely, yeah. Definitely, definitely. Definitely, yeah, definitely. Yes. Yeah. Correct. Yeah. Yes. Okay. So have you ever paid for any Google product? Okay. So most of the things that Google offers are free for all. And that's the motto. Search wants to be free for everyone always. And I don't speak on Google's behalf. This is my personal experience. This is a big challenge. Same thing that I was trying to tell ma'am. It is extremely difficult to procure any kind of GPUs and TPUs. we are fighting with our teams because, and then we find optimization ways of using the same TPU among 10 teams, So that's also another challenge. That said, there is a big concept of sensitivity, especially in the tech industry. You're talking about a butterfly, right? Now, say you are looking at, you can say, take the same thing, you're studying, trying to understand breast cancer. You want to look at some cell mass nuclei. You go Google something. You don't know when that imagery will shift from breast cancer analysis to pornographic imagery. And it is a daily challenge for people working in my industry to make sure that, and it's you. I mean, you're an adult, but a 14-year-old or 16-year-old could be doing something like that for their project. Who is responsible for that individual's growth or that individual's well-being? And that is why lens is so tricky, because it's an image and every single pixel and change in that pixel can really change what that image means. I've just come up with one example, but there are any number of examples. So for us to be very, very sensitive on people of color, people of all kinds of gender, animals, everything, it's extremely important. And that's the challenge that we face. We have to run several rounds of sensitivity just to be able to get a product out for testing. And yeah. Thank you.
I would request for the Momento presentation, please. Yeah.
Thank you, ma'am. Now, I would like to request the honorable chairpersons, Dr. Alok Saxena sir and Dr. Aruni Saxena sir, for presenting a memento to the speakers of the session. And I request Professor Dr. Shubhra Dasidar sir and Ms. Anushri, she's already here. I request sir to please come on the dais. Now, I would like to request Professor Dr. Deepak Tewatia sir and Dr. Shishir Nandi sir to present the memento of the chairpersons of this session.
Thanks once again, honorable chairpersons and speakers for this session. And Anusri is a big symbol of very smart and young speakers. And now it's a time for taking lunch. I request all the dignitaries to please have a quick lunch. And during the lunch session, poster session is also included. I would request Dr. D.K. Dixit sir, chairman of the poster session, and honorable members are Professor Dr. Subhra Dosidar and Professor Dr. Abhijit Oja sir. So these three professors, Dr. D.K. Dixit, Professor Subhra Dosidar, and Professor Abhijit Oja sir. So you are the judge for evaluation of the offline posters, and I will hand over the evaluation sheets. So please have a moment, sir, single moment, I request you.
So welcome to all, and moving forward to commence our fourth scientific session, I would like to invite Professor Dr. Indra Ghosh ma'am and Dr. S.K. Raj sir to chair the session. Professor Dr. Indra Ghosh ma'am is working in JNU since 2009. Please, ma'am, and sir, please come on the dais. Indra Ghosh ma'am is working in JNU since 2008, and she's Dean and Professor to steer the School of Computational and Integrative Sciences, which deals with the computational approach of biology, chemistry, and economics. Dr. Shrikant Kumar Raj sir is a Chief Scientist and Professor at ACSIR, CDRI. He is a highly skilled researcher in molecular and cell biology with over 30 years of experience in academic research and leading various scientific projects. Over to you, sir.
Well, we like to start the session, but seems the students at least haven't come. So please request the students to come back quickly. And already the speaker is waiting online. So our first speaker is Professor Athena Geronikaki. She is from Italy, sorry, Greece, and yes, and she is quite well known in most of the QSR committees, and internationally also she has held conference in the QSR. So her topics of research is that mostly in the designing innovators. Basically, she is a medicinal chemistry, then turned into the field of molecular medicines and others. So if you can, I think that will be online. So let us check with her and then go ahead. She is already there. Athena, how are you? Nice to meet, and we can start then her talk. Okay. Athena, are you there? Can we hear her? We are unable to listen. Yeah, we don't hear you. We don't hear you. No. Maybe mute. Switch on. Something. Your slides are there. Yes, but your voice is not there. Yes, slide is coming. Voice is not coming.
Are you muted? Please unmute you. Again it is muted. Athena ma'am, it is muted.
Yeah. Please do switch on. Please unmute. Kindly unmute. Yeah. Yes. We can see you. Yes, we can see you. And we can listen. Okay. Okay.
You can hear me?
Yes.
Okay. So may I start?
Yeah. Yes. Yes. Please start. Please start your talk. Yes.
Good morning, good day to everybody. First of all, I would like to thank the organizing committee, especially my good friend, Professor Anissa Xena, and Sister Nandi for the kind invitation that we talk in this conference. And also I would like to thank the chairpersons, especially my good friend Indira Ghost, for their presentation. I'm going to speak today about thiazole-based benzodiazole-based thiazole derivatives as potential SARS-CoV-2 main protease design, synthesis, and biology. Before starting my talk, I have to say that I am medicinal chemist and not computational chemist, but I use these tools for my research. I cannot change my slides. What is going on? This year, I cannot change my slides.
Hello. Ma'am, can you hear me? Ma'am, please do one thing. First of all, open your PowerPoint presentation and then go to the slideshow.
I don't hear you, Cecile.
Hello. Am I audible, ma'am?
Yes, I'm here, not very good.
Okay. Ma'am, please open your PPTs
Yes,
and then go to the slideshow.
wait a moment.
There is an option of the setup slideshow. Browse by an individual. And then share.
Now it's moved. You hear me?
Yes.
But you see my screen?
No, right? No, ma'am.
That's why I don't like the online presentation. It's always a problem. Now you see me?
Share all the screen.
You see my screen?
Ma'am, you can share your all the screen. Your complete screen, you can share that. When you will click the share button, then you will see different type of the windows. There is a PowerPoint presentation window, ma'am.
I don't know what. Now you see?
Yes, yes. Your screen is visible, ma'am.
But I cannot move the slides.
Ma'am, go to the slideshow.
What?
Slideshow.
Slide off?
Yes.
There is no indication on the screen.
Ma'am, lower right corner, ma'am. There is a window for the slideshow. Near the plus and minus zoom button.
Can you hear me? It's Aruni Shobhit here. So if you go your cursor down, yeah, go down the stairs of the temple. Go down. There you would see plus 71 minus.
It doesn't move.
No, it's moving. We can see.
It doesn't move. I cannot change slides.
Oh, you can do one thing. You can also use down button on the keyboard and it goes to other slide.
Keyboard doesn't move anything. I use keyboard.
Yeah. I see. Yeah, like this.
Yes, yes,
Yes, the slide is changed.
Okay, ma'am.
Yes, perfect. So, during my talk, I will point out an introduction, design of compound, synthesis, biological evaluation, and conclusion. In December 2019, a novel virus outbreak was first reported in Wuhan, China. Despite many attempts to contain it, the virus, named severe active respiratory syndrome coronavirus 2, spread worldwide, causing pandemic. This virus belongs to a family of RNA viruses, namely coronaviruses, responsible for human respiratory tract infections ranging from common cold to severe pneumonia. Coronaviruses are highly diverse family of enveloped positive cells, single-stranded RNA viruses. They infect humans, other mammals, and other species, including livestock and companion animals, and are therefore not only a challenge for public health, but also veterinary and economic concept. This ongoing COVID-19 pandemic is today responsible for more than 6.7 million deaths worldwide. SARS-CoV-2 is transmitted via respiratory droplets, infected cases to oral and respiratory mucosal cells. And here we see what kind of symptoms this SARS-CoV-2 causes: fever, weakness, headache, stroke, dyspnea, rhinorrhea, anosmia, asphyxia, and many other symptoms. Despite significant progress in COVID-19 disease treatment, effective therapy for managing the novel coronavirus disease long-term complications are still missing. COVID-19 is now found to affect also other parts of the body and except of respiratory system. There is now available vaccine which show high efficacy in preventing serious respiratory complications. But continuous mutation of the virus reduce their effectiveness. So the development of safe and effective drugs to treat active forms of COVID-19 remain options. In order to fight any virus, not only SARS-CoV-2, somebody should know his life cycle. So the life cycle of SARS-CoV-2 consists of 9 stages. The first one is virus entry, either through the fusion or endocytosis, followed by viral RNA release. And the third step is protein synthesis, including ribosomes viral RNA to form peptide chain. And in the third step, there is glycolysis with the L-protease, followed by RNA replication, subgenomic transcription, and several-step translation of viral structure protein, followed by viral packaging and viral release. At this time, when this disease appeared, scientists didn't know anything about this and have no weapons how to fight with this. So they turned their attention to already approved drugs for other viral diseases, such as, for example, HIV. And so here you see some drugs which were quite successfully used to treat COVID in the beginning of this disease: chloroquine, remdesivir, lopinavir, and so on. And here we see the molecular pharmacological mechanism of ongoing potential therapeutic agents for management of COVID-19. So there is a drugs which were used, their mechanism of action, and also it is shown on what step of the viral replication they work. All enzymes and proteins involved in viral replication are potential druggable targets in research for therapeutic options to SARS-CoV-2. And the critical targets for antiviral drug development include the spike protein, the M-pro protein, the papain-like protease, and the dependent RNA polymerase. Among the limited SARS-CoV-2 molecular targets for treatment of SARS-CoV-2 disease, the MPRO has emerged as the most important for the development of antiviral treatment against SARS-CoV-2. This is a key enzyme involved in viral application by cleaving polyproteins, which are necessary for the formation of functional viral protein. Besides, this enzyme have high degree of conservation among coronaviridae family members, making it invaluable target for development of pan-coronavirus direct-acting antiviral. Moreover, a binding site of this enzyme is highly different from that of human cysteine proteases, allowing development of selected inhibitors with reduced toxicity. According to crystallographic studies, SARS-CoV-2 main protease is homodimeric protease with two subunits, 1 and 2. Each subunit has a length of 306 residues and formed by 3 domains. The first domain includes 8 to 100 residues, the second 101 to 184, and the third one 199 306. And here we see the location of lopinavir in active site of the enzyme. It's a screen like this. The catalytic diad that perform proteolysis cleavage formed by residue cysteine 41 and cysteine 145. Taking all this into account, our aim was design and synthesis of compounds that could be effective of SARS-CoV-2 main protease and potent drugs for antiviral treatment. Actually, the present research is a combination of traditional medicinal chemistry, structural biology, and computational chemistry. In particular, this compound designed based on the following structural features and interactions provided to enhance their activity. It is known that SARS-CoV-2 main protease cleaves its substrate after glutamate, which follow lectin and before a serine or alanine or glycemic amino acid. The analogs of the substrate we used to study the structure of main protein. The results showed that amino acid lectin is placed in vicinity to residue cysteine 41, while glutamine is placed in vicinity to cysteine 145 residue. As I already mentioned, these two residues form catalytic diad that is essential to SARS-CoV-2 chemotrypsin-like protease activity. Cysteine 41 is function as a proton acceptor, while cysteine 145 has a nucleophile. So, the achieving potent aminic affinity requires a molecule that will interact strongly with these catalytic diad residues. Therefore, a molecule that could act as a mimetic and interact with at least one of these residue, catalytic residue, might strongly increase its enzyme. Taking this into account, and based on our previous results, we show that the Zabianone moiety seems to be to mimic a glutamine amino acid, and furthermore, oxygen atom of the Zabianone ring can form the hydrogen bond with cysteine 145, and also that fluoride, the presence of fluoride substituents increase the inhibitory activity because they form electrostatic interaction strongly than H1. We include, keeping these two moieties in our new design molecules, we include benzothiazole moiety and aromatic rings because due to their hydrophobic nature, they can form PP and interactions. Continue building our molecule, we add sulfonamide group, which is for the reason to mimic the amide linker of the natural substrate, and which probably would interact with the rest of 45. And finally, we iterated our molecules with a variety of substituents to enhance their distribution. What actually we did? We designed compounds, make prediction of their antiviral activity by computer program parts about each Professor Faroikov, and after this, we made dosing studies in order to select and pro-inhibitors for further research, to choose the compounds which show which will strongly inhibit SARS-CoV-2 and PRO. Actually, molecular docking studies are powerful too for rapid discovery of hit compounds for clinical use. Their key contribution is their significant reduce of time and cost, mainly for emergent disease such as COVID-19, and the ability to speed up the analysis of target interactions with drug candidates. After this, we made automated prediction because it's very important for the drug development process, and choose and choose 15 compounds for synthesis and in vitro evaluation. The same we did with the second group of compounds, which is 85, and the same that they follow the same process. The structure of SARS-CoV-2 and PRO with PDB code 6LU7 was used for molecular docking studies on virus designed as a genome derivative. All compounds re-docked as racemate, and the best conformance we selected based on the RMCD values and the estimated free energy of binding. So that's out of 85 selected compounds, 15, the most 15 show free energy ranging from -1054 to -741, while initial inhibitor has a dot score 7-7.7. For the selection of the best conformers, was influenced also by the number of intramolecular interactions, which such as hydrogen bond formation, aromatic or hydrophobic interactions, which accept by docking the compound in the active center of the enzyme, and especially with the cysteine 145, make cysteine 41 and glutamic acid 166. The next step, as I told you, was the evaluation of prediction of ADMET properties. One of the most critical challenges for oral medication is the ability to cross intestinal epithelial barrier, which controls the rate and the extent of human absorption and consequently their bioavailability. Bioavailability is very important point because many drugs, many projects fail, even the compounds has very good activity because of very low bioavailability. So the CACO2 permeability assay was used to predict how well the administered drug will be absorbed. A value of higher than 8 multiplied 10 minus 6 centimeter nanosecond indicates good permeability. With the exception of compound 7 and N3 inhibitor, all compounds have higher values than this threshold, indicating good permeability. The positive values of compound 1, 2, and 11 shows that they can be transported across the cell membrane by the ATP binding cassette transporter, which is component of the LP glycoprotein. The value of distribution indicates the extent to which a drug is evenly distributed throughout the body. A value of distribution below 0.15 log suggests low distribution, while the values exceeding 0.45 log indicate high distribution. In this context, compounds 4, 5, 10, 11, 12, and 15 are considered high value of the substitution compound. With the exception of compound 10 and 15, the log BP values for all compounds indicate low blood-brain barrier permeability. Similarly, the majority of compounds exhibit low permeability to the central nervous system. Metabolic prediction suggests that all compounds above substrate and indicator of KIP2B6 and KIP3A4, compounds 2 to 5, as well as reference inhibitor N3, give default hepatotoxicity, while the other, they are not toxic. Here you see the tables which indicate the water solubility, kappa permeability, and all what I spoke about a few minutes ago. All compounds be computationally evaluated for their drug likeness and the availability score, and the results show that the availability score for most of the compounds was 0.55, with no violation of the Lipinski rule of 5, and in combination with their drug likeness scores, which range from minus 0.24 to 0.49, it can be concluded that they have good oral bioavailability, except of compound 14, which has bioavailability score 0.15 and drug likeness score minus 24, and have two violations from the Lipinski rule. Finishing with this prediction and the work which it's needful before starting the synthesis, then to the synthesis of this compound, which the first group of compound was synthesized by 3 steps, starting from 2-amino 5-adamantane thiazole or 2-amino benzothiazole derivative, which reacted with fluoroacetyl chloride to give fluoroacetyl derivative, which in turn, in the presence of ammonium thiocyanate cyclides to give the thiazolidinone derivative, and the last one, upon reaction with the different aromatic aldehydes, gave the target compounds. Here you see the structure of compounds, 15 compounds chosen among the group of benzothiazole and thiazole-based thiazolidinone derivatives. The second group of compounds, where we included sulfonamide group, they are synthesized by 1-pot 3-component reaction using conventional and microwave system methods to enhance the yield and decrease the time of the reaction. And structure of 15 and other 15 compounds which they are chosen, you see here. Since we finish with the synthesis, compounds be characterized by different physicochemical properties, and they are to prove their structure and purity, and after this they went to biological evaluation. Evaluation of SARS-CoV-2 and-PRO inhibitory activity was assessed by fluorogenic enzymatic assay, according to which peptide substrate exhibits low fluorescence. Then protease cleaves the substrate and breaks the rigidity of addons and doublets. So we take 2 moieties, non-fluorescent addons fragment and fluorescent the terminal addons moiety, which highly fluorescent addons fragment increase in fluorescent signal, and this increase in signal is proportional to the protease activity. So, according to the obtained results, for the group of benzothiazole-based and thiazole-based thiazolidinone, 3 compounds showed to appear to be active. Compound C1 with IC5476, K3 with IC50-0010 and N2 with 99084. Compound K3, was the most active, being showing IC50 value lower than compound non-reference drugs GC376. The evaluation of second group of compound revealed that 5 of them were active, 7, and among these 5, 2 compounds showed IC50 value better than reference inhibitor. This compound 12 with IC50-0192 and compound 7 with IC50-0234. Talking study of compounds of benzodiazole, of group of benzotiazole-based diazolidinone derivatives revealed that P3, this one magenta, binds to the enzyme in a similar way as a reference inhibitor GC376, the diazolidinone ring, being placed at the S1 subsite in the active site of the enzyme, where glutamine is naturally placed between residue glutamine 166 and catalytic 15 1. The oxygen atom carbonyl group forms hydrogen bond interaction with cysteine 145, while the nitrogen oxime group forms hydrogen bond with glutamate 192. Furthermore, the aromatic ring of benzotiazole and aromatic ring with fluoride forms hydrophobic interaction with plenty of amino acids, thus enhancing the stability of complex enzyme ligand. And here we see the structure of the reference drug, and again we see that oxygen atom of carbonyl group takes the hydrogen bond to cysteine 145. Compound CN and N2, CN is purple and C2 is green one. The compounds of the same group with K3 have got the same orientation inside the enzyme, but the compound N2 interact only through this enzyme moiety with the 25 and 1827, while compound C1 interact hydrophobically with both the ring and benzotiazole with plenty of amino acids, which stabilize more the complex of enzyme and ligand. This absence of stability in compound N2 maybe is the reason of high IC50 value except for compound N2, which is N998 compared to C1, which is 4736 respectively. For the evaluation of hepatotoxicity compounds, they are tested in the human normal fetal lung hemoplasm NRC5 cell lines. Each substance was tested in 3 different concentrations, 0, 1, 1 and 10 micromolar. After the incubation period of 24 hours, no notable negative impact was observed on these normal cell lines and at the 2 lowest concentrations. K3 is the most promising compound and it exhibits the highest protease activity with IC50-001 micromolar and affects cellular reactivity more than 57% at this concentration after 48 hours in culture. But at the highest concentration of 10 micromolar, compound 14 and 15 exhibit the highest activity. But overall, we can conclude that this series of compounds toxicity allowing to select the most promising compound for antiviral evaluation. The in vitro antiviral activity of compounds 1, 4, 7, 11, and 12 was assessed against 2 SARS-CoV-2 variants of concern, Delta and Omicron strain, was assessed in their E6 cells simultaneously evaluated with evaluation of their cytotoxicity. Remdesivir as potential as positive control exhibit no cytotoxicity at the highest concentration tested at 100 micromolar and demonstrated equally pronounced antiviral activity against both Delta and Omicron SARS-CoV-2 variants with a range of the IC50 in 5 micromolar. Our compounds too exhibit no cytotoxicity at the highest concentration test. Although compound 12 is one of the most active compounds of the second series, is the most active compound of the second series of the IC50-0192, it is devoid of antiviral activity against Delta strain. On the contrary, compound 7, which was the second more active compound, showed with IC50-0234 micromolar, showed moderate activity in ranging between 15 and 35 micromolar EC. Compound 11, which was the least active as a pro inhibitor, showed weak activity against both strains, EC50 90 and 71, against Delta and Omicron variant of concern. Finally, compound 1 and 4 not only did show any enzymatic activity against Mpro enzyme, but they lack the antiviral activity. The molecular diagram simulation study for the top of compound 12 and 7 revealed that compound 12 consistently exhibits strong binding affinity and robust interaction with essential residue of the enzyme. You see here the data of this. At the end of my talk, I can conclude that in this study we synthesized 30 compounds as possible inhibitors and properties of SARS-CoV-2 out of 197 totally designed compounds. The analog with the lowest binding energy is selected for biological evaluation. The most potent analog we endorsed with IC50 values of less than 1 micromolar in this enzymatic assay, and this series of compounds induced low cytotoxicity relevant to select compounds on the viral evaluation. And showing the reactivity of compounds K3, 12, and 7 exceeds that of reference compound GC376. Additionally, compound 7 shows promise as an inhibitor of SARS-CoV-2 replication in 0E6 cells. According to docking studies, compound IP binds to the reference inhibitor GC376 and molecular docking simulation study for the compound 12 and 7 reveals that compound 12 consistently exhibits strong binding affinity and robust interactions with the essential of the enzyme. So, in a way, we reach our goal. And to close my talk, I would like to thank my collaborators, Dr. Petru, my postgraduate, my postdoc student, Dr. Pisapidou, my PhD student, Professor Gennady Kulik from my university, Professor Rungrat Mongol from University Ceylon of Hong Kong, Professor John and Scholz from the University of Lincoln, and Professor Makaev from Moldova. And thank you all of you for your attention.
Anyone wants questions, maybe questions can be asked, please. Hello? Yes, so questions? Anyone wants to ask question? Is there? What are the students? Have you heard it at all?
One question from my side. Ma'am, have you tried with nanomole concentration? I see.
Micromolar.
All micromole, right?
Yes.
So are you planning to some modification and something so that
My nanomolar.
I have a question, Ethena. That is because this is very similar to the human. Also, the human has also the same type of proteins. So, have you checked for selectivity also? Yeah, the compounds what you have. So, whether they bind to human cell line or not. Hmm?
Yes.
Yeah. Okay. Okay. That's what. So, okay. Thank you. So, yes, thanks a lot, Athena, for coming and having. And Professor Anil ji wants to. Anil ji wants to have a question. Please hold on. Sir. Yes, Anil Saxena wants to ask question, Professor.
So it's a very nice presentation and I think good work. I congratulate you. And this I want to ask that when you have the docking studies, have you made docking for all compounds, whole series?
Yes, we did, Anil, we did, thank you for the question. We did docking for all compounds, all 197, and choose the compounds which have the best binding affinity to the and properties.
Okay, good. Thank you.
Thank you very much, Athena. And yes, it's nice to meet you and see you here. Yes.
Through the screen, it was nice to see you after 1 year.
The next, yes, next, there are 3 speakers, but next speaker, I'll just say Asmita. She's going to present here. So I request Asmita, please check your slides and can be presentations. Okay, please. Thank you. She's from Kalyani. Next to you. Okay, so well, going on, Ashmita is one of the young scientists upcoming scientists, I would say, working on nanobiology, and she's working at Kalyani University. And she and her team has many successful work, and she works very much with Dr. Shishir here in Jaipur, hence quite familiar face to most of the students. So Ashmita, are you ready?
Is it okay?
Please carry.
Is it
Thank
okay?
you. Thank you. Have that voice. What do you use these?
Okay, slides. Good afternoon, respected chair, the organizers, Professor Saxena sir, all the faculty members and all the dignitaries, my students. It's an honor and privilege for me that I've been invited, and I feel it not only for myself but also for my scholars who often visit Jaipur, because a part of my work is happening in Jaipur and the other part is happening in University of Kalyani at West Bengal. So I'll be giving a short talk on strategic formulation and evaluation of nanophytocompound for therapeutic management of diabetes. So, coming to the background, actually, diabetic is a serious metabolic disorder, and India has been the diabetic capital of the world till last year, when it has become second. It has become second right after China, and around 101 million of people are being diagnosed with diabetes, and over 136 millions of people are diagnosed with pre-diabetic condition. So the count is huge. There are several orthodox medicines with their own scientifics and problems, and people are sometimes more inclined towards the usage of phytomedicines, which also have dosage issue scientific validation. So with the advent of nanoscience and technology, we have been trying to, put forward these selected phytocompounds, and the selection is done through in silico molecular docking at Jaipur, and with the selected phytocompound, we formulate the nanoforms and we undertake the preventive application of this phytocompound in cell line as well as is in animal model. So we choose the phytocompounds which have structural similarity, not 100%, but some structural similarity with the regularly used drugs for diabetes. And we choose the phytocompound from the lap of mother's nature. The phytocompounds that we choose based on they're easily available, we try to make sense with that. They are cost effective. They have some reported anti-inflammatory, antioxidant, and anti-genotoxic role. And with that purview, what we do is that we make drug repurposing. Next. These are some of the data for the usage of the compounds. Next. And the phytocompounds that we have been used in many cases, like for cytotoxic, anti-cytotoxic and anti-genotoxic effect against pesticide toxicity. Next. In fish model. So we have now created a drug repurposing to understand whether it has the efficacy against preventing or halting the progression of diabetes and its complication. So with this objective, the first objective becomes screening of the phytocompounds based on similarity with the original drug substances that are used regularly to treat diabetes, then to formulate the particles in a polymer which is approved by the FDA, which is non-toxic and biodegradable in nature. And that polymer is polylactide co-glycolic acid. After that, we characterize the particle that whether they are in the nano range, and then we try to elucidate whether this particle are actually affecting the mice model or the cell line model. That means in experimental model, we use them and we see whether the target proteins that are already done in the in silico, whether they are actually modulating these proteins so that there is a kind of prevention or inhibition in the progression or its complication of diabetes. So coming to the methodology, this first part of the methodology is done in Jaipur, the in silico part. The second is the characterization part, and we have to undertake many sophisticated tools like UV spectroscopy, FTR, AFM, DLS, SEM, TEM, encapsulation efficiency. We undertake the drug-drug interaction by circular diachrosome spectroscopy. We then understand the bioavailability, which is very important, and then the overall biological parameters related to diabetes and its complication. So this is just a representation of how we formulate the drug. That means there will be a cage of the polymer and a drug will be inside. And this cage will dissolve or get biodegraded when it has been administered in vivo or in vitro model. Next. So the strategic formulation of the nanoparticle are made by the standard protocol of Facey et al., which was there from 1989. Here we have used stabilized pluronic. This is PLJ, that is the polymer. The phytocompound of choice is used, and this is how the particle looks. That means there is a cage, the capsule, and the phytocompound is within. And then we administer in cell line and in mice model. We have to undergo several ethical issues of animals, and then we can undertake these experiments. These are the parameters that are used for the characterization part, and these are the parameters that are being used to elucidate the effectivity of the nanoparticles against the disease. Coming to the observation section. Now, this is the 2D and 3D demonstration of the nanoparticles by atomic force microscopy. And this represents the size of the nanoparticle. The FFT data shows the uniformity of the particle that are being formed. The more particles are closer to the center or their eccentric, the more uniform particles are being formed. We also undertake the dynamic lag scattering to understand the charge of the nanoparticles. The negative charge of the particles helps greater internalization of the particle when administered in vitro and in vivo model. This is another particle that are being used. Actually, I have given data for 2-3 particles. So this is another particle. This is curcumin. And the 2D, the 3D, the FFT, these data shows that there is a uniformity in the formation of the nanoparticle and the particles are in the nano range. The DLS data is also negative. This is the FESIM data, which shows the purity of the particle that has been formed. Now, the essence of the nanoparticle has two senses. First, whether it is in nano range, and the second, whether in the capsule the actual particle is present or not. And that, because we are dealing with nanoparticle. And what is the range? It is 10 to the power minus 9, and you cannot see it with open eyes. We can only see it through sophisticated instrument, or we can see this, we can visualize it as an agglomerated form. So we have to characterize whether the actual component of the particle is present within the polymer. Here, the FTI data shows there are the respective peaks of the curcumin. This is the representative data. And the similar peaks are present in the nano-curcumin, which represent that, yes, we wanted formulation of nano-curcumin and we have the actual curcumin component inside the PLGA. Now, after administration, it becomes very important. Say, if we are administrating in cell line model, we are administrating in vivo model, that is mice model, it is very important to understand whether the particle that is being getting inside the model, that is in mice or any other organism, it is there totally, or it is, or the metabolites which are useful to control or inhibit the progression or the onset or initiation or the complication, whether the metabolites are present or the particles are present or not. That means component of curcumin or component of the phytocompounds are present or not. So we have taken the LCMS data of the tissue homogenate from control mice of liver, next, then curcumin fed mice, liver, next, and then nanocurcumin treated mice, and we found that yes, the metabolites are present in the liver tissue homogenate. Similarly, next, we have done it in the same process for the pancreas, next, the curcumin fed pancreas, next, the nanocurcumin fed pancreatic tissue homogenate. We have also undertaken XRD study showing the respective peaks for curcumin. Now, this is a very interesting study. There are two consecutive slides. First slide shows the circular diachrist spectroscopy, which directly shows whether the particle has the capacity to bind with the DNA, and we take the cuff thymase DNA as the model. And from the data which is shown here, we find that piperin and nanopiperin, both of them bind with the DNA, and there is an elicit of the intensity of the band in the spectral data. Now, interestingly, in the next slide, for the curcumin, it is very interesting to see that curcumin, definitely, yes, it is binding with the DNA. But for the nanocurcumin, it is binding with a greater efficacy, but at the same dose. The dose was similar. That means from this, we presume that, okay, the nanocurcumin or the nanophytoproduct has the capacity even at the dose that we are using for better binding. And for the dose, there is a calculation for the faciatal protocol. The dose automatically, after it is being formulated, the calculated part is 1/10 of the dose is being used for a study. We also undertake some study for the blood-brain barrier, and we found that the TEM study shows that the particle could cross the blood-brain barrier. Next. Now, this is a very interesting part. The bioavailability study for the phytocompound in perspective of the days concerned and in perspective of the tissues concerned, and we find that yes, they are available in different tissues in different time intervals. Now, this is another important perspective for nanoparticle study, because look, nano is in a shell. Now, if after being administered orally or through injection, we have done the oral administration, if the actual drug component cannot come out from the shell, then it is of no use. So we have to anticipate, we have to understand, we have to verify experimentally whether the actual drug component comes out of the PLGA core, and we found that there is a phase after which the drug is being released. And from this data, it is evident it is not a burst release. It is a suspended release. That means all of the drug is not being released just pick. It is released in a suspended manner. So that's the beauty of the nanoparticle that we have formulated. This is the encapsulation efficiency. So the more the drug is being encapsulated, the more it will be, the loading will be high and the capacity will be low. The more the drug is being loaded in the capsule, the more the capsulation efficiency will be less. And we have found that the expected entrapment efficiency is around 86 to 90%. Next. Now we undertook some of the diabetic parameters like blood glucose level, media glucose level, glycosidated hemoglobin, and insulin level. Next. And these all are measured in the either in the mice model or in the in vitro cell line model. We also undertook the bioavailability test in the cell line model, and we found from the TEM imaging and the fluorescence imaging that the particles are going inside. Particles are seen, and it is going after a time interval. It is moving inside the nucleus as well. Next. This is an interesting study that we did with limonene, and we find that the diabetic inducer that we used, that was alloxan, it could morphologically, the architecture of the cells were degraded, and this limonene pre-treatment had the potential, of restricting the morphological disorientation that was caused by alloxan. Next. We also undertake the mitochondrial membrane potential studies. Next. The cytochrome C study. Next. These are protein study. Now, previous one. Now, this is the genotoxicity study that we undertook in the mice model. Alloxan induces several, these are chromosomes. These are the chromosome of the mice. Alloxan could alter the normal shape and size of the chromosomes, and we found that the curcumin or the nanocurcumin and the nanoparpirine both had the efficacy to halt or inhibit the extent of chromosomal aberration that was happening. Next. Similarly, this is the comet assay showing that alloxan could impose a lengthening of the comet tail, which directly depicts that there is a chromosomal, there was a DNA damage. But in case of the nanoparticle, the damage was controlled. Similar results were also obtained for the DNA fragmentation assay. Next. Now, this is the tissue architecture. This is for the kidney tissue, and we find that the curcumin had the potential of inhibiting the morphological changes in the tissues that were circumvented by the alloxan induction. And the halting of the tissue architectural changes could, in a way, inhibit the complication, diabetes-induced complication, but that was that of kidney tissue. That means complication related to like diabetic-induced kidney disease, those can also be halted or inhibited. We also checked the generation of ROS in both the pancreatic and the hepatic tissues, and we found that the nanoparticles had the potential of, modulating the generation of ROS. Next. We undertook some mitochondrial membrane potential study and immunohistochemistry. Next. And as predicted by the in silico molecular docking study, we also understood whether like P53 protein and other proteins were also screened and found that the curcumin or piperine or limonene had the potential of targeting some of the proteins. And we checked whether it is actually happening in the mice model or it is actually happening in the in vitro model. And we found that there was an alteration in the p53 protein, and which might be that this might, this is a DNA repair protein, and overexpression of the protein might, play a role in inhibiting alloxan for beta cell damage or any other cellular damage. And this is the essence of, getting a party view from the in silico data and doing and seeing actually what is happening in the experimental model. Next. This is the intensity profile. And finally, we undertook the RT-PCR and the Western blot studies. We undertook the study with the GLUT4, that is the glucose transporter, with the GLUT2. This is another glucose transporter. GLUT4 is present in the muscle cells, and GLUT2 is present in the pancreatic beta cells. So, coming to the conclusion of the study, nano-encapsulated form of the drugs that were being chosen by the in silico molecular docking, where they were nanonized, I should say, in a polymer that is biodegradable, non-toxic, and they have an increase of drug penetration, bioavailability, thereby having better efficacy than the unencapsulated counterparts. The drugs can then be easily and safely used as a protective measurement for the therapeutic agent at a 10-fold reduced dose. That cuts the cost effectivity that were imposed on use of some finest phytocompounds available. And the oral route was found to be very effective, which is also a new way that we have done. Presently, we are in collaboration with AIMS Kalyani, and we hope for some work preceding our preclinical work. Let's see what's best, so that we can give a little better life for the pre-diabetics. We are concerned about diabetics also, but we are concerned about the pre-diabetics, because all these phytocompounds are readily available, like piperine is available in pepper, curcumin is available in turmeric, limonene is available in the lemon or citrus. So the pre-diabetics who already have been diagnosed, like 186 millions or 156 million in India alone, they get readily transformed. Any chance they will readily get transformed into diabetic stage. And if we can halt that or delay that onset, with this concept, we started our journey with the in silico coupled with in vivo and in vitro approach. Next. The work with the nanopiperine was published in Scientific Report in 2024. This is a Nature publishing group. Next. The work with nanocarcumin was published in Colloidal Surfaces, Physicochemical and Engineering Aspect in 2025. I gratefully acknowledge all the funding agencies that have given me grant so that I can conduct the work. And definitely I'm grateful to Jaipur for providing the in silico molecular docking workstation. And I'm grateful to my University of Kalyani for giving me the infrastructure and the opportunity, and my laboratory for doing the work. So this is my part of my lab. My Dr. Nandi visited my lab, and they are my research scholars, without whom I don't know whether any product would have come out. And Rishita has, Professor Nandi is co-supervisor of Rishita. Presently, she could not come today. Sorry. And Arnab is here. He's the next person after Rishita who will take the baton to the next. Next. And the group of scholar, and I'm always indebted to my professor, Professor Anishur Rahman Khudabaksh, who was my PhD supervisor, and he has been the top 2% scientist from the last 5 years. I'm always grateful to him and to Almighty. Thank you so much.
Thank you. Any questions from the audience?
Not only diabetes, I think there is a wide range of diseases that were not at all diagnosed or that did not come into diagnosis. I'm telling, not only diabetes, years back, the concerness of the people were not, I think, that much. And if not, uh, going to a doctor, I have seen my grandparents. I was lucky to see my grandparents and his father also. So they were so much reluctant to go to visit to a doctor, even if the doctor came, did not sit with them. So as of the little knowledge, whatever I have, I can only say that as years pass on, my father has become a little bit of concerned about the thing that okay, death has to be diagnosed. I do not disagree with your point that my grandfather wasn't no, I have to fetch a doctor, I have to see a doctor. But yes, the state of the art way that the patients are being going the concernedness of going on fetching a clinician has increased, and that is why the diagnosis, the data collection has increased from every part, from every part. Hmm.
Arun, I think it is a very big debate we can start. Uh, yeah. Because diagnostics things
It
were
changes,
different.
it
Okay.
changes generation after generation.
Sir, let us take another students' questions.
Yeah, I would like the students. Look, this was a very lucrative thing. You have seen all the hands-on experience. Yes, sir.
We'll discuss. Mhm.
No, I'm not telling before and after. As per the protocol of FACI et al. 1989, suppose, means that's a calculation. It's completely a calculative part. Suppose we are taking X mg of the drug. Suppose we are taking X mg of curcumin. After the nano has been formed with everything, well, the polymer and everything, we have found, suppose YMG. We have calculated that each YMG of the drug has 1/10 amount of X. That has been calculated. And while administration, we make sure, like if we are administrating, say, A amount of nano, we are administrating A amount of the phyto compound only. So whatever effect we are having, like if you are having a similar effect also, that means the phyto compound is with the A amount and the nano is with A, actually having 1/10 of the actual drug component in it. Yes. Are those drug? No, we use it first and foremost for the dose. We use it in the cell line. It is microgram per microliter or ml, whatever we find it in the range finding trial. For the mice model, we have to put it in mg per kg body weight. So it ranges with the drug finding trial. So it is in the mg per kg body weight for the mice. Per kg body weight. So mice is 40 gram only, and we are 80 kgs or 90 kgs or 70 kgs as for human. So in that, for the in vivo model, we take mg per kg body weight, and for the cell line, we take it for the microgram. We have found it from the range angle. No, no. First and foremost, we have screened it through in silico first. Second is that we do the immunoblotting, we do the PCR study, and we understand whether it is binding with the glucose transporter, it is affecting or not, whether it is affecting the P53, whether it is affecting the insulin receptor or not. These are the targets that we see, and we also verify whether it is corroborating with the study that we get result from the in silico molecular docking studies.
Okay, let's I'm sure that there would be many other, because this type of delivery system are very new, and questions will be more. I'd be very thankful if you can talk to her. She's going to be here, and all of you want to ask her questions, because we have to go through the next speaker.
Yeah, sure.
I have a question.
Yes.
What is the positive control you used for genotoxicity study?
The positive, first and foremost, we had to eat metformin. We have done.
But toxicity, toxic.
So the dose that we have used, the dose that we have.
Metformin is for diabetes for positive control, right?
Yeah, for diabetes.
But for positive control for toxicity, genotoxicity, whether it is chromosome is altering or
So
not.
we have used alloxan as a diabetic inducer. So we know the dose at which it will promote genotoxicity and at which it will promote diabetes and genotoxicity as a whole, 12 rule. So we have taken the dose so that we have standardized the dose so that the diabetes is being controlled and the genotoxicity is being controlled.
Okay, thank you so much. Let us invite the next.
Thank you so much.
Next speaker, that is 10th speaker of the day, Dr. Shishir Nandi, who is known to all of us perhaps in this stage. I don't want to further give introduction about him. Please, Shishir. This is
Pointer. Job. Hello. Thank you. 10 minutes. I will take. So I would like to take permission from Honorable Chairperson. Yeah. Thank you very much, sir. Thank you very much, ma'am. And I thank my organizer for giving me this opportunity to explain this topic, to explore the chemopreventives against colorectal cancer utilizing a docking simulation in this international platform. You know, early in the morning, when we get up from the bed, if our bowel movement is clear, we feel that we are the happiest person in this world. It depends on regular activities, exercises, intake of good food, fruits, vegetable. But we are the persons doing mistake in our life, having great affinity towards intake of spicy food, chili food, oily food, fast food, intake of excess amount of the red meat, smoking, drinking, alcoholism, intake of sweets become obesity, fattiness, lack of exercises, long sitting work under pressure in banking, post offices, software companies, lack of sleeping in the night, long gap in the food intake, improper dieting, narcotic drug addiction, drug side effect, lack of drinking water. So these unhealthy factors cumulatively pressurizes our GIT, and GIT disturbances occur like flatulence, indigestion, gastritis, irritable bowel syndrome, constipation. So now, what is gut pressure? You know, this is the human gut. It contains favorable collection of microorganisms that is called microbiome that maintain the ecosystem of the gut. Under the gut pressure, gut stress, it become inflamed, this layer become inflammation, loss of this layer, loss of the microbiome. And then when the intestinal tight junctional protein getting inflamed and lost, loss of microbiome, and colorectal diseases occurs. Now, you see, I can show you some experiments. You see this picture? Farmers often, very often, spray pesticides, insecticides in the field for a long time without wearing the mask. After the work, they are feeling problem like GITs. What? Vomiting, then nausea, lack of food intake, irritable bowel syndrome, gastritis, constipation. What is the reason? Does this pesticides molecule, insecticides molecule goes through the mouth and nose and attack the gut? Does inhalation, ingestion kill the microbiome? This is a question. I assigned this work one of my M.Pharma students. You see, Yakult, commercially available suspension. What? It contains Lactobacillus casei. MRS broth has been used to grow. This Lactobacillus colonies grow, and this colonies, it is diluted, turbidity produced. And by using simple UV visible spectrophotometer, the student take the reading, it becomes 1.287. Now you see, in this experiment, the pesticide data contains copper oxychloride and thiophenate methyl. These are useful to protect, to kill the pest. Yeah, and these are used to protect cabbage, cauliflower, potato, tomato, chili. Very often these are used. Now, in very dilution, 10 to the power minus 3 dilution, this test compound has been added. And this, again, after adding the test compound, the UV visible absorbance of these two compounds were taken. And you see, these are the absorbance rate. For copper oxychloride, 0.323, and for thiophenate methyl, it is 0.563. Now, the control absorbance is 1.287. So from 1.287, it becomes this decrease after adding this test compound. So easily we can say that there is a killing of microbiome using the in vitro test. Now, this work has been published in the Springer. This is the work of my M.Pharma student. Very easy work, very easy. Next.
Now you see, second experiment I am telling. Have you seen this picture? Food additives we are using in our daily life. You see, like wheat flour, red chili, curcuma, sugar, color sweets. Do you know what does it contain? Coloring material, food additives, sweetening agent, food additives, preservatives, stabilizers, food additives. So cumulatively, excess intake of food additives, it can attack the DNA. Do you know what would be the side effect? DNA, gene toxicity, gene aberration. Now you see, these are the sweetening agents: disodium glyceride, sunset yellow tartrazine, propyl gallate, alloxan. These are the used. These are very useful as food additives. You know, wheat flour means this maida, atta, it becomes whiteness, multigrain atta. What does it contain? It contains alloxan. Do you know regular use of this kind of food additives, what will happen? Next. Now you see, in an experimental model, this alloxan has been treated with the mice. This is the control, blood sugar level 112, and after administration of alloxan 207. So it becomes diabetic. Percentage fluorescent cells was tested. For alloxan, it is 89.45%, and for control, it is 52.55%. So intake of alloxan, it becomes diabetic. And now this problem is trying to get solved by using the natural molecules, by using the two formulations of this pelarogolidine. This is a natural compound. Formulation 1, formulation 2, and you see the blood glucose level tends to normal, decrease. Now, GNA aberration, chromosomal aberration test was done in our experimental collaboration laboratory with Osmita. You see, this is the control, and these are alloxan treated, and now these are drug treated, means PG, pelarogolidine, with two formulations treated. And here you see gene aberration was more, chromosomal aberration was more, it becomes less because this is the natural molecule treated. This work getting published in European Journal of Pharmaceutical Sciences. This is the PhD work of our student who is Ritita De, is working under my supervision, co-supervision, and Osmita supervision. Now you see why colorectal issues happen. You see, this is our gastrointestinal system. You know, there is a good talk between lungs and gut, good friendship. Who is giving the friendship? Gut microbiota, it is responsible for giving the friendship between lungs and gut. So, you know, when gut is under pressure, microbiota is getting lost. Communication is getting lost between gut and lungs. Lungs is under pressure. Automatically, heart is under pressure. So, total digestive system, including the main machine of the human body, destroyed. Next. Now, you see, what would be the long effect? You see, anal fissure due to passing, due to pass of hard stool, anorectal orifice getting ruptured, inflamed, bleeding occurs. Flute-like appearance happens, that is called fistula. Now you see, piles, inflamed veins. We are very common, these problems are very common. Fecal impaction, some of the material of fecal material, stool material getting inside the colon, who is constantly in touch with this ruptured vein or inflamed area, and then it becomes, what will happen? Burning sensation happens, bleeding happens, infection persists. It causes, what it will causes? Abnormal expression of the proteins and genes, tends to colorectal cancer. Next. Now you see preventive measurements. So when a patient is suffering from cancer, no answer. But before getting attacked by the cancer, What would be the preventive measurement? Shall we have preventive measurement? Yes, I have preventive measurement. What? You see, we can avoid this kind of food I have shown you, like chili food, spicy food, we must have to avoid. We must have to avoid this kind of, we must have to take this kind of food, fermented vegetables, you know, dahi, buttermilk, pickle, we must have to take because it gives favorable environment. It helps to grow the microbiota inside the gut. Intake of the fiber food, you know, roti, like papaya, ripe papaya, mango, pineapple, apple, guava, dates, orange, strawberry, cucumber. Intake of sufficient amount of the water to decrease the colon pressure, easy defecation of the bowel. Now, when a patient is suffering from this kind of problem, anorectal problem, doctors suggesting the siege bath with alum. This is true. And also some stool softeners like milk of magnesia suspension, liquid paraffin. But you know, long use of this milk of magnesia liquid paraffin have some limitations. Now, next, doctor suggests lactulose along with antioxidants like vitamin A, C, E, N-acetylcysteine. Experimental findings said that these 5 components are helpful against cancer, but there is no biochemical mechanism explained till date, molecular mechanism. So in our laboratory, scientists like me targeted these 5 components and tried to explain the molecular mechanism utilizing in silico modeling, in silico software. Next, you know, so KRAS G12C, this protein is getting abnormally expressed in colon cancer, and this protein has been targeted, and these components are taken as a ligand to bind the active site of this protein, whether having binding affinity or inhibition. You see, this is sotorasib, standard drug given for the treatment of colorectal cancer, and this is the active site. All these 5 components are getting bind oil in the active site of this sotorasib. Next. So this is the standard drug mode of binding. These are the amino acids. Next. You see, this is lactulose binding, vitamin C, N-acetylcysteine, vitamin A, vitamin E. Next. Now, most interesting point you see, lactulose, vitamin A, vitamin C, N-acetylcysteine. These 4 components, this is the alignment of the 4 components along with sotorasib. This, and when vitamin E is added, you see some of the groups of vitamin E is deflected from this alignment zone. What does it indicate? It indicates that, but some of the groups of vitamin E is getting deflected, but it is bound in the active cavity. Now the question is, how does it fit? It fits oil. It indicates it has something special mechanism of action, and the study said, you see, this valine 9 and histidine 95, these are the 2 more amino acids which are present in this binding. Next. So one of the component lactulose was taken into molecular dynamics simulation study, and this is the report of molecular dynamics study, and the complex is fit oil. Now you see the video. No? Okay. Next. Now the conclusion is, my message is, we must have to take a lot of natural antioxidants to make us healthy in our daily life. We must have to avoid our bad habits. This is my conclusion. And next, you see, our experimental lab is in West Bengal, and I am very much happy that my authority of the institute, management of the institute gives permission for going there and doing work with Asmita. Next. And you see, they are my collaborator, Professor Dimitri from Russia, who came in the last meeting at Dehradun. Dr. Aruni from Nottingham University. Asmita, my elders, my sister. Amit Rai from University Pune, Savitribai Phule University Pune, and Dr. Hare Krishna Rai from Nirmala College of Pharmacy, Guntur. Next. You see, I acknowledge the Water for Welfare Government of Uttarakhand for giving grants for the research, and you see how I can express my thanks and gratitude to the teachers. I don't know, because there is no substitute for mother and there is no substitute for teachers. And my Guruji, academic Guruji, I am very much happy that my academic Guruji is Dr. A.K. Saxena, and my academic Guruma, Dr. Midula Saxena, and Dr. Monish Bagchi. He is my parent teacher, my guide, PhD supervisor from IICB, and my parent teacher, Professor Kunal Rai from Jadavpur University, and Guru Brahma, Guru Vishnu, Guru Deva Maheshwara. So they are my Guruji, my Guruma. Next. And how can I express my gratitude to my students? Because we are the teachers, we are able to see on the eyes of the students. They are my active students who are always doing the work in the laboratory, And this year, 2025 is very happy for me, happy new year for me, because my management approved 34 lakhs BioVia software in this year. I am dancing, my hat is dancing. And these are some in silico expertise with us in our laboratory, and I am the very expert person in this field. Next. And really, our students, they are getting publication in high impact factor journal like Nature Group. And you see, these are the some publications in 2024-23. These are all collaborative works. Even M Pharma, B Pharma students, they are giving publication in Springer, in GPAR. And now I really salute my mother India. I feel proud that I am India. India is great. Thank you very much.
Thank you, Sisir. Wonderful presentation.
Please,
There
sir.
is only
Please.
one question.
Yes, sir. सर गेहूं का रोटी दिल खोल के खाइए आप. Sir, I can give you the answer now. You see, now ecotoxicity is a big issue. Ecotoxicity, whatever, yes sir, whatever lifestyle diseases are coming due to environmental pollution and ecotoxicity, and our very bad practice, we are the citizens responsible for it.
Okay, Arun, let us stop here. Any student, one question? You don't want to ask your teacher? Thank you, Sisir. You really presented well, and teachership full bloom. Thank you so much. Congratulations. So next, we will invite the next
Rajan.
speaker, which is Aruni Saxena. Please.
Thank you. Yeah. Take it in. Okay. Can you all hear me?
Yeah. Good afternoon, everyone. First of all, thank you for a lovely presentation, Dr. Nandi. And thank you, Chairman, the board, for inviting me on this occasion of speaking on this interesting topic. And the session itself being on cancer. We had few sessions before, which was on AI applications. Now you would see in this presentation, potentially food for everyone, whether it is a software person, whether it is a clinician, or whether it is a drug designer. From the clinician side, we have a big team. I only mentioned 2 names right now, but we would be the main presenter. It's Dr. Jitesh Arora, who is online joining from UK. So he is the part. And of course, I would take the lead. Specifically on the radiological scans, Dr. Jitesh would be taking up that as it's part of his expressive learning and expertise. So going ahead, not wasting any time, this is the title. Refractive hypercalcemia and neuroendocrine tumors. There's a discordance between histology and imaging and tumor progression. So just as I know, it's a variety of audience. We have people from life sciences from different areas. Tumors, I think everybody understands. It's cancer. It's a common way. Whether a tumor could be a non-cancerous one, it can be a cancer. That's a different sort of histology. But at this point of time, I would make it very basic. So we have a type of tumors which we call neuroendocrine. And why they are called endocrine is because they affect the hormones. Any tumor, potentially it might be in an endocrine gland or maybe outside, but my cells might behave in expression of excessive hormones, which create symptoms. And that's one of the problems with that when it comes for diagnosis, because people always think, oh, it's a hormone problem. Next slide, please. So if you see the incidence and the epidemiology, you won't be hearing about neuroendocrine tumors a lot as when you compare with lung cancer, colon cancer, breast cancers. But still, there is estimated 6.98 incidence notice per 100,000 persons in a year. If you see epidemiologically, it can happen in any part. Now, when I'm saying any part, well, any cancer can got a metastasis and can go to other parts of the body. So a lung cancer, when we say it is metastasis or it's a stage which is final, everybody for common use, it can reach your brain, it can reach your bones, it can migrate. Here, the peculiar part is this cancer, neuroendocrine, mostly can happen anywhere. You are seeing it's in gastrointestinal tract, it might happen in pancreas, it might happen in lung. But then it's labeled as neuroendocrine tumor of lung and not a lung cancer. So that's the difference. And that's a very rare peculiarity of these tumors. Next slide, please. So here it's for anatomical distribution. If you say it can happen in lung, if you say it can happen in pancreas and small intestines, most of the cases are noted in the gut system. So again, Dr. Nandi, your presentation links with me because here also you can have a type of cancer in your gut region that could be small intestine, it might be pancreas, which is most often seen in clinic, although other are also present. Next slide, please. So key features. Now, I'm not a histologist, but just for the purpose, I have made a slide with where you can see something. Origin is neuroendocrine cells, that we should label it when the definition comes, because they are present in different organs. And then WHO classifies it to grade 1, grade 2, grade 3. Ki-67, just mark it for now. You would see its application AI later on. Ki-67 is grade 1, grade 2, grade 3, depending, and it is a prognostic index. You can say how many, so basically cancer is the uncontrolled growth of cells can be cancer. And this shows somehow the proliferation index, which is the growth index of any cells being multiplied. So logically, if it is more, it would be bad. That's why it's poorly different. Next slide, please. Now, these are the, as I mentioned early on, why we are saying at neuroendocrine and why they become potentially problematic to get diagnosed at early stages is because they are involved in the release of some neurohormone secretory products, which normally everybody has. So for example, It could be somatostatin, which is related with growth, or a glucagon, for example, which is regulated for your blood sugar. So you have insulin, which helps in controlling the level of blood sugars, where you have the antagonist of its glucagon. So if that suddenly increase, you are again getting a risk of hypoglycemia, hyperglycemia individually. So initially, most of the diagnosis happens in endocrine, or endocrine specialist comes to know something is not very right with this person. And then we later on come to know that person has a cancer. It's not a hormone problem. So that's the thing. Common ones are when they are happening in the GI tract, their maximum incidence we have seen of serotonin going too much, which causes flushing, dilation of face, redness. They are typical things. Long sustained diarrhea, not getting controlled by any of the antidiarrheal medications easily. Non-infective form of diarrhea, that could be the symptoms. And these are, you can say, stimulatory and inhibitory. In this slide, when you say they are inhibitory neurotransmitters, they are stimulatory neurotransmitters. So in a very layman language, any neuroendocrine tumor, stimulatory might be high, or even inhibitory might go high or low. Next slide, please. Risk factors, of course, genetics comes into play. You would later on see there are genetic syndromes which are linked with it. Multiple endocrine neoplasia, von Hippel-Lindau, chronic inflammation, again, it's a pros, and then smoking, alcohol, hormonal factors, environment or the ecosystem toxicities is also one of the risk factors. Next slide, please. This is something which is important and might be a point for learning, because everyone nowadays, if you see, cancer is like a heart attack. And there are reasons for it. The reasons is not because the incidence has gone high, of course it is, but the diagnosis and access to medicine and health equity has increased when we see it in the Asian 50 years before. For early signs, one should always remember, if you're having flushing or like diarrhea, always think it might be also cancer. So don't neglect it. If you have hypoglycemia, hyperglycemias, and your diabetes is not under control, your HbA1c is, in spite of everything, getting too high or too, mostly in a high case, it could be again cancer. Parathyroid hormones, the topic of the day, patients comes with a high calcium, and I would explain that. My calcium levels are always high, somebody says, that's not right, it could be cancer. So these are the early signs. The late signs, of course, cancer is known is a deadly disease just because it is a stealth. It's like a stealth burma, which nobody can detect on radar. And they often come to us in clinic when they have jaundice, bone pain, or they have just breathlessness. But then we come to know that breathlessness was not because of the heart or lung problem, but in fact, it was a cancer. General warning signs is, of course, anyway, if somebody has a weight loss, which is unexplained, if somebody is having sweating in the night, which is unexplained, if somebody have fatigue, loss of appetite, abdominal pain, or having problems with digestion, which people tend to neglect, thinking it's bad fooding habits. It could be, of course, that is. You should be always be thinking of cancer in your background. Next slide, please. So this is WHO system, which was for the classification of gastrointestinal neuroendocrine tumors. I had already shown you the previous one, so I would go running over it. There is always a Ki-67 index and the mitotic count. So for students, mitotic, you must be hearing from your cell cycle. So that's a similar, if the cells are multiplying too rapidly, it is counted as a mitotic count. Next slide, please. So here it comes for the staging. So we as oncologists, we always, for us, the most important part is what stage a cancer is, because that would determine all the things. What is the treatment? What is the prognosis? What are the possibilities? A general system is TNM. I've just mentioned it for the students to have an idea what it is. So TNM is used in every type of cancer, in which T stands for the tumor, whether it is penetrated, how much it is size, what's the growth. N refers to lymph nodes, if the lymph nodes are affected. If yes, how many? And of course, on that, we use NO, NX, NO, N1 regional. And then M stands for metastasis. If any cancer has reached other organs, it's certainly M1 organ. If it reaches 2 organs, it may be M2. And after that, we don't need any grade because we know that it's gone. If you consider why it is important, I explained in stage 1 to 2, a surgery could be an option without requiring anything else to be done. Stage 3 could be site of a multimodal treatment. What we mean is surgery plus chemo plus radio, or maybe chemo radio. Stage 4 is metastatic. Now, what is specific for neuroendocrine is we have surgery as an option, and we would be that if it gets detected in a very localized area, surgery would be the first part to be done. Somatostatin analogs, why? Because somatostatin tends to be the most common neuroendocrine transmitter, which is in excess state causing problems to the patients. So we use it octreotide or lanreotide for that. Then nowadays, you must have heard of new things coming up like immunotherapy, biologics, targeted therapy, what that means. So you have a target therapy that is for progressive pancreatic. Normally, it's more or less most of them are targeting tyrosine kinase, which is supposed to help in proliferation. Theirs are inhibit those so that the cell cycle doesn't happen anymore, or the growth restriction is there. Classically, we use chemotherapy most, carboplatin etoposide, but the problem here comes to side effects. Carboplatin are platinum-based drugs with irreversible side effects causing neuropathy and also having effect on kidneys. Similarly with etoposide, apart from general side effects of vomiting and other loss of appetite and diarrhea. New thing which is coming up nowadays, which might be a new term for you, is PRRT, it is peptide receptor radionuclide therapy. Now, this is very much important because this thing is coming up. It's now being involved, in which we are giving a radioactive substance in form of tablets with binding to the receptors which we are using for chemotherapy. So you're using a two-way approach. You tend to bind it on the receptor and take the radionuclide directly there on the site. It has got a very promising results, although it is quite expensive. There are challenges with it. And then, of course, you have supportive care at the end of the day if you can't do much. Next slide, please. So these are the novel targets, if you see, which I have already explained. So I would be going a little bit fast on these slides. So you have tyrosine kinase inhibitors, which I already told. Sunitinib, cabozantinib. Main common thing, we need to control the blood pressure. All the patients, it affects the proliferation of the blood vessels. It changes its supply to the tumor, but on the same hand, it causes problems with the blood pressure management. So there are challenges with it too. Then you have chemotherapies, the other ones which have been involved is capecitabine and temozolomide, the more common one which I'm using. You have immunotherapies. Now, immunotherapy is coming up or being studied now. Problem is, of course, all immunotherapies can cause inflammation in anywhere, in any part of your body. Those are irreversible. And the only medication we have to treat is steroids, and that too at very high dose. So that remains a challenge. And then, of course, the novel compounds, which I have said, peptide receptor radionuclide. Next slide, please. So now it's a case, a real world study. Interesting one. We had a patient, 62-year-old. I personally saw him in the clinic. He was presented with hypercalcemia and acute kidney injury. So for non-medicals, I can say hypercalcemia is either the calcium levels are too high, comes to the hospital, he was vomiting. Acute kidney injury, we say when the kidney functions are very bad, in very bad condition because of the electrolyte imbalance. Because it is reversible, we say it acute. If it is a chronic one, it means it has been for a long time and we cannot change the functioning of anymore. Now, what first things doctors do is they do a scan. I mean, they do the blood test for sure, which I have not mentioned, but radiology, they do the scan and they see that there is a goiter in thyroid. There's something in the liver, not yet known what it is. We do a PET scan, which shows some high uptake in sigmoid colon. That means tumor might be sitting in colon somewhere. Now, till this point of time, we don't know whether it is a neuroendocrine tumor, whether it is something else. Histology helps doing it. That's why biopsy is always important. And that shows poor differentiated Ki 67 with index of 30%. We come to the conclusion, of course, it's a neuroendocrine. And then we give carboplatin and etoposide, which is the standard one, as I mentioned. Partial response seen. We planned for 4 cycles, but partial response was seen, and recurrent hyper. Now, only clinical sign which was coming was recurrent hypercalcemia. Next slide, please. This is the timeline, if you see. What were the early signs? Hypercalcemia. We do a CT scan in May 2024. We do an ultrasound guided biopsy that was done in June 2024, in which you say metastatic grade G3 neuroendocrine tumor. Then we did a octreotide scan that was in July 2024 and planned with chemotherapy. We repeat in August, FDG PET scan, biopsy, and surgery. And the surgery was for parathyroid gland to be removed and thyroid to be removed. And then in December, chemotherapy was stopped. We did a scan to check the parathyroid tissue remaining, and these scans would be told by Dr. Jitesh, my colleague. Next slide, please. Over to Dr. Jitesh.
Thank you, Dr. Saxena. So I'll be talking a little bit about the imaging that this patient has had, with the first being a CT scan of the thorax, abdomen, and pelvis, taken with IV contrast in the portal venous phase back in May of 2024. So here on this slide, we can see two images. On the left, we see an enlarged heterogeneous thyroid gland. So what I mean by heterogeneous is you can see different shades of gray where this arrow is, showing different densities within the thyroid. And on the right, bilobar liver metastases. So the liver has two lobes and metastases present in both of these. However, no primary lesion, however, was able to be identified on this CT scan. Next slide, please.
Next slide, please.
Because of this, this patient was then subsequently referred to interventional radiology, or what we call an ultrasound-guided percutaneous liver biopsy. So this is a biopsy of the liver taken through the skin. The histology was then analyzed, which showed metastatic grade 3 neuroendocrine tumor. So that's quite an aggressive neuroendocrine tumor. As Dr. Saxena mentioned, a Ki-67 of 30%. And it was also what we call CDX2 positive. Now, CDX2 means that the tumor expresses the CDX2 protein, which is a marker of intestinal differentiation, suggesting that this tumor is likely coming from either the gastrointestinal tract or a pancreatic origin. The patient also had a biopsy from a colonoscopy done in August of 2024, which showed tubulovillous adenoma, and on review, whilst it was initially a well-differentiated neuroendocrine tumor, later confirmed what to be a poorly differentiated neuroendocrine tumor with a Ki-67 of 30%. Next slide, please.
Next slide, please.
So the patient went on to be discussed at what we call a multidisciplinary team meeting in July of 2024. And it was decided that this patient would require an octreotide scan and what we call an FDG PET scan, as it was quite a high-grade neuroendocrine tumor. Now, on this slide, we can see the octreotide scan this patient had in July 2024. So just a little bit more information about what an octreotide scan is. It uses nuclear imaging to detect somatostatin receptor expressing tumors and involves the intravenous injection of what we call tetraetide, which is a radio-labeled somatostatin analog. That's where you can see highlighted on this scan in red. Unfortunately, once again, no primary tumor was identified. However, the liver metastases were avid for tetraetide. Now, when I say avid, I mean they had a high uptake of this radio-labeled somatostatin analog. Notable other findings included a new right lung mass seen on a scan, which is not indicated on this image here, as well as some low-grade uptake in the thyroid gland. So in the previous slides, we showed that the thyroid gland was significantly enlarged. It once again had low-grade uptake of this radio-labeled tracer. However, it was deemed that this is likely to be from a pre-existing multinodular goiter rather than a metastatic neuroendocrine tumor going to the thyroid itself. Next slide, please.
Next slide.
So the patient also had what we call an FDG PET scan in August of 2024. This is once again a type of nuclear medicine scan involving what we call intravenous radioactive glucose, FDG. So cancer cells are typically more metabolically active than other cells in the body. Therefore, they take up more of this radioactive glucose sugar. Essentially, you become radioactive and are detected by the scanner. And that's what you can see highlighted on this scan in bright. So it showed extensive metastatic disease in both lobes of the liver, as well as in the right lung as well, likely coming from what Dr. Saxena said earlier, a primary tumor within the sigmoid colon itself. So we underwent a colonoscopy, as we mentioned previously, which was then analyzed. Next slide, please. Now, I include this slide because we've spoken a lot about this thyroid that was enlarged, and Dr. Saxena has also mentioned this patient had high calcium levels and also high parathyroid hormone levels, and these cause chemical disruption in the body, and the patient presents with varying signs and symptoms of these. For that reason, the patient in August of 2024 had their parathyroid gland, their thyroid gland fully removed. However, we're still having parathyroidism. Essentially, their parathyroid hormone levels were still raised in the body. So the surgeons wanted to look for any remaining gland tissue from the parathyroid glands. So this scan was requested, a nuclear medicine scan of the parathyroid. And as you can, so imaging was performed at 10 and 90 minutes post-injection of a pharmaceutical tracer with SPECT CT taken in the delayed phase. And as you can see here, there was no focal uptake of the radiopharmaceutical to indicate a parathyroid adenoma, and there was no residual parathyroid tissue seen either. Next slide, please. So I've just included this slide just to give basically an up-to-date reference of the most recent imaging this patient has had, taken last month in January of 2025, a CT scan of the thorax, abdomen, and pelvis, once again with intravenous contrast. Here, as you can see, the patient has lost their sort of subcutaneous outline of fat on the CT scan, showing, the patient's had severe weight loss from the cancer. There's severe disease progression with hepatomegaly. So essentially the liver has become enlarged with innumerable metastases. We can see metastases all throughout the liver. It's impossible to know how many there are. The vast majority of this scan is taken up by the liver itself, which has then displaced some of the other organs, such as the bowel, and also caused compression of the intrahepatic inferior vena cava also. I'll now hand over to Dr. Saxena to talk a little bit about the rest. Thank you.
So thank you. Thank you, Jitesh. Next slide, please. So as we see in this real case scenario, we had some clear learnings and of course some complications which we came through. The biggest one was that neuroendocrine tumors, which we were thinking could be controlled, but at the initial stages was in fact an advanced one with a really poor prognosis. Now, mechanism, you came to know that surgeons went in, they removed the thyroid completely, they removed even parathyroid completely, thinking that would control the calcium levels, because that was causing patient to have vomiting. He was having dry mouth. He had extreme weight loss in later stages, not able to eat just because of that. Even that did not change the course of disease. And the mechanism possibly could be that they have a parathyroid hormone-related peptide secretions, which we want to test, but the test is 6 months of duration, and I don't think our patients would be having such a prognosis. So that was the other cause. Potentially is the tumor lysis. However, it is not applicable in this case scenario because the chemotherapy which we planned to be given for 4 cycles could not be completed much because of the electrolyte imbalance and poor state. We could not give the last 2 cycles to the patient. Management challenges. Of course, we went to endocrinologists. We thought, okay, we cannot try to find where the parathyroid is. Maybe that this tablet Sincalit, this is a classical medication which we use for hyperparathyroidism, can be used to suppress somehow PTHRP, which helped us to a certain extent. The calcium levels did come down, I would say, but then need regular monitoring because it went too low sometimes and once too high. We tried to search in the literature for other similar cases. I was just a bit curious if some other cases have been reported. There are some cases, one reference we could find, but not many. And then prognostic significance. Now, what we can see over here in this case scenario specifically is hypercalcemia could be for prognostic significance. But of course, we have only one case. And now I'm going further down. Can the next slide, please? Yes. So as I said, generative AI, what I did is, and I tried to say, okay, let's try to do using generative AI. If I can program and know the patient prognosis initially, because the key challenge is here that what is the problem statement? So for anything, if you want to use an AI, or any site of modeling, and AI Gen is the common thing, you need to identify your problem statements. Computers are dumb. They cannot understand your language if you put them straight. So you need to understand what is the key thing which you want at a very microscopic level. So that's why I said sudden change over well to poorly differentiated neuroendocrine tumor, why, and can we predict it? Why it happened? So somebody says, why I developed a cancer, right? Of course, we have number of risk factors. Few have been already mentioned: environment, ecotoxicities. Some is, of course, man-induced. Smoking is man-induced. Alcohol is man-induced. You are taking artificial things. You say it in a general ecosystem. And then, of course, genetics was one of the most important ones. And I think there are some unknown viruses which remains unidentified that could be behind, which I believe, because few cancers we were able to identify. So there are a number of factors, okay? So genetic mutations I took into account because we know there are genes TP53 and RB1, which is known, which could be associated. There's microenvironment changes. Of course, hypoxia interaction with stroma cells, that might be. Then is therapeutic interventions, such as might have some sort of prognostic significance, right? And of course, as an oncologist, when I sit with the patient, the main thing is choices. First of all, if the patient is fit for treatment and what he chooses, because there's no yes and no clearly, in the most case of cancers and advanced stages, right? So treatment. I don't know how many of you do Python programming, but few who are good with who are burgers. So I did a Python programming, simple ones, to set, like I don't have any data set, right? So, but I need to identify what to do with it. And then I come up with this equation of log P, beta 0 plus beta 1 x1 till beta n xn. So you have the probability of transformation that you want to catch. You have factors, this is b1, b2, bn, this is coefficient of predicted variable, right? And then you can take it up to n, in which we can use predictor genetic mutations, microenvironment factors. Next slide. So if this is the Python code which you get generated, I don't know how many computer buddies are left. Few have left now the sessions. That was for more interest. So if you do with Python code snippet, you get XY predictor transform at constant sin. So this is the program. This is a logistic regression model for which you can start using this code. Next slide, please. Yeah. So as I said, the main thing is we don't have much data, but expected predicted accuracy could be quite good if we have that. And then how it can influence in progressing in determining prognosis and what are potential advantages is you can use it for prognostic stratification, first of all, then personalized treatment plan and improve patient counseling. So right now, most of the things which we do is, of course, evidence-based. But these cases which are very rare and which are sort of a very rare possible cause, it becomes a problem because we don't have the evidences too much. And of course, it may be a step in because we have the regression Python model. If people get feeding the data into it, which of course remains a challenge, I do accept it, then potentially in the clinic, once the patient moves with this kind, I can show him what are the chances, how much are the chances that the histology findings which was there in August, how it can differentiate till December. And I think Dr. Jitesh Arora explained it, that histology was taken 2 times in this patient, because we also thought when we were not able to control the cancer, we might have diagnosed wrong, or not diagnosed wrong would be a bad statement, but in a sense, we underestimate the potentiality of the cancer. So cancer tend to change also even when you start treatment. I need to tell the patient how much time is left because most of them want that answer. Of course, nobody can give it. But with generative AI, if I use this, I can increase the efficacy and the treatment predictions to a very high extent. Of course, the data remains a challenge, but it's possible. Next slide, please. So these are the key takeaways. I have summarized it. It's that it is what are the early symptoms, what imaging we can use. Refractory hypercalcemia is a thing which we have noticed in our case. Tumor heterogeneity is a problem which we saw. Initially, tumor was in a different histological state, and that was well differentiated, went into poor differentiated ones. And then ongoing research, what are the personalized treatment we are having, or undergoing research, and generative AI might have solutions for it. Next slide, please. Okay, there's a references. Next slide. Yeah, thank you. So I come from University of Nottingham City Hospitals. And this character, I don't know how many of you know about this character. Who is it? It's Robin Hood. I don't know how many people know about Robin Hood. He was a person who used to take what you say Robin Hood of medieval period, to take gold from rich for poor. So I come from city of Robin Hood. It's Nottingham. This is the University of Nottingham. Our department is in City Hospital, and this is QMC, which you see, the Queen's Medical Center. It's one of the third largest hospital in UK, with 4,500 beds. Thank you, everyone. Now, any questions?
I suppose, yeah. Is that? Don't get in the place, but I must see when the moment is based on very then you can expect from the generative AI, this is very difficult. So, if you want to use my voice is quite large. So please do take home this message. If not the medical message, I'm supposed to be, yes, and very limited. I'm really surprised with a very limited test. You come out with absolutely corrected. Probably this is the only way the patient can survive, So, when you face traumatic scenario, which nobody faces, the doctor with this much knowledge and having understanding. So this is one of the best presentation, I will say, in this session, where we learned how to use even the dumb computer. So great thanks to him. And any questions anyone has, please. Any questions? Yes, sir. You have? One question.
No. I think ask Dr. Ashmita, do you have the question you asked? Yeah, he wanted us to go that question.
I think for generative AI, available data is posing constraints. But do you think with increased integration of system, computer data, accessibility data, and real-time basis, can this problem be addressed in near future?
I'm very hopeful. Like there are many people who are starting thinking in the similar ways. Of course, it needs extra time and incentives, I would say, initiatives to be taken. But I think in next 10 to 15 years, in field of oncology, particularly because we always work with a lot of patient data, we do share it. It is possible. Not in all sort of medicines, because some of the like blood pressure management and things, but because things are saturated. Everybody knows most of the stuff, but in oncology specifically, it does stands, and hematology too. Yeah.
Running out of time, so that's why I would have discussed it with the outside.
No problem.
So I was just wondering, I'll talk to you later, means what about the multi-parametric and physiological perspective of other diseases that does not follow a single route?
Yeah.
So how can this AI give a mental peace, or at least make the patients little distress, means lower the distress that are anxious and anticipate in this scenario? That's my question. I can discuss with you.
Sorry. No, I can give you the answer, and we can discuss in the break too. And it's a very common problem, and that's why here the AI helps you, because we have been using multi-parameter regression analysis since childhood. At least for me, it was nothing new for me, and maybe it's because of my father. I knew it from when we were driving car, he's discussing about this model, other models, right? Of course, I'm not a medicinal chemist, but I know it. You have applications, but if you remember, at least for me to sit and make a logistic regression, I need hours of work together. I have to sit 10 hours continuously to really take the things out. But if you know Python and AI and the phosphor, you can reduce your workload. See, you have, if my equation, you see it's bx, x till n, right? N, and then you can say it till infinity. So the number of influences, what you're, I mean, the problem here in the medical science or life sciences, you have so many microenvironment factors, age, BMI, and I don't know what blood group, most of them are, that it's unlimited. It's unlimited data points, if you think. But now you cannot use everything in your, so you have someone to help you to just reduce it in a fraction of a second. And of course, you have to do it. Then afterwards, you have to test, you have to do the data set, you have to do the test set. It might be completed, but that time is reduced. It's the same way like my father used to use scientific calculator. Imagine using scientific calculator, calculating for each equation your R value, how much time it would take. For me, it's so much simple. I simply use SPSS, MATLAB. It is done in fraction of a second, right? But then, I don't know what you are using, but then you use with AI tools. So you can use them to reduce your workload, and you have to, the process is the same. The process is not different. You have to have the data training set and everything.
It was very enjoyable to, chair this. And on behalf of both of chairs, I think I really thank all the speakers for keeping more or less we managed to get in time.
Okay, thank you.
Thank you very much all.
Now I would like to request honorable chairpersons. Honorable chairpersons to present the memento to the speakers of the session, for which I would like to invite Dr. Smita Samadar ma'am, Dr. Shashi Nandi sir, and Arun sir is already on the stage. And I would like to request Rajan sir to show the e-memento of Professor Dr. Athena ma'am. Thank you, sir. Now I would like to request Professor Dr. Deepak Tewatia sir and Professor Dr. Alok Saxena sir to present a memento to the chairpersons of the session. Thank you, sir. Thank you, ma'am. So there is a short tea break, and as we are running short of time, so I request everyone to join the conference room after 10 minutes. Thank you. And the chairpersons of next scientific sessions are Dr. A.K. Dwivedi sir and Dr. Arun Kumar sir. And the speakers are Professor Dr. Indra Ghosh ma'am, Dr. Shrikant Rat sir, Dr. Alok Saxena sir, Professor B. Jairam sir, and Professor Dr. Lakshmi Kotra ma'am. Department of Pharmaceutical Sciences, Gurugram University, Haryana. He has authored 5 books and many research articles. And he has been also appointed as the founder, a faculty member of NIPER Raebareli. Over to you, sir.
In this scientific session, we have first speaker, Professor Indra Ghosh, a very eminent researcher, academician of this country, working in a GNU since 2008 as a dean and professor in the School of Computational Integrative Sciences. It's a very famous department of computer science, which deals with the computational approach in biology, chemistry, multiple disciplinary department. She has nourished the school as a career of Center of Excellence under the Department of Biotechnology in the computational biology to initiate a new stream spotted by the UGC central regulatory body of this country called complex system. She has been amongst the earlier scientists to realize the importance of the bioinformation. It's a very long journey of, we cannot read in the simpler words. Eminent professor and academician and researcher, we invite Professor Indra Ghosh, ma'am. Thank you, ma'am. Please.
Yes, so I must first thanks to organizers that they give me the opportunity to speak about something which is not necessarily drug design, but it may help to excel us in drug design. So it's not necessary the topics I will be talking is very much easy for everybody to get, particularly the students, because as you can see, well, this talk is, the title of my talk is, if you can just have, yes, one more click. It's called, you can press, please. One more. Protein function and structure. Whereas most of my talk will be addressed to the young people, the students. And why this is because I chose to talk on this topic. I had a little bit, purpose, I will tell you. Quite a long time, so we are working in the field of drug design, etc., everything. And mainly technical, The experimentalists are doing their work quite well, but most of the theoretical people, enough good theory we develop, but then even beautiful machines. But we haven't been solving real problem of if in a smallest molecule, if little change happens in that molecule, everything goes over haywire. This prediction we are unable to handle. So I thought, why don't we look into the backward? And there was a student also, I'll tell you, the student picture will come maybe very end. He joined, he is a biotech student, and he didn't know much of programming. So nobody took him as student, but I asked him, what do you think you would like to do? He said, I like to predict structure of protein. Everyone likes from sequence to structure of protein prediction. So I asked him this question, and which was bugging me actually. So this question was, yes, given a sequence, how many possibilities of structures are there? Most of you probably never thought that. But this was bugging me really, because given one sequence, can we have many structures? We should be able to have many structures. But when you build structures by all our theoretical modeling tools, boxes, everything, it turned out to be one or two structures.
And most often, if you submit the sequences in the sequence data bank, you get a huge number of sequences which are similar to this. However, nobody has done all the structures, okay? Most of the things which you require, you will go and hunt and find out, no, that structure is not there. Let's say you would like to find a protein, maybe in Mycobacterium tuberculosis. You would not see that in Mycobacterium tuberculosis, but you may find something in Staph aureus, equivalent protein. So what you do normally, you take the sequence, align them, and then predict a structure. And then you are very happy, you take the structure, dock, do everything, and come out with a chemical. This will work. Most often, theoretical people don't have a problem. They just have to find it out. Is there anybody equivalent chemists have done? Hard work of medicinal chemists probably has produced a lot of data, a lot of chemical structures. You just find how good structural overlap it has and publish a paper. Nobody touched that. And your paper actually titles, I found the drug design of anti-tuberculosis, very big word. But unfortunately, we don't have. We haven't really found so good drug for tuberculosis itself. Forget about Staph aureus and other bacteria. I don't have to tell you anything. YKG, we call him always Professor YKG. Experts in all these infectious disease, he told you what a difficult scenario it is. So, to come back, I had this question in my mind. All we do these similar sequences, and then we just run into some software and build the structure. And then we do everything possible. Really, if a sequence given, should it have that structure? Who told it to have that structure? That's my first question. Yes, I know many people, thermodynamics. First, the thermodynamics, physical chemistry, it's the stability, so many folds cannot be done, etc., etc., etc. Some people will say, no, it's a good kinetics. However, we never attempted to find what happened. So this student of mine, he went for doing PhD quite long back. These works are really long back, 2010 to 2015, he submitted thesis and got. So why I'm showing this? Because I realize last 10 years almost, people are trusting, most of the students and designing and compounds, they're trusting too much on this black box. So I thought, let me shake all of you. So my title of the talk is Function and Structure, not Structure and Function. So basically, I like to put in your mind, given any sequences, it is the function which determines what should be the structure, not the sequence and the structure. That's my last slide. Let me go through slowly. So please go ahead, next slide. Yes. So mainly two papers, so you don't have to actually read or take a screenshot. Papers are there, not only that I've handed over my slides also here. We have developed actually now a software which you can probably access by certain typing something, where you can put the coordinates what you want, and then you get actually whatever I've done. Most of the structures will come in that form. Let me go to. And this talk is 100 years celebration going on for last 1 year almost on Professor G.N. Ramachandran. This is also to make sure all of you that Indian scientists who won so many things, so many, he didn't get Nobel Prize, but he got, his things are written in every book possible. And any crystal structure solved, they come to check whether their structure is correct or not by using this map, which is called G.N. Ramachandran and Ramachandran map. Most of the structural biologists, almost all remembers him for that, and it's not India, everywhere in the world. So don't think India doesn't produce great honorable people. India has produced, and he has done this work in Tamil Nadu, Chennai, Madras University, and then he shifted. When he produced all this map, his student, who was my teacher also, he was also my teacher, He told me, Professor Siramakrishnan, we did all calculation in logbooks, slide rule, etc. Hence, please do understand, great science really doesn't need computer. Great science need thinking. So, and he did develop this before even 2 crystal structures were done, only 2, and both of them were myoglobin structure. So everything was right, and I just share with you how good this plot is. After that, lysozyme structure was solved in Oxford, and they sent the coordinates to him, finding whether it is plotted properly or not. And C. Ramakrishnan did plot all, and he figured out only one point is not, only one amino acid is not coming to correct point. So let me see. Yes, if this works, I can you see? Yes, here. There was here one outlier point. So he said, sir, I calculated 3 times. I always get that particular amino acid to fall in this place, which is not allowed. So Professor J.N. Ravichandran sent all these used to be those days, not by email, not by internet, not by anything, downloadable Google, nothing. It was done by telegraph. So he wrote a telegraph that to D.C. Phillips in Oxford, please check your sequences. Imagine, still that time, we just got independence. This paper came out in 1954. So I just born them. So this is what he looked at, not now. Just go back, So I really wanted to give this slide to all of you to remind you, we have a great culture of science. So he challenged D.C. Phillips. D.C. Phillips got Nobel Prize later, but he challenged him. D.C. Phillips took it seriously, sent across Sanger, who also got Nobel Prize, to find out whether the sequence is right or wrong. And after 3 months of struggling, they got that particular amino acid is a glycine. This is made non-glycine. And if there is a glycine actually can cover equal space here also. The reason is glycine is what? It's a symmetric molecule. All of you know. Probably you never noticed. So go back and look at glycine. What is specific of glycine? But I just told you the story. It's not the story. I have heard it from C. Ramakrishnan, the gentleman who was the student that time. And he said, we are very happily sent across the paper and said, let D.C. Phillips correct his sequence and coordinates. Please do understand, this is how Indian science has grown up. So if any one of you think that what we are saying and all, yes, we have a very good foundation. And if we can grow, it's just because our foundation, nothing else. So let me go to my talk. Now, we have been told many, by thousands, millions scientists all over the world, if you have a sequence, you should get a structure. So from this sequence, we have a predicting software. You run the software quickly. It gives you something like this picture. And then you say, well, I want to get the crystal structure of such and such. So you get a beautiful crystal structure. You just map. These parts, yellow parts should be beta units and this alpha helix. So we got all these and very nicely packed structure. So what we talk about, anything changes over there, doesn't matter. You know, main structure should be like that. So sequence decide structure. Hence we have been taught sequence, structure, and then structure decides what function it will give. So be prepared. I'm going to throw this out. Ulta. Next slide. Yes. So I asked my student, as I told you, given 100 amino acid, it's more or less 100 amino acid, how many structures you can get? So he said, wow, how will find it? I said, why? All these proteins have only 2 types of very good structures, rest of them are loops, etc. So find how many possible alpha helix, beta, etc. can. He did work and he said, well, if all of them are alpha helices, then 120 amino acids, then we may have 2 to the power this many ways of making 2 to the 12 ways. And if there are different number of alpha helices, then we'll have more and more possibilities of structure. So this is like combinatorial. So please, next. This is the plot, what exponentially increasing is possible structures. However, protein doesn't have at all this many structures. They only have this many structures. We did study first this many structures available in the PDB. And then we were asked by our own colleagues, oh, you are looking at only the scope structures. Maybe there is another technique by which you modular structures you can calculate. Yes, we did that. We figured out that's also not much. So if you notice, actually, if there are like 3 helices, they are almost falling on how many we can predict. But more and more number of helices increases. Actually, conformers and the structures topology doesn't go like that. They change. They actually don't go many ways. They are folding very less. This was a big puzzle. And my student came back and said, so I don't have a PhD. I cannot go through so many structures. I said, just don't worry. Find why nature choose only these many structures, not so many. And already there were a lot of data people have developed in Europe, in US, and also in Oxford. How many folds are there? And papers were coming like pouring because of certain folds are stable by several systems, several ways. That's why, we don't have many different new folds. So we look at what are folds, etc. So if you can go, what we saw. So my question, always this green space is for you to sort of notice more. So why a small fraction of fold are observed in structure space? This was our question. So every student, I always say, if you have some genuine question, just start your research. But then you have to find some data which you can follow. So I'll go through that. So it's kind of teaching research methodology. Next slide. Yes. Why I thought so. This was a question my student asked. He said, for a long time you were working, and why you asked this stupid question? Everybody accepted this is structure. So there are only that many scope folds. Why you worry? So I said, no. Why are you worry? I'll come to. Number one is this. Look at these structure. These two, this is same molecule, 100% similar molecule. Please trust me, only 2 mutations. 2 mutations at this level happen. Suddenly the structure, and this is where the binding happens of this. This is a repression protein. What its job is to go bind to DNA and don't allow it to go for next level of working. What the trouble is, if any mutation happens, it changes the structure into this, and immediately it loses how to bind, and effectively we get too many DNA being copied. Obviously, cell becomes uncontrolled. So next. So basically, there is a switch, looks like. The switch is 2 mutations, just 2 amino acid mutation. They're consensus mutation. Trust me. But nobody can explain why they just switch to another structure. Nobody. Then comes this protein, which is I choose and look for. DHFR, which is very famous protein for people who are worked on anti-malarial drugs. A long, almost 25 years, this scientist worked in UCSF to find drugs. He found pyromethamine, which binds to it. But the moment he started before even his clinical trial was sanctioned, pyromethamine gone because mutation. The mutation taken care of, pyromethamine doesn't bind at all, low and high resistance. However, please note, there were many mutations. I told you these side chains all are mutations, but structure didn't change at all. Structure stayed. Only mutation happens, pyromethamine cannot bind. Drug designing has been done, huge money spent, no help. These are cases why I was so worried about what is happening, why. Some proteins don't bother, but they don't bind to any longer. And some proteins just change 2 mutations, only small 2 mutations, that 2 consensus mutation. Let me go to the next. I will unfold to you more troubles. So basically, this relationship of sequence to structure is actually governed by function, not necessarily by structure in between. So, and mostly probably is evolutionary, which is everyone knows that happens. The next. ample example. Same fold, if you can look at them, and anyone who does structures and all these softwares available with Shishir sir, just you can take these two and superimpose beautifully those folds. Absolutely same, but totally different functions. You can read. One is alpha lactobin. Its work is different, and this is lysozyme. Actually, ulta work. Lactobin is good. It makes lots of carbohydrates business, but this is not. This is the other end. It breaks the carbohydrate. There are three more, which very obvious picture that shows exactly similar structure. This is growth factor receptor. This is phospholipase receptor. Names tells you what they do. And this is tyrosine kinase. Three. And most of their sequence similarity is less than 30%. Whoever is planning to do homology modeling, watch out. You may get this. Totally different function, same structure. Next one. Next slide shows other one. And yes, just go back. That's interesting. First picture is this. This is carbonic anhydrase, mostly well-known. Initial structures have been found. Same carbonic anhydrase. This is chicken or birds. One, I think chicken. This is human. Their structures are totally different. They have identity less than 30%. And this is called alpha beta role. You can see all the alphas are outside, betas are here. This is left-handed beta helix, much less alpha. But they do same function. Usse bhi simple. Usse bhi simple. These two proteins, everyone knows, subtilisin and trypsin. Any one of you working on whatever, protein digestion. These are the favorite ones. Trypsin is cheapest, so we buy and use it. Subtilisin, little costly, but still we use it. Both of their structures are totally different, but function is identical. They will make basic form. Now, everyone will say to you structural people, oh, those active sites are very preserved. But how they keep it? My question is, in advance, when protein sequence comes out, how they know any different fold, we will keep the active site protein. How they know? Who tells them? That's my question. Thermodynamic doesn't help in that. Okay, next. Yes, so we look into, let us look at the structures, all possible PDB database, and pick up few selective database. This is for the students when you have a question and you are going to work on that topic. First, be sure that you are taking many number of data and variety, not the similar data. Don't take from PDB this, PDB that, no. So I'll show you, and that is the key of your findings, not the finding, the error bar of the findings. And if you do not do in theory error bar, as bad it is the experiment without error bar. So this is the database we choose from all these. Now, first question we all ask this huge amount of structures are coming, but if you look at their resolution of structures, actually not many are having good resolution. There are very less who goes to higher. These are the poor resolution. These are the better resolution. Actually, only 1.8 to 2.2 maybe will get a good and sufficient number of proteins. So we focus that. Why? Because if we don't have a good resolution structures, we cannot do any conclusion. Then the next slide. Yes, we did data generation in 4 different ways. So 1, 2, 3, 4. As you notice, we did some data set which was from 2013, 14, and some of the data set 10 and 11. Why we choose this? Because we figure out that is the year your resolution was quite high. So when you choose data, have proper mind in it. So this 4 data set gave us all the work we do it. Also everywhere I will show you the error bar, and you don't ask then where you got error bar. You didn't do a 3 experiment. We did 4 experiments. Every database we run, everything. Let's go to next. Yes. So question comes, Should we go the scope and Kathy, every one of you should have known and not even known. And now you have BioVia. Oh, you just type it. I want this many. Oh, they will show you beautiful structures. All comes, but they don't tell you how they got the structure, how they got so many small modules which they combine and make the proteins very active, etc. So if we look at, we decided if alpha and beta are the major component of it, instead of following what SCOP says, let us build our own method to identify. And I wanted this method to be so simple, it's like a linear plot. We should not have too many confusion. Hence, we figure out there is a huge structure surge, what you call, I learned this word from one of these guys who presented. Next, same slide have many, so please go ahead. Yes, but you look at this scope, that how many folds are available. Suddenly you see around 2007 onwards, not many folds are coming. So the question may come, where are the folds coming? Not coming. Are people forgotten or they are not doing something? No, folds have gone. That many only protein can have. But nobody asked that question. They said, oh, we'll do more crystal structures and we'll get more. Nothing has come since then. It is, I'm talking about 2000, and this data is 2013. I have a data in 2024. No more new scope. But let's just go through. So why this happened? That was my question. So let us see that. Can we do this? So could not this explain what the topology space is? Means how restricted this structure space. So let me please go through. Also the structure function troubles. So we did this contact screen. Now this is also for students. When you work on any scientific topics, of course, you must have a question. Then you must have some data or generate your data or do some experiments or something. Next, represent properly. If you take the old type of stuff, what already published paper, etc., you are bound to get the old result. So don't be proud of publishing a paper. As the many papers I see reviewed, as it was earlier also found, I found the same thing. Why should you then publish? My question is that, haven't you? The reason not that they publish like that, because they use the same technique, same method. Hence, you should have your own representation, particularly when you work on theory. So this is my representation, what we come out called contact string. Don't worry about this flowchart, all our paper and things there. So basically what we do is just go to the next, please. So next slide would tell you, and we did develop a program which will do all these works for you in Python. It was so simple to work on Python. So that's great. What we did actually was, for alpha helix sample I'm just showing, they have like this cross so many, and it can have parallel all these angles. So every alpha helix, how good they contact and how moved they are, you just plan all these and develop. So next slide, please. You may need to twice because I don't know why this slide is like this, but go to the next. Yes. So from protein structures like this, if you have the coordinates, we developed the contact string. Can you have the next? This is called representation by our own way. Just the next slide. So this is how it will look. All of them will look like this. Just simple, if you see the simple notation. So basically they will be something like this kind of letter. So beta 0, 0, this numbering we have done. And from N terminal to C terminal we have followed. And that's why we have got the contract. Now these are one-dimensional way, representation of three-dimensional structure. Yes, so we go to the next. So developing that, first thing we did, then he applied to helical state, all the helical protein. What we found, this is the number, and you can look at it. What we noticed, there are some helix for every 3 helix, 4 helix, 5 helix, some structures. There are plenty of structures possible. Because we have this contact string, we can make many contact strings. Combinatorially we can have, but out of so many, only few are occurring actually as a functional protein. That was the first shock for us. But anyway, so rest of them are like others. So if you look at numbers, This is the percentage. It's very highly, 66%, 62%. Almost every of the higher number of helices have higher number of possibilities or probability of occurrence. Whereas this is others, means hundreds of other conformers. They just topology doesn't work. Very few available. So this is the result for, so what we come up is, next please, that is structural space topology. These are prevalent and these are non-prevalent. Here this is highly prevalent and this is totally non-prevalent. That means only few topology space are occupied. Very asymmetric way of having same sequence, having difference, doesn't matter. They just go into these functions or topology space. So now we did modularity. So this is a question. So why they go like that? So we found out very repeatedly, we did this juggling. We figure out even in larger space, that is if you have 10 helices, these 3 helix or 6 helix bundles or 5 helix bundles repeating, they're just repeating in the superstructure. They love this small one. And so we did this modularity check and then found, yes, the patterns are just repeating. Next one, please. No problem. Then comes beta protein. Same things happening, only one great advantage. Next please. That we saw the same thing happening. Prevalence are very high, non-prevalent. Only thing happened is, you can look at, I think, about it. The majority are like this, not like this. So majority of the modularity goes beta structures are anti-parallel from the N-terminal to C-terminal. However, it doesn't matter to us. We just calculated and just run this all work. Next slide, please. So not only, and so many are available space. That's why you want to show. These are only 1 or 2 or 3, that is in the 3 beta structures. If you have increased number, so many possibilities, but only one will be highly possible. Rest of them are not. That occurs in actual space, means crystal structures. Next slide, please. So now comes alpha beta protein. So this is necessary for all of you to know. Just apply to something and publish a great paper is not the work. You have to establish whether it is an alpha helix or beta, alpha beta, beta alpha, whatever may be the combination of proteins. All prevailing, are they following the same prevalent, very high prevalent, few, rest of them not prevalent. If you are not doing for all, then your research is not really a rigorous research. Please do note this. I am mentioned this because I am sure most of the people here are in GLP and all. Theoretical work also needs GLP. If somebody is thinking, oh, I will do modeling and I will tell blame on computer, sorry. This is what you have to do. Next, yes. So basically what we figure out, evidence of this modularity. See, in alpha helix, we figure out like this, repeats after repeats after repeats in larger structure. Beta structure, something like this, goes on and on. Double, that is alpha beta, something like smaller ones. And the most interesting thing what we found was the next slide probably. So this is like modular. So we identified systematic manner. We have using context string. Actually, we got this systematic manner. Otherwise, we wouldn't have seen it. And the next is then, yes, now comes our next question. When you develop everything, you remember, so what? That is the question I asked my students. Yes, you found this observation, but why? So what? Why you got this? So find it out why you got it. So he had a bigger problem now. So what he did is all type of descriptor, computers are the great facility that way. So hundreds of descriptors available in published papers. He just got all descriptors in proteins, amino acids, structure, conformer, everything. He took all these physical chemical descriptors and some of them I showed here. Many of them he did. And then he plotted for the non-prevalent. Those are not occurring so much. And for those who are prevalent in every class, alpha helix, beta, beta alpha, everywhere he plotted. Very interesting. What do you expect? Some property must be. So beginning, all experts told me hydrophobicity. You know, the proteins falls because of hydrophobicity. This is a terrible dismay for my student. By third year, or end of third year, he said, oh, my thesis is going to be on dogs because I didn't find why. Yes, they would have been in dogs. This plot shows you. Now, do you see this error bar? That's because I took 4 data sets from the beginning. Our error bars are much larger than differences. So prevalent ones are darker and big ones blobs. And those who are not prevalent structures, they are like this. So none of them, these are only 4 properties, 3 properties. None of the properties had any relation. Sorry to tell you, none of them had any correlation with this prevalent one should have something. At least it should have hydrophobic property, good. No, not a single amino acid property did tally with any one of them. Next slide, please. So that was troubles. Even accessible surface area, a little bit less accessible surface area for beta anti-parallel. We saw little correlation. That's not sufficient. You know, there are statistical methods to identify. I'm sure some of you would have published paper immediately. There is hydrophobicity, everybody says. But you know, this is what I always tell everyone. Research is like Satya Sandhan. Research is not solving your problem only. You have to find the truth, why it is happening. We could have published a paper, and he was very keen because 4 years over, his scholarship will be over, and he wanted to submit the thesis. I said, no, we are just hiding the truth. We are just you know, sigma, well, p-value is a little significant, and we publish. Is it the truth? So I asked him, ask yourself, what for the proteins are? Why the proteins have such structures? There is something, no? In the cell doesn't make proteins of its own choice or its own sequence. Proteins are there only because they have some functions to do. So why don't you correlate with function? That was a problem, how to do the function. There are plenty of functions, different kind of functions, but then next slide will, so you can see bad results over there. But now you see, prevalent and non-prevalent classes, we had 2 methods. One is based on the annotation comparison, what they do. And the second one is metabolic reactions, substrate, etc. based. Hard work. Next slide, the same slide. So these 2 ways we compare the functional properties of whole lot of these proteins, which have preferred conformers and non-preferred structures they are happening. So I already told you preferred means they occur all the time in the protein database, and non-preferred means very less they occur. Now comes the interesting, so next slide, and we found the correlation. So you see, even though error bars are high, you can see the prevalent average is here, same class of fold, and non-prevalence are much low. However, if there are less and less and less, that is large number, data being short and short and short, we don't get very well things. But this was good enough to tell us the story. What the protein structure wants is diversity, many functions. Of course, the next slide actually, I think all these prevalent topologies can afford to have many functions. We just had a list of functions. I think maybe the second slide. Okay. This is the most interesting slide I like you to remember. We did plot functions of so many functions, and all belongs to the prevalence, all prevalence classes. So it can be, you know, this EC number tells you function. So EC numbers, majority EC numbers with the prevalence. So what we notice, it has some characteristics of forming the structure because it can do, the nature can do many functions with the same 3-dimensional structure. So nature is very lazy. It found few most important core motifs and just does that. Hence, this is one of the most famous motif called Rossmann motif or Peter-Rollmer motif. Maximum, when we study, maximum number of proteins, structures come with this motif, and all of them Almost all of them belong to everything. Nuclease, protease, carbohydrates, polymerase, reaction. Every biological function you find, the same motif repeats somewhere here or somewhere there, either part of it or center of it. What we then come out with this plot, which is interesting, we thought, just press one by one. Guess what we figure out, 40% of proteins, all active site residues always are in that core region, where small core, what we defined as maximum prevalent structure. 40% of them, 50% of them, at least one catalytic group occurs in the scaffold, and only 5% doesn't have many. What it tells us, not only these are prevalent one structures, maybe stability. Actually, we did that. I don't want to show that. I had folding done by these prevalent structures and non-prevalent structures, and we figure out prevalent structures quickly fold. The other ones have a lot of hassle. But over and above, they can afford so many active amino acids or catalytic amino acids. That was the crux of the story. Why most of the scientists use the catalytic active site and make medicinal chemists are perfect in designing things because they don't care the other part. Other part nature knows that only will be. And they just find that. And this slide was, that's why I say, why we have now functional diversity actually makes the structure. Those structures doesn't have functional diversity, they won't occur even. They won't be folded even. Yes, so these are like most prevalent ones what we found, which I showed you in structure, a picture earlier, and many more other here. Next slide would tell, more or less, I'm come to the end of the slides. So the summary, and summary is, what we figure out, somehow, if we use this representation, we are able to find, actually pinpoint, that particular distinct class called real, the prevalent modular. And modularity emerged from the functional requirement in nature. And that now you can explain why same modularity was having many mutations, still it has the same. If you remember, DHFR had the Tim barrel structure. So even if you change so many amino acids, it doesn't bother. However, carbonic anhydrase has beta role structures, which can change anyway. So those questions, which I could not answer then, now we can answer them depending on the functional system, and they got many divergences. And of course, the kinetic we did, I didn't show you so that it would take another half an hour, but what we decided that we should have this. So anybody has a sequence and builds a structure from a homology modeling or something, they should check one important thing. What is the function of that particular protein? Before they choose their whatever homology, motif or a structure, number one. Number two, they must check whether the active site residues mostly siting on those common maximum possible motifs. If you don't have these two, most of the structures you will interpret with docking and everything is really useless, literally useless. So this being my summary, I want to go to the final who people. These are for most of the students. I really like, please go ahead and do, which has been asked always, so I always keep these slides always with me. You know what? Go ahead. There are three things to becoming successful. Number one is science, but in spite of. Everybody will tell you, no, don't do it. The student was so dissolute. He said, everybody is saying I'm doing on the wrong way. But I said, just you try to find whether you're wrong or right. So in spite of everybody telling you wrong way, please do. That's the science. And use science for your guidance. Next is teamwork. Well, you need some technology to do it, whether you are experimentalist or a theoretician. But the most important is, I have seen people saying, oh, this guy is better, that guy is good. I have found in my career some 30 pages, not many, only few 30 PhD students. Each one of them are so unique. And why they're unique? Because they taught me lesson instead of I am teaching them. They taught me, each one of them taught me so many lessons. So every person should compete with themselves. Actually, the best way is that do it. Fine. Well, this year I have done this much work. Why don't I have a publication next year? Aim yourself, you will do it. And next year you say, I should target 2 publications or maybe 2 more research ones. It is, you should continuously do it because next click will come. Never stop learning. And that's most, important thing. And it goes whole life. We do learn every time. Only then we succeed. So next slide would give you my teams. So many. These are all my great friends and the teamwork people. These are JNU. These are from Pune. And these are my, I would say, mentors. They always come and anchor me to the down. I know whatever vague things I start thinking about, they will say, hey, this will not happen. But that's the best. I go against their will and try something. Next, just like this is the guy Tawsif. He's presently working in, he's pure experimentalist now. He has published a paper in Nature with some 25 teams in working on this famous rats laboratory or whatever you call, where from we get all these rats. Okay. And this is the guy Shailesh, just to click. He is the guy Shailesh who has also worked on developing this pro-Lego functioning way. Main people are mostly Dick. And right now, means last 2 years, I'm working with a company, which is a startup company, whose name is Teura. They have this laboratory in 23. Now we have got a office and staff for working on computer and as well designing, etc. and doing some field work trials. They work on mostly, at present, we work on the aquaculture. So my job is to guide these guys to work out. So many people ask, so after retirement, you should sleep home and go and give advices to university, become university faculty, retired faculty of university, go and teach them. I'm deadly against it. The reason is, if I go and teach them, then the universities will never hire a young person who is my student waiting on the job. I said, no. Rather, I take a biggest risk to improve the aquaculture life. That is mostly prawn, shrimp, and these business people who invest their whole life on it, they are the middle and small businessmen all over the world. And full Southeast Asia has this problem. Moreover, if we spoil our water by dumping a lot of antibiotics, I'm sure that we are bound to have worse problem. Sir was telling all the time, you people got dumped all these, fast food and all. That's why we got diabetes. Forget fast food. We will be dead. We all have cancer only because if you dump so much, number one, antibiotics. It's not a wise idea to put not only antibiotics, they put a lot of pest control system in water. And water cannot be stored. So these guys keep me quite highly active. So you can think of how I'm spending. I don't have much time really to spend myself as such. So thank you for next slide. Actually, I will wish whole lot of work I did because of them. Just next slide, please. So mostly the softwares and things are over here. I have a small, I'm trying to make it domain working very nice, but most of my ex-students, they load all sorts of softwares and say, it's not good, so I'm trying. So if you people see that it's not working, blame on me that I'm not yet made it. But I really thank you for all that, okay? Thank you. So I hope I have managed. I've exhausted time.
Hello. Thank you very much, Professor Indra Ghosh. It look like God had gifted you gray matter more. You explained well about this topic, and you told what researchers should do and what else when they're designing the pharmacophore using a structure of protein or unknown structure of the protein, using computational approach to the biology. Any audience want to share or explore this topic? Anybody else?
Very nice presentation as usual. What I think one should explore, you say same protein has got different functions and protein has different structures and you predict the structure which should be there. So the thing is that it will be it may be because of different protein is a big molecule. So the different functionalities can be from different regions or sites. That's what you say. This is the active site for this particular activity. There can be other active site for other. So this may be one of the reasons for their functional difference. For protein-protein interaction can be different. But I am talking for the small molecule interaction. I think we are running short of time, perhaps. I think we'll discuss very.
I think there is no other question. So thank you, Dr. Ghosh, for such a nice talk. Now I invite, Now I invite Dr. Srikant Rath for the next presentation. Dr. Srikant Rath is Chief Scientist and Professor of ACSR CDRI Lucknow. He is a highly skilled researcher in molecular and cell biology with over 30 years of experience in academics research, leading to various scientific projects. He has extensive hands-on experience in protein biochemistry, molecular biology, cell biology, microscopy, and actually there's some problem in reading, and experimental design. Dr. Rath is very important member of the team, which is responsible for the implementation of GLP in CDRI. So now I invite Dr. Rath for his presentation.
Thank you, Chairman, sir. Thank you, Arun, for giving me this, calling me to this stage. I am grateful to Saxena, sir. Actually, I got a call from him on 28th while I was taking exam of Lucknow University students. I just told, yes, sir. When you are calling, I will be there. And I came with the two big people, Dr. Dikshit and Dr. Dwivedi ji, who gave a nice company to this place. Having said that, let me go to my topic, that is working in a good laboratory practice environment. We all do research, right? And more or less all of us are handling laboratory practices, maintaining records, doing experiments, getting the things done properly. Perhaps if we will ask that whether you have done it properly or not, many of us may fail because we are not recording them properly. If you cannot reproduce a reliable results, then your results are actually not reliable. Since the yesterday evening, Professor Y.K. Gupta gave a stress on that. He was talking, he is heading at the moment, the NGCMA Technical Committee and responsible for implementing good laboratory practice system in around more than 60 places in the country. And only very surprisingly, you will know that only 3 government laboratories are under GLP accreditation and CDRI is one of them. Having said that, you will be also surprised to know that CDRI is the first government organization to initiate all these thinking process in the country. But unfortunately, the 3rd laboratory in the government setup who got this accreditation because we do not compromise in small things. That is what my seniors, mentors taught us. And the day I was implanted in CDRI in 1998, I actually came to know that what actually good laboratory practice is. After that, I have undergone several trainings by OECD, by Indian NGCMA, and we are trying to do our best in CDRI. In the 3rd cycle, we are actually running the good laboratory practice. Every cycle has got 3 years, and we are running in the 3rd cycle. And I shall be talking about what are the advantage and disadvantage of this. Actually, majority of the things are very advantageous. And since most of you are chemists and trying to develop drugs, you must align to the system of good laboratory practice. Next slide, please. So why good laboratory practice is required? This is little bit of history. In the early 70s, FDA became aware of cases, poor laboratory practices all over the USA. Basically, they were looking back to see the adverse effects which were happened in the society, whether the experiments done in relation to those molecules are actually done correctly or not. So they found that a lot of fraudulent activities and a lot of poor laboratory practices were discovered. Some of these poor laboratory practices are unethical and equipments were not used properly. They are not calibrated. Some of them are not actually appropriate instruments, inaccurate accounts of the actual lab studies, and inadequate test systems. The animals were not there, animal house were not there, but the molecule is tested in animals and reports were supplied to the public regulators. So GLP was first introduced in New Zealand before US taken any action. Actually, GLP is already in the implementation in New Zealand and Denmark in 1972. US started it in 1978, actually implemented in 1979. Next. So this is basically a practice which is now handled by OECD, that is Organization for Economic Cooperation and Development, which basically tells about how the experiment should be planned, performed, monitored, recorded, archived, and reported. Planned means it has to be a written document, which can show that it is actually planned on which date, who are the people who will be involved, who are the people who are experienced or not, whether they are educated properly, whether they are trained in the proper business, proper experiments, and performance, how they were performed, what are the conditions of the laboratory, what are the instruments they used, how the environment were controlled, what are the mice species they used, what are the cell lines they have used, and who has monitored that time to time. You have a quality assurance system who monitored this, and the data in every step has to be recorded. Majority of time nowadays in research scholars, if you ask them question that you bring what you have done, they will nowadays they bring the laptop and show you. If the laptop is crashed, then what you will do? You will actually face the trouble. So basic practice is it has to be recorded properly in the hard copy, and you can transform into computerized records. There is no problem. But the first thing is that it has to be recorded properly in the record books. Then it has to be archived properly so that in after 3 years, 4 years, 10 years, because you are dealing with drugs, many adverse effects will come, and if data is audited after some time, the data should be produced by the organization. So it has to be archived properly, and the reports actually show what exactly the observations were. If there is an adverse effect, it has to be also recorded and reported so that the company or the investigator or the father of the molecule should know that what would be the expected adverse effect maybe in the human when it will be tried in clinical trials or in the society. Next. So the objective of good laboratory practice is to make sure that the data submitted are a true reflection of the results obtained from the studies, and GLP makes sure that the data is traceable and reproducible and promotes international acceptance of tests and studies. We are not developing the drugs for ourselves. We are not developing for our country. We are developing the drug for the entire society. It consists of several nations, several continents. So we have to be careful about how the data is recorded, and it will be once you are developing the data in the GLP format, all the OECD countries, including US and Japan, they will accept your data, and you don't have to repeat the experiment. They don't have to repeat the experiments. 2-3 incidences actually NGCMA faced in last 3-4 years. Some of the data they doubted, they came back, and it was found that the data was not actually developed in a GLP environment, but it is non-GLP data actually added to the GLP data. So it could be traceable and caught. So that is the reason one has to be very, very careful about the recording and keeping them in the archives. Next. So the advantages are many. It can guarantee the quality of non-clinical laboratory studies. Non-clinical studies are executed ethically, planned, performed, monitored, recorded, and assures that the data are true reflection of the results, assures preclinical safety and residue safety, assures generations of high quality and reliable test data, assures mutual acceptance of data, and assures increase of public confidence. And if it is developed in CDRI, naturally the confidence is very big. If the data submitted by CDRI to CDSCO, even today, the CDSCO never ask a question. The same data what developed yesterday, Dr. Saxena was talking about Dr. Gulati, who is taken the permission from the CDSCO for some other biological effects, and the drug is now accepted in 2022. And because of it is developed in CDRI, actually half of the things is done. The other things they have done in USA, because in that committee I was there, I asked specific questions because I was thinking of the CDRI's right is being marred. But he told, sir, no, we have taken, certainly we agree, we have written in the document that the data, most of the non-clinical data were developed in CDRI, but later whatever data in the for the particular disease, we again developed and tried it, and all the documents they have submitted that. So the confidence level increases when you are actually working in a good laboratory practice condition. But disadvantages are very, very less. Of course, whenever you will make a quality system, quality management implementation, naturally expensive things will be happening. It is expensive process, time consuming process, and more skilled manpower is required, because most of the all of the activities, if it is not done by skilled manpower, it will not be recorded properly or archived properly. Next. So GLP governs the GLP principles, which addresses almost all the things from apparatus to the quality assurance unit, test systems, test reference substances, standard operating procedures. All the procedures are being governed by standard operating procedures, and performance of the study is being monitored, and reports are being prepared in time, showing the data integration. Next, so all these activity what is governed by the GLP actually is taken by the test facility management. So test facility management ensures starting from the apparatus to all the conditions. Starting from the apparatus, it is IQ, PQ, and OQ. Generally, we don't bother for that. Once the instrument is functioning, every time we actually generate data without checking its integrity in particular time. And we also never check the in vitro systems whether the cell is actually behaving properly or not. Several cells, hundreds of cells nowadays people are using. So in the GLP system, you have to actually provide a data that the cells what you are using are actually in the particular condition was in proper condition and it is not contaminated. Then in vivo systems like particular strain, particular species, you have to spell it out and where the animals came from, how they were maintained, how they are handled, everything, and who treated the drug or test material or test item. All these things has to be recorded, and the test reference substances which were procured from the generally from the standard companies, every details has to be there. Where is the expiry date? How many experiments were done with the reference system? Everything has to be recorded properly, and everything actually this quality assurance people, they come time to time to check. Surprise checks were being done. Very at least 4 surprise checks in 1 year we used to do in CDRI. So basically your tension is over when your test facility management is ensuring everything by putting maximum people to monitor everybody. But the studies are actually conducted by the highly qualified study directors. That is, in GLP system, all scientists are being called as study director, and other people who are associated with them, like technical officers, they are called as study personals. And these people are responsible for generating data in proper way. If any problem happens, they have to actually inform the study director, and study director has to inform the test facility management. Parallelly, quality assurance people also inform every happenings to the test facility management, so that the test facility actually regulated properly under particular conditions, so that it is fit for the recording and fit for the conducting study. For example, regulatory studies like the regulatory toxicology or pharmacology studies. Next slide. So the biggest responsibility is actually the people who are working in the quality assurance unit, because they work like CBCID people who actually surprisingly check everything, starting from the documents of protocols, study plans, to the quality of the material which are being utilized, and the quality of the animals, animal health records. They monitor the environment. They check the AHUs, how many times the air flow is being regulated, who is checking this HEPA filter is there or not in the animal house, and of course the cell culture system is working properly or not. All these documents, they are being checked, because they are the responsible people, they are the eyes of actually the test facility, and they check and inform the test facility manager that everything is going smoothly during the conduct of the study. Next, the most, as I have already told, that study directors are the only responsible people for the all studies. So starting from the approval of the protocol to the study plans and education of the study personals, ensuring ethics clearance nowadays. Ethics clearance are required for all the activities you do in the handling with these animals. If you are handling these cells, you have to actually take from bioethics clearance. If you are handling the animals, you have to take the permission from the animal ethics committee. All these ethics responsibilities laid on the study director's responsibility. Then ensure the quality assurance personnel to know, to inform that your study is going to happen, and this is the study plan and the time plan. All these things has to be communicated to the quality assurance as well as test facility manager. Then, of course, when the data is generated, you have to interpret the data. You are the sole responsible person to interpret the data, analyze accurately, and they document, every document has to be checked and report well, so that every adverse, everything which is not up to the mark, even if you are leaving a outlier, you have to report that 1 or 2 outliers we found and we have reported it indifferently. So, and it has to check. Sometimes it happens that while conducting the study, you have to amend the things, plan the amendment and deviations. So you have to also record that what are the amendments and what conditions you have done the amendments and what condition you have made the deviation, and how these amendments and deviations are not influencing the study report. You have to report that. Then, of course, all documents, it has to be signed by yourself. And the day you sign the report, that is the final day of the study. Next day, you have to archive, so that you cannot further manipulate or check or recheck or correct anything in the report, and it has to go to the archive directly. Next slide. Similarly, the study personals, they are the pillars of the study, because upon them only the study director actually lies his faith, so that the data is generated properly. So they have to be appropriately educated, so that all the parameters, why they are relevant and when, in what conditions it has to be taken, one has to check. For example, if the reproductive study is going on, proper cycling females are there or not, whether it is checked or not, where the females were allowed with the males, and whether the cycle, which stage of cycle the females are allowed with the males, and where the checking was done and when the checking was done for the mating studies. All this has to be the study personnel should know, so then only he can accurately generate the data. Then he should know this standard operating procedure, whatever standard operating procedures he is using, all the standard operating procedure should be known to him or her, and the training has to be properly. Whether any doubt is there, so he has to get it clarified from the study director or the scientist under whom he is working. And he has to also have some trainings of how the laboratory general management is there, where the drainage system is there, how the waste management is there, where the specific training of data collection, translation, and protection has to be known to him. Then personal sanitation and health precautions has to be taken by him. Without PPP, he should not do any experiment. Without PPP, he should not enter into the animal house and handle the animals. So segregation of waste and how they are being disposed, all these things, all these SOPs has to be trained properly by them, so that they can conduct the study properly. Next, persons who are responsible for are actually the facilities, which are in vitro facilities and in vivo facilities. In vitro facility, of course, the bacterial facility and the cell culture facilities. So the personnel should know actually how the cultures are being done, what are the cell types, how the morphology is there, if the cell is disintegrated, how the cell will behave, and if they are contaminated, what are how under the microscope they are being observed. All these gyan should be there for the study personnel, so that they can actually conduct the study properly. Similarly, bacterial, for example, most popular study in bacterial studies actually AIMS-SA, where we do regularly the mutagenic studies. If there is tester strains are not behaving properly, then all your data will show that all the molecules what you have made are mutagenic. So that will mislead the investigator, and the chemist who is actually preparing the molecules will be frustrated, because all the biology labs, they will say that the molecule is mutagenic without any things, because the bacterial reverse mutation system AIMS-SA actually handles Salmonella typhimurium test strains. Actually, if they are not maintained properly, they will actually misbehave, and these instead of 50 revertants, they will go on 300, 400, and it may be very difficult to understand how a molecule which is not expected to be mutagenic is showing so many of revertants. So naturally, in such conditions, the study personnel has to be highly educated, and he has to show all the data to the study director or scientist, so that scientist can decide what is happening actually. Next. Similarly, animal house facility, if the healthy animals are not being utilized in your experiments, then all your results which are derived out of those animals will go wrong. And therefore, the animal has to be, all the animals has to be healthy, and they have to, if you are getting the animals from outside, they have to be in the quarantine for 7 days, other in acclimatization for, I am talking of rodents, for another 7 days. If you are using monkey, of course, we are putting them for around 3 months and treat them properly for tuberculosis and other infections, check them properly in 3 times, then actually implementing the study after 3 months. So it takes a lot of time, and that's why we have not taken the GLP accreditation for monkey facility, but we do monkey studies in a GLP GLP-like protocol following, and we have separate areas for animal for different species and studies, and we have separate for rooms for sacrificing the animal. We have separate rooms for taking out the blood, so that the animal ethics is being followed very strictly, so that one animal should not see the other animal, so that the animal experience are perfect. While maintaining the animals in the animal room, we have room temperature monitoring, we have humidity monitoring, we have air change monitoring, we have a room pressure monitoring, so that no infection infection goes to the animal house, because the all persons, including the study director and QA persons, including TFM, has to go to the animal house with proper personal protection equipments, so that animals are protected from us and we are protected from them. Next. We have other services. Since we have a government facility, we have all the people who are working for us, but in the private setup, of course, it is very difficult, and this part is actually very cost effective, expensive, and it is not cost effective. That's why majority of CROs, they charge a lot of money for conducting a study. For example, if we are studying and spending some 20 lakhs for monkey studies nowadays in India, for one monkey study for 28 days, people are demanding more than 3 crores, 3.5 crores they are asking, because all this they are spending all this money most of the time in this section, which are actually not very expensive for the maintaining these other laboratory services. Since government system, we have all these things. We actually do the things, pest control, fire extinguishers, yearly basis, and monitor them, so that all the test facility manager actually checks and review properly. You don't have to, as a scientist, bother for all these things, because all these are taken care by the test facility manager. Next. So that is what we are doing. I want to give the impression that working in a GLP facility is certainly very smooth for the scientist, but it is for the management to take care whether they will take the accreditation for test facility or not. But for institutes like this, I would rather recommend a small facility. We have to first practice the recording system properly. Whoever is doing research properly, a unique coded book should be utilized for every researcher to write down everything, whatever they do every day's work, so that you can actually monitor your observations, so that many molecules, what we are thinking that it will not work, many times it may work, because biology behaves very differently. For example, madam was talking Lipinski rule, every time all the molecules are not following. Many of our successful drugs are actually, if you analyze, they may not be following the Lipinski rule, but they are working. If you are nowadays, if you develop the same molecule, you may throw them out because it is not following the rule. But many times it may work if you are actually go back and see the records that it may have some biological effects. Ultimately, we have to use the molecule for the efficacy and cure the diseases. So we must not leave any opportunity to miss our data, and we must not miss any opportunity to question by the other people that your data is not reliable. That is pathetic thing. And in this age of pop peer, I think each of us should follow a disciplined recording life. Do whatever you do, record it, record it, record it. We are very outstanding people. In India, people are generally more intelligent than any other country, but they fail to record the things properly. Everything we carry on our brain, and many times we overdo the things and fail to justify that what we have done is right. Thank you so much.
Hello. Hello. Hello.
Thank you.
Hello.
Thank you for the representation and about GLP practices. Are there in India any regulations for private institutions or private laboratories to get GLP certified for animal house and things? Or is it constricted to public sector? I mean, only to the major institute? Yeah. Yeah.
We got the certification for human purpose, but there's no the time is pumping.
And what is the accreditation body in India for ensuring?
It's governed by DST, Department of Science and Technology, and they have a unit called National DST compliance, where the technology and Dr. Diwan is also a healthy monitor, but before becoming a monitor, but still already in PDRI, so we do not conduct any inspection for other academic stakeholders. But there are many 30 around and all centers are not public. That is what other countries like India.
Thank you. Thank you.
Any other question? Dr. Rath, I have one question. How long we have to keep a GLP study in archive?
Actually, I for the department of we have to even for 3,000, 130 for 3 years. So 9 years you have to keep the record. And there are the And the material is there for so you can with proper documentation it can be started. But the documentation is the minimum requirement for in India. But anyway, they have somewhere in US, they have UK, they have 4 years. But something around that, like 10 to 12 years. All it depends upon by 10 years if you are unable to develop the services, all the jobs are come to regulatory services 2 years of regulatory service. So literally another 8 years within that 2 years. Effect of the molecule human effect. So literally the state of the molecule is divided. If it is going well, and most serious going to take up. Minimum 9 years.
How long we have to keep the biological specification, biological samples?
Yes, biological samples. So you take all the people around 40 required. So we take all the keep all the people for and I will go to 3 like that. Yes, 5 2 years. We have to do for every time. So many times for
I think in GLP, the integrity of the persons who is conducting the experiment, that is more important. I come across with a case where I asked for something regarding GLP studies, and that study was for 6 months. And surprisingly, I got the report for that, the GLP compliant, fully compliant GLP compliant report of that within 2 months. So how we can prevent that?
Yeah, obviously the main part qualitative and I will go for this program and that will be beneficial without knowledge. But for God's grace for us, you got me and you said. The other way I would say, if you are honest, you do not have any loss. For example, you lost your smartphone in life, internet not valid. It's very difficult to prove that you are very, very difficult. Reason why, even if you are honest, you have to have record to keep that your molecule was in perspective.
And this is true for the persons who are not working in the GLP environment. They must also keep the records properly so that if there is any query regarding that or regarding the integrity of that experiment, they have to produce that and they can prove that whatever they have done, that is all correct. So GLP is on one side, but the integrity and the record keeping is on the other side, which is very, very important for a researcher who is doing research, active research in the area of, especially in the drug design, drug development, or other areas of research, healthcare. Thank you, Dr. Rath, for such a nice thing. Now I invite Dr. Alok Kumar Saxena for the next lecture. Dr. Alok Kumar Saxena, superannuated as Managing Director of Selger GTC India Private Limited and is currently the proprietor of AA Consultant. He is a chemical engineer with a doctorate from IIT Delhi and a postdoctorate from University of Oldenburg, Germany. He has over 44 years of experience, including roles such as Deputy Director at the Indian Institute of Petroleum, Dehradun, Chief R&D Manager at BPCL, General Manager of Process Licensing for Excellence in India, and Professor at UPES, Dehradun. Dr. Saxena has significantly contributed to the development of refining technologies and commercialization through close R&D and process design interactions, modeling and simulation, and scaling up from lab scale concept to the commercial design. He has published more than 50 papers in journals, encyclopedias, and conferences, and holds 4 patents. He is a recipient of CSIR Young Scientist Award in Engineering Science and the CSIR Technology Award. So, Dr. Alok Kumar Saxena, please.
Based on artificial intelligence. So that support and that seems to be a future trend. Just a minute, let me find. Yeah. Anyway, why it is. I don't want this way. Anyway, just a minute. Let us see. Okay. So I'll be presenting on current trends in healthcare technology. We have been listening various speakers, and I have seen lot of compounds have been developed, lot of testing has been done, lot of data has been generated. This is in the last one day we have seen it. But just imagine how much data we have generated in the last many years and how it can be exploited to develop more cost-effective medical support and medical treatment. So current trends are basically targeting in this direction, and AI is basically supporting this approach. It is not only medical sector or healthcare sector. If you see, based on tremendous computing power, real-time computation network, and vast data bank, what is happening? All sectors are driving their new business models. Now people don't go to the bank. Am I right? Things are available on mobile. See the transport sector. Uber is a one example. Food, you get everything at home from the required restaurant. So what all these things are based on the current computing power, data bank, networking. This is changing the scenario in different sectors, and healthcare industry is not an exception to it. Actually, after COVID, this sector has felt the need to prove its resilience. Situation was very different 3-4 years back when COVID struck, and that time digitization took really revolution in medical sector, and that was the time You could see on your mobile how many cases are there in which area, where is the peak, where it has gone. So it was some immediate response, a digitization effect, which triggered the use of the digitization or exploitation of digitization in medical sector. This trend has to continue, and there are various factors which are responsible for current trends. This includes different patient expectations after COVID, reform in healthcare system, which are being emphasized again and again. Government of India is also announcing various policies time to time for treatment, for cost-effective treatment, and demographic shifts. So these factors are responsible for current trends, which are mostly AI based or networking based. Regarding patient expectations, there is a report which this is 2024 JS Future Health Report, which reports a summary based on survey conducted of 9,500 healthcare consumers and 1,400 healthcare providers. This sample is distributed over different countries, different healthcare systems, different local challenges. Despite of these variations, the inferences are all are agreeing that healthcare system should be more accessible, it should be more equitable, it should be affordable, it should be well connected and human centered. So these are the things which are affecting or trying to exploit the current tools available, current computation power available to improve or to meet these expectations of the patients. Preventive care that is accessible for everyone, greater coordination of care between providers, seamless access, transparent billing, easier appointment. These are the expectations which can be addressed using current tools, which are being exploited in all industries. Now, banking system is based on internet. You need not go to bank. You can transfer money. You can encash money. You can go to ATM. So everything is network based. If we can do that, why not all the records can be prevented on the net and accessible at anywhere if any patient goes. So these things are basically taking shape. And today, Anushri mentioned about AI application, as well as Prakhar mentioned about SAP, which can even tell which drug is sold in which market, what identification. All that information is available online. How can it be exploited to not only for treatment, but also for drug development? There are a lot of reforms going on in healthcare system, which is for enhanced affordability, which requires to ensure that drugs are available at affordable prices. Generic medicine in India is currently buzzword. In US also, the government has announced the first 10 products, including healthcare costs, that will be subject to price negotiation under Inflation Reduction Act. European Union is also forming policies that any drug which has been introduced, not introduced in all the 27 member countries, has the exclusive window only for 8 years, which is reduced from 10 years. Germany also, the government is working to address perceived gaps in national pharmaceutical regulations. Similarly, Italy, the long-awaited reorganization of Italian Medicine Agency is currently in progress. Japan and China as well, governments are pursuing comparable initiatives to reduce drug costs. So it's a global phenomena, and affordability of drugs is an important, and there are government regulations coming in which are affecting the current trends in healthcare system. Demographic shift is the third reason which are responsible for current trends. If you see in India, aging population and declining birth rates. India's annual population growth declined from 2.5% in 1971-81 to approximately 1.3% today. Population is expected to peak at 1.7 billion, projected in 2060, before it starts declining at these rates. Elderly population expected to increase from 139 million in 2021 to 319 million by 2050. So this shift, this increase in elderly population is going to bring different requirements or different pressures on healthcare system. So what are these pressures? Double burden of diseases, rising prevalence of non-communicable diseases, continued presence of communicable diseases, increasing chronic illness rates, with 23% of elderly suffering with chronic diseases. If chronic population is increasing, numbers are increasing. So this, even with this percentage, pressure is going to be different. Healthcare systems, in India, there is uneven healthcare system between rural and urban areas, limited healthcare insurance coverage with only 37% of population insured, insufficient public health funding, growing demand for geriatric and palliative care. Professor Valdemir mentioned a lot about this geriatric treatment, and he also mentioned Russia and India have this issue to address in the time to come. Policy implications: reform healthcare system, ensure universal healthcare, affordable community-based health insurance, investment in healthcare infrastructure, and leverage emerging technology. So all these requires a coordinated effort. We have to address all these issues involving policymakers, private sectors, and local communities, means all stakeholders. As Professor Y.K. Gupta told yesterday, all stakeholders are we, you, and everybody. So they have to participate and come out to meet all these challenges. The demographic shift in global scenario, Spain, this is one interesting data. Spain expects to complete reversal of contribution of its labor force to GDP growth from positive 0.6% between 18 to 20 to negative 0.6% by 2040. What does it mean? Shifting from a historical labor-driven boost to a future of economic burden. This is the effect of demographic shift. Switzerland also anticipates a 30% increase in public health expenditure by 2050. South Korea and China are already facing the economic challenges of shrinking working age population. And because of shrinking working age population has compounded effect. Why? Working age population puts pressure on the ability to generate the tax revenues. If taxpayers are reduced, non-taxpayers are increased, tax reduction will be there, and growing healthcare requires more revenues. So this is the compounding effect, which brings demographic shift is bringing these pressures. These demographic shifts highlights the preventive measures and proactive healthcare. So there has to be more emphasis on preventive measures so that the 23% of elderly people are suffering from chronic diseases, that number at least can be reduced. In view of these factors, we talked about demographic shifts, changing expectation, and implementing the new regulations. What are the trends? What are the emerging trends? Current trends: increased R&D focus and investment, big data analytics, artificial intelligence, NLP-aided advanced communication, precision medicine, integration of blockchain technology, digital transformation, and requirement of reduced carbon footprint. This is something very generic. It is required for all technology, but these are specific, which are mainly based on network connectivity as well as data availability. Let us briefly touch on these points. If you see the increased R&D focus on investment, in India, a strong commitment to R&D in pharmaceutical sectors played a pivotal recently. Pfizer R&D Center in Chennai, Novartis facility in Hyderabad, Marks Healthcare R&D Excellence Center in Bangalore, Bayer's Data Science and Analytics Center in Hyderabad. These are the centers which have been activated in recent past to meet future challenges. In the wake of COVID-19, pharmaceutical companies have further amplified their R&D investments. Government of India also introduced a production-linked incentive, PLI. Professor Y.K. Gupta was mentioning about it yesterday. A scheme for pharmaceuticals with a financial outlay of 15,000 crore. Dr. Reddy Laboratories, Biocon, Strides Pharma Science, and Premier Medical Corporation have already received the first tranche of these initiatives, totaling to up to 166 crores. So this is all. These are the policy implications. Last budget, Government of India reinforced the commitment to innovation by allocating 3,200 crores for research. So on global arena also, pharmaceutical industry is undergoing transformative phase. Trends are basically going with a focus on innovative technologies, personalized medicines, strategic collaborations. Investors are putting more funds in targeted therapies and personalized treatments, and these shifts already driven the breakthroughs for genetic research and rising demand for more effective healthcare solutions. Notably, investments in biopharmaceuticals, especially areas of gene therapy and regenerative medicines, has rebounded strongly after slowdown in 2022 and 2023. Means after COVID, there is a fast response on these techniques, and globally also pay more emphasis on R&D. Now, all artificial intelligence, any inferences is to depend on how to analyze and combine the data. Big data analysis is a systematic process of examining large complex data sets to uncover valuable insights and patterns. Means we talk about a lot of evaluation, lot of response to different drugs. If these data are available, this data is available globally and accessible through computer networks, one can draw the fast inference with lesser resources required and less time required. Driving innovation as energies at every stage of drug development, from discovery to manufacturing and marketing. It helps with the SEP is, for example, the yesterday morning Prakhar mentioned, SAP data can be used, which chemical was imported, where, how much it was, which market, how much it was consumed. All that information can be derived from that. The key enabler to make informed decision to optimize process of ultimately improve the patient outcome for improved affordability and access. If we can reduce the time and investment in evaluation and development of drugs, it will reflect on the affordability of, plays a key role to allow researchers to analyze vast publication with clinical trial results. Right now, I've seen that people hardly go to library. Everything is available on system, computers. It is easy to dig out all relevant recent references, and that is really helpful, and it should increase the efficiency and cost involved in drug development by avoiding non potential candidates for drug. Artificial intelligence is also part of current trend, as we have been, which we have been discussing, and there have been previous speakers have spoken about it. It is basically computer science that enables human intelligence and perform complex tasks, enables machine to simulate human abilities like learning, problem solving, decision making, and comprehension. AI revolutionizing drug discovery with enhanced efficiency, automation and optimization of drug manufacturing, health risk associated with clinical trials, and reduced costs. So AI is supporting in all these, which should make the healthcare system more affordable and equitable. NLP aided advanced communication, natural language processing is important subfield of AI. Everybody must have experience with Alexa. You ask the temperature, you get the temperature of the city. You ask, listen any song, just talk to Alexa. This is the, if such things, even in cars, the features, you just tell the call this person. You will, while driving, you can talk to that person. So all such easy communication with the system, with machine, is basically should revolutionize the healthcare system also. Bridges the gap between human communication and machine understanding, enables more natural and seamless interaction between humans, revolutionizing the pharma industry. Vast amount of unstructured data, if it is put and we can talk to machine and it can compile and give the right information at right time, life is much simpler and much healthcare system is much effective. Even in I think in during COVID period, nobody was allowed to visit doctor. Everything was on phone. I had to consult doctor, do the medicine, injections to my wife during COVID. Those, no support is available, and all those things are coming, and need was felt during that period, and now things are taking shape, which are forming the future trends in healthcare system. Precision medicines is also another area, means personalized medicines. Patients' unique characteristics are considered for to have personalized medicines, integrates genetic information, environmental factors, and lifestyle choices. So all these factors have been discussed, but if all these things are customized for each patient based on database available for individual, for the region, for the country, it will make the decision making much simpler for the doctors. Enhance disease prevention, diagnosis, treatment, leading to more effective targeted care, an approach to make healthcare system better connected and human-centered. Integration of blockchain technologies, another part of networking we are talking. Blockchain technology is basically decentralized, distributed digital ledger system that securely records and transact across the networks of computers. Ensures data integrity. Data integrity is the point. Reproducibility was emphasized by Dr. Y.K. Gupta yesterday. And if it is data integrity is maintained and it is immutable, and this blockchain technology should facilitate the transformation or withdraw the information at right time. This will also affect supply chain integrity, data security, and regulatory compliances, which are also important requirement to maintain the data to be used in future. These are centralized treatments. Nature blockchain technology offers innovative solutions to improve trust and efficiency. Digital transformation is important part to modernize, enhance, and drive business growth for decision making. And it is that's why digitization and digital transformation is helping to utilize all available tools, high computation power to have well-considered decisions. The key components though include it demands cultural shift within organization. Once we go for a digitization system, SAP system, I've seen in an organization, if SAP system comes, all staff is first, there is a lot of inertia. Oh, it's something new has come. But that should not be the culture. One has to adopt to new approach, new techniques to be in line with global trends, to be in line with facilities available, or to exploit the tools which are available, which is going to improve the convenience and efficiency of the system. Some notable points I would like to summarize here. Pandemic acted as a catalyst for digital transformation that we must appreciate, highlighted the need for rapid adoption of drug development and distribution. And things are progressing in that direction, setting up the trends as of now. Fast vaccine development through digital tools and streamlined supply, and that too in a situation where there are not much validation scope, validation window, validation subjects were available. So this was based on the remote interaction with doctors. This is a culture which adopted during that period, and continued treatment. So this is the effect of all digital transformation, which was catalyzed during COVID and taking shape in the coming trends, coming years to come. So, but all this digital transformation can be achieved, or all these tools can be managed if there is digital transformation there. All data is compiled, data is available, well connected with computers. So all those things are we are able to exploit to support and make the healthcare system more affordable and equitable. And this digital transformation basically forms the backbone for innovations, improved decision-making, seamless process, and ultimately driving efficiency, accuracy, and drug development. The requirement of reduced carbon footprint is common to all technology. It is not specific to healthcare or pharmaceutical, but it's also applicable here. We have to be worried about the greenhouse emissions. We have to find the ways in each technology, in new trends, how greenhouse gases release is reduced. For each technology, there is a term life cycle assessment, which assess how much carbon is emitted from the source of drug, ultimately it is consumed, or any raw material produced when it is consumed. and after consumption, whether it is produced any CO2. It is very critical for petroleum industry, and also in healthcare system. There are steps, there are approaches to reduce the carbon footprint. It includes switching off to continuous manufacturing, and there are cases which they have reduced the carbon emission by 80%, adoption of renewable energy. Schneider Electric program can support achieve Johnson and Johnson and Pfizer have ambition to achieve this. Means by recycling, by going to a continuous system, we can reduce the energy consumption in the system. Reduction in energy consumption and optimization, energy optimization leads to carbon saving, and it also gives a carbon credit. So to summarize, healthcare are on the path of major change. Experiences during the COVID-19 pandemic are already responsible for this trigger. Rapid digitization, along with integration of blockchain technology, big data analytics, and artificial intelligence are drivers for the shift. Factors such as changing patient expectations, demographic shift, implementation of more stringent healthcare systems are further fueling this change. This makes the sector a preferred space for more investments. Higher R&D investments are on the card. Precision medicines and remote consulting are becoming reality. And I have a video to show here, if I could run it. Just a minute. Is it no sound? Sound?
Test, including body scan, heart check, genetic testing. This is an AI-powered doctor's office. So the craziest part is that there's no doctor inside. It's called CarePod. When you set in your CarePod, you can access 86 different medical tests, including body scan, heart check, genetic testing, and even blood draw. Once you do the test, the CarePod dispenses the necessary equipment and sends your data directly to a remote doctor for analysis. The remote doctor reviews your data and gives you feedback, often in judgment. The whole system is built by a company called Forward, and their goal is to create a care for and offer affordable healthcare to billions of people.
To be frank, I am not advertising for Forward. I am only mentioning about the trend or situation, what we can anticipate in time to come, based on available tools, based on artificial intelligence, based on networking of computers, based on data availability. And at the same time, it's also important for us to share whatever data we evaluate, develop, to be accessible to all. I was discussing to Dr. Aruni that there are some ethical issues in that. But yellow or stalwart sitting here, we can emphasize the policy makers, how this can be shared for the benefit of humanity. Ethics is one part, agreed, but humanity is also equally important. There has to be some discussion and debate on this to resolve this. I think with this, I conclude my talk. Thank you.
Thank you, Dr. Saxena, for such a nice presentation. Now this talk is open for a discussion. So if there is any question, please ask.
You want to correct?
Any question?
Too late in the evening.
Actually, now I have one or two suggestions. These are not questions. In a country like India, where most of the population is very poor, most of the population is very poor. The cost of medicines in India are very high as compared to their production cost. I will give you one example. About 10 years back, I visited Kerala State Drugs and Pharmaceuticals Company, which is owned by Kerala government. At that time, citizen of a company was sold at 11 rupees per strip of 10 tablets. And the other company was selling the same drug at 33 rupees per strip of 10 tablets. And the Kerala Drugs and Pharmaceuticals, after taking the profit, they were supplying that 10 strip pack, 10 tablets pack for only 1 rupee and 10 paisa. So you can imagine how much amount they are just gathering from the poor public.
I think that's why there is an emphasis on generic medicine. Our government has been emphasizing to go for generic medicine rather than specialized medicines. There is a buzzword for that.
Actually, all these products are generic medicine. Even then, how much amount they are charging?
Correct.
Another incident, I will tell you. I was a member of one of the committee in drug pricing control organization. At that time, a tablet of a particular drug was of 10 milligram was available at a cost of 3 rupees 97 paisa per tablet. And the company wants to introduce a tablet with 20 milligram. And they ask that fix the price of this because it is a 3 rupees 97 paisa. So 10 milligram will be 7 rupees and 98 paisa. So we will fix it at 7 rupees 85 paisa. In that meeting, I asked them, what is the cost of 10 milligram of medicine? Because what you are changing in that?
Correct.
You are only changing the 10 milligram of the material, the drug. They told in the next meeting, I will come. So in the next meeting, they come with a proposal that, okay, we will set this price at 7 rupees 25 paisa. I further emphasize, what is the cost of 10 milligram of the medicine? And they were not able to say that this is the cost because all the other things are same. The packaging will remain same. The whatever they expend on the say marketing of that will remain same. The price difference is only 10 milligram of that drug. So my emphasis is that there should be a cap by the government on the greediness of the drug manufacturing companies, which is the requirement of today's India, especially for the purpose of the poor, very poor people, so that they can also have the proper treatment. If they want, they can give any money for the companies, but with a cap. Please keep your greediness under control.
Yeah, I think all stakeholders, including policymakers and consumers and everybody has to take a view and implement the policies accordingly.
Actually, in my opinion, as Yeah, yeah, yeah.
I think such solutions should be given to charitable trusts. They invest a lot of money. Adani is feeding Kumbh everybody. So they can also easily manage it with 15 kg plant, less plant, and so cheap. I think it can be done. That is, there's some policies to be reduced.
One more thing which yeah.
Yeah.
One more thing which I want to suggest, that on the wrapper of the medicine, we are mentioning the expiry date, the manufacturing date, the batch number, and the price. One thing should also be made, that what is the production cost of that. You mentioned that. I'm not asking to reduce the price, but you only mentioned what is the production cost of that drug which you are selling. Because a huge profit, more than 80% of they are having the profit on that.
Correct.
Yeah. But yeah, but as per
Okay, thanks.
Dr. Saxena,
Thanks.
for such a nice talk. Thank you.
Thank you, sir. In this journey of this scientific session, we have another renowned speaker, Professor B. Jairam. He will be online. Yes, Professor B. Jairam. Currently, he is serving as a mentor at the SCF Bio IIT Delhi. What is SCF Bio IIT Delhi? It is a supercomputing facility for bioinformatics and computational biology. It's a specialized area in IIT Delhi. Professor B. Jairam completed his PhD from the City University, New York, postdoctoral fellowship from the Columbia University, USA, then senior research associate from USA. And after that, he joined IIT Delhi as a academician, as a researcher, and he served as an head in the chemistry department since from the 2006 to 9. And he's a founder coordinator of the Kusama School of the Biological Science and the told SCF Bio IIT Delhi. He played a well important key role in the development of the several scientific software tools, including ChemGenome, Bhagirath, Dhanvantri. These software tools are freely accessible to the global scientific community through the SCF Bio website. And this is all about Professor B. Jairam, a brief biodata. I am now inviting Professor B. Jairam for the presentation on the very well established and well exploring topic towards the molecular Ayurveda. Please, sir.
Yeah, thank you. Thank you very much. Thank you, distinguished chairs, dear Professor Sisir Nandi, and various very well established distinguished scientists in the audience and my students. I know it's very late. I've started sharing my screen. I hope you can see this. Is this visible? Is the screen okay?
Yes, sir.
Oh, thank you very much. So I will share with you some ideas on how do we go towards a molecular interpretation of Ayurveda and how do we bridge the connection between allopathy and Ayurveda from target directed drug discovery towards a holistic medicine that we all believe in, traditional medicine, right? So this is something that we started long time ago, about 20 years ago, as a dream plan. At 10 a.m. disease reported, 5 p.m. drug is delivered, and like through an Amazon or Zomato or something. And how do we plan to do that? Let us say you had access to the genome, then you have its genome's annotation software, and you find the malfunctioning protein, or this could even be bacterial viral protein, right? And then you develop a structure, then you using your computational technologies as tools, and then you figure out a molecule which acts like an inhibitor, and then this inhibitor can be sent to a panel of doctors, and they will prescribe the dosage and so on, and eventually, if there is no toxicity issue involved, very initially just administer the patient, finish is running, jogging happily, and so that was the general flow chart. I think 20 years from now this will probably happen. But what's involved here? Genomics, proteomics, metabolomics, lots of information technology, HPC, and chemistry, because these are molecules after. That was dream part one. And now how far did we go? Actually, we did cover the entire pathway. So we have created a KM genome software which can do genome annotation. Earlier prokaryotes and now eukaryotes as well. So we can do that. 2024, we managed to complete the eukaryotic genome annotation software tool development as well. So that is our KM genome, chemistry-based genome annotation software. Then we have created a software called Bhagirath, which is force field based or ab initio physics-based methodology for structure prediction. I know a lot of you are aware of AlphaFold, which is doing much better job. I think in a Bhagirath reaches 50% accuracy in very difficult cases. In easy cases, of course, it's as good as AlphaFold. So I didn't bring the comparison. Talk is not about it, but otherwise, Bhagirath is one of these contenders for structure prediction. There are other tools as well in the world for structure prediction. So once you have the structure of the drug molecule of the target, biomolecular target, then you can create a molecule using our Sanjeevani softwares. So there are commercial softwares again, I would say, and very well established commercial softwares. But the good thing is Sanjeevani is as good as any one of them, and it's freely available. Any student who wants to do a CADD can easily access the Sanjeevani software as well as all the other softwares. Now moving on. So this, how do you do it? In fact, this entire pathway, I call this a Dhanvantari pathway from genome to protein, proteome to eventually drug molecule, etc. It is got pipeline got published in 2020 with lots of validations, right? So KM genome is the genome annotation, then structure prediction. Once you have the structure, then you do active site finding, quick screening of millions of molecules, and eventually atomic level docking scoring, and then filters, and finally a drug-like molecule, right? That's our Dhanvantari pipeline. So the 3, 4, 5, 6 are the Sanjeevani part, and 1 and 2 are the genome and the protein part. So now what I would say is that you could come up with a million molecule library, or you could have your own database, or you can try a molecule. Methodologies are in place. In place for what? For a single molecule discovery. Some success stories have also already been reported here. So this is a estrogen receptor, and we found a molecule. When I say success story, it's been validated, computer prediction validated experimentally, in vitro studies mostly, and so also for some cell lines. So this is a molecule for biphenyl molecule. And then we have something about anti-Alzheimer's here, and then anti-malarials, antivirals. Antivirals, we had HIV, then chikungunya virus, and even more recently, drug-resistant HPV as well. So these are some zinc molecules identified by the Sanjeevani software. Out of a million molecules, we managed to capture 4 or 5, and they seem to work quite well. So the summary of these first few slides is that these methods are in place. Now, what about molecules? Well, more recently, we said, why zinc database when we have so many Indian medicinal plants? So why not do that? So my student Dheeraj got into the act, and he presented a poster. Some of you may have had chance to look at the poster in this. He's currently attending the conference, and he's in the audience, and he's presented. And what he did, along with other students, is to compile information on about more than 100,000 phytochemicals from 6,000 plants, Indian medicinal plants. You would say, is this really good? People have been doing this all along. Well, if you compare that with other softwares, other available databases. In fact, BIMP is one of the largest databases, which has a maximum number of molecules as of now, right? So this is good news. So we have now a collection of molecules, and all the properties have been worked out, including, the various drug-like rules and so on. As someone pointed out, some drug molecules may not obey all the various rules here, but what we find is that this BIMP molecule, that is in phytochemicals from Indian medicinal plants, 68% of them seem to obey at least one drug-like rule, not all of them, at least one drug-like rule. Now, once you have the molecules and let us say you have identified a target, well, you could do a quick scanning. So this RSPD plus, again, my student Dheeraj there in the audience developed this plus web tool. It can scan 1 million molecules or 1 million compounds. So 1 lakh is not easy story to do, right? And this can be done against a single protein target with a defined active site. So you have the summary of all this is that we did in fact find some molecules. I'm sure all of you recall during COVID time, we expected that Tulsi will cure or Giloy will do it. And then we did a calculation at that time. We did find some molecules from Tulsi blocking the NSP5. These are COVID protein, NSP16, NSP16, etc. And turmeric, neem, they all seem to be active. And we also did there was some experimental validation, and we were wondering whether our methodologies are able to capture or BIM molecules. And they do, in fact, ranked 5 here in this case, and then it's also ranked 23rd in the case of binding affinity. So the story then is computational methods for screening, docking, scoring, they're available, right? Whether you use Sanjeevani methodologies or made in India kind of thing, or you want to use commercial softwares, these are available now. Molecules, that is phytochemicals from Indian medicinal plants, are available. Again, BIMP is one database. Alternative, people have been using Zinc database, which promises almost 1 billion molecules now. Alternatively, one can draw a molecule or one can do a random design based on some fragments and so on. I'm sure all of you have heard of different methodologies for creating molecules to fit an active site. The question then is, from an Ayurvedic point of view, where is the target? Where are the biomolecular targets? Which proteins to target? So this is a primary question that we are addressing right now, and we are figuring out how do we handle this question, right? Earlier, based from a allopathic point of view, it was very clear that you eventually got onto a pathway, and from a pathway to a protein target, and then you started looking for an inhibitor molecule. But from an Ayurvedic point of view, I'm not an expert. Or some of you probably know much more than me on this Ayurveda part. Now, we have at least 7 Ayurvedic human body types, right? Vata, Pitta, Kapha, and different combinations. Apparently, this type 7 people seem to be very, very healthy people, right? And this is due to imbalances of the doshas. Now, various sources are available. This is our part 2, our dream plan. The dream plan 1 is done from a docking, scoring, etc. From single molecule, single target, no issue. Now, how do we handle Ayurvedic type? So now, for example, Vata, say there is an imbalance. Imbalance could be various things. Now, can we convert this into a set of proteins? That is a big question that we are right now facing. And if you're saying, well, here are the 100 proteins which are influenced or which are affected because of this phenotypical issues involved. So what we are seeing from this Ayurveda is a phenotypic description, and our urge here is to figure out what are the molecules that we are target. Similarly, for Pitta, again, imbalances are described. This is typical Ayurvedic way of describing. And now what we need to do is to convert this information or the knowledge into proteomics. So what we are proposing and what we are pursuing right now is that it's not just one human genome or human proteome. We probably have to define 7 types of, proteomes or genomes based on the three doshas or prakritis or vata, pitta, kapha combinations. So once you have these 7 standards, right, and defined, and you have them in your library, then of course conventionally this is done through Nadi Pariksha, pulse, etc. Diagnosis is done. But why not do a proteomic analysis? So this is with the assumption that 90% of the diseases are due to the proteins. Our proteins are good drug targets, right? It's not to say anything less about gene of the DNA or RNA. We have methods for tackling DNA-based or RNA-based drug discovery as well. Now, Ricky-3s are dosha imbalances that all of you know. Now, how do we compare them? So once you have the proteomic profiles of the 7 standards, and you know that the patient belongs to one of the 7, then you compare them and figure out which proteins are upregulated or downregulated. If you can do this, then rest of the story is very simple. Rest of the story is, well, go back to the database of phytochemicals and then do a docking scoring methodology to figure out what are the inhibitors, activators. So what we are now currently doing is to take these functional proteins in the human and create a library of molecules which will do inhibitors, activators. This is apart from the experiment. So there is no patient involved here, just to make a library, a lookup table, basically. Suppose now, tomorrow, let us say we have the proteomic, differential proteomic expression for the patient, right, for a given dosha, then then our task will be these 10 proteins have to be upregulated, these 10 have to be downregulated. Then we just do a lookup table from this. Number 3 or number 4 here. I should have written number 4, right? And of course, then there are issues of right solvent medium and how do we administer it. So those are issues probably Ayurvedic doctors can tell more about it. So I think this three prime are the ones that we are currently pursuing. Now, summary is need data on protein expression profiles, preferably with concentration, need a database of potential inhibitors. This is something that we are doing for all relevant proteins, although we don't know what is a relevant protein. So we decided we'll do it for entire human proteome. But then it's ideal that we define these 7 types and figure out what are the protein expression profiles for 7 standards as well as for the patients with a particular disease. We could be tissue specific protein expression profile as well. Right? Then of course need an estimate of concentrations and a functional a scoring function. These are some computational issues that I'm sure we'll figure out. How do we handle, right? Chemicals based on body type administration. How do we administer it? So essentially it's a multi-ligand, multi-target problem. So far from an allopathy drug discovery, we were looking at single target and a single molecule. Occasionally we do a cocktail, and then we figure out, well, maybe we should do 2 drug molecules targeting 2 biomolecular targets, right? But here is a Ayurveda will be a problem of multi-target, right? That's kind of a problem. Now, in terms of computational requirements, of course, there is a possibility of data storage, pre-calculated ones, and then create a lookup table. But if you want to do a priori, then you're dealing with a 100 petaflop kind of a compute resource. Right now, NSM machines across the country can handle petaflops easily. 100 petaflops, of course, is still a far cry. Hopefully someday we will get this kind of a compute power accessible to all our students and scientific community. So overall, if you were to scan and do this, modulate the concentrations and modulate the solvent issues, etc., then probably you're going to need exaflop machines. I think some people in the NSM community are thinking about creating exaflops in the country. So hopefully this will be a reality soon. Essentially, what I've presented to you is a possibility that this computational methodologies, especially with the AI emerging, can give you a combination of these chemicals from Indian medicinal plants for a particular disease based on the differential proteome expression profiles and suggest, well, to the Ayurvedic doctor, to the expert, that this is combination, this is the concoction that you probably have to give to this patient. And of course, he can verify, he can ensure that it is true and probably administer, and the patient probably gets cured, right? That is where things are happening, and I hope we'll get there sooner than I expect, right? But 10 years from now, this should be a reality. So that's usher in molecular Ayurveda. Thank you very much. I tried to cut it short because you're running late, and I admire your patience for staying so late. Thank you again for your attention. So some of my students who contributed to various software developments and acknowledgements to the funding agency, and this is our website. And all our softwares are free for usage, including the bioactivity information of medicinal plants. So that BEAM database is also freely accessible and downloadable. Thank you again.
Thank you, Professor B.J. Aram sir. It's a wonderful talk. You explained well about towards the molecular Ayurveda. And we have seen the many examples in the history of this India that Ayurveda molecules are the supreme in the biological action, and we human being cannot compete with them. As morphine is the example that we cannot able to discover a more competent painkiller than the morphine. And the another example is Sanjeevani booty. We cannot find the another Sanjeevani booty from the source of the organic compounds, but it's from the natural compounds. So it's a well explained topic, sir. And now I request to the audience, if you want to explore the topic, please share your words and questions.
Yeah, first of all, Professor Jairam, thank you so much for your very nice presentation and very new topic you've entered it. And I feel that the idea which you are thinking that to find out, it is very difficult and complex. I do understand that which proteins level go up, down by which you can then that's a very good idea, I think. And I hope that I think you will be able to do something so that at least one can find out some targets where they act, which we don't, but otherwise I think the idea has got a lot of potential. So I think it's a fantastic idea, and I think you will be successful in that. Thank you.
Oh, thank you, sir. Thank you. Thank you. Yeah, I think. Yeah, please feel free to email me in case you have any questions or doubts or any problems with the software. So I'll be happy to answer and share.
Yes, sir. Thank you for this wonderful talk and session. Now, in this scientific section, we have another speaker, a very renowned academician and researcher, Dr. Kotra. Kotra is an academic entrepreneur. Thank you, Professor B. Jairam, sir. Thank you.
Yeah, I'm closing. Yeah.
Yes, in this scientific session, we have another speaker, Dr. Kotra, academic entrepreneur with the experience in the drug discovery and the development. Kotra Group specializes in the area of medicinal chemistry, preclinical, clinical development of the small molecule and natural product based drug. It's a basic group behind all this research and development and the work. The Kotra Research Group has contributed a number of research papers and publications and various patents in this field. And Dr. Kotra recipient of several awards, including Premier Research Excellent Award from the Ontario, Canada, and R&D awards, various R&D awards, various research awards from the various pharmaceutical companies like Glaxo and other Canadian Society for the Pharmaceutical Sciences. Main aim of this Kotra Research Group is to discover and development of the drug candidates and the dietary supplements from the discovery to the clinical development to the market for the multiple areas. And Dr. Kotra is a co-founder of Vincentor Inc., San Diego, California, USA. Now we invite Dr. Kotra to explore the topic drug efficacy and the sex difference in the male in immune response. We welcome you, sir, and kindly start your presentation, sir.
Thank you very much, Mr. Chairman. And I appreciate the invitation to speak to the audience, and hello to everyone in the audience there, distinguished professors and scientists from all over the world. And I do recognize Dr. Dwivedi. I remember you from my undergraduate summer training at CDRI 35 years ago. And I'm sure Dr. Saxena is sitting somewhere in the front there. Hello, Dr. Saxena. And let me get started. I know I'm standing between dinner and yourself right now. Just a couple of, let me get my slides going here. Okay, so a couple of disclosures. I sit on a couple of companies' boards. This is publicly traded company. This is a private company, and anything I say here purely is my personal opinion. And these are all various organizations over the years that supported my research in Canada. And most of the work I'm going to be talking about today is coming from Eva Vasilevsky, a longtime researcher in my group. And most of the drug development that happens in my group, basically, all these guys contributed, both my collaborators and others. So we do a variety of things in my research group. This is a very recent introduction into the market. It's a dietary supplement for alcohol and alcohol intoxication called Unbuzzed. We have a number of other programs, including multiple sclerosis at phase 1, just cleared phase 1, diabetic neuropathy in Santor, phase 2 cleared, and few others that we deal with from market to the basic science level as well. And here in the next 15, 20 minutes or so, what I'd like to communicate is the importance of sex differences that probably are ignored universally in everything that we talk about, and why that should not be, especially in this day and age. So the immune system, which basically is extremely important in general in animals and certainly in the human being, we know from daily activities that it's different in males and females, from pain threshold, things of that nature. In drug development in general, whether small molecule drugs or biologics also, we do in vitro studies, but rarely we pay attention to the cell lines, whether they're male or female, or in vivo animal studies, and many times we tend to rationalize based on a variety of things, if we can develop the disease in the mouse or not, or a rat or a dog. And sometimes we do pay attention to the ratios of the males and females, but in efficacy studies tends to be a little less important. As well as in human, most of the times in development, the idea is to go fast into the market versus paying any types of these attentions in research. Post-approval, of course, we do epidemiological studies and then try to gather information and then try to rationalize, but that takes several years, if you will. And so we've been working on this area of generally ECS or endocannabinoid system for a number of years now. And I came across some literature, especially estrogen, testosterone, and our endogenous steroids, and their receptor systems, how integrated they are into endocannabinoid system. And these pathways are sometimes different in males and females. And because of this integration between the immune system and endocannabinoid system and our interactions with the endocannabinoid system and endocannabinoids, we thought, okay, why don't we explore this further and try to understand it? There's also evidence, and it goes back almost 2 decades, that in human as well as in rodents, this endocannabinoid system signaling is different, and also it affects the fertility as well in males and females. So it is an important system, plays a very integral role, if you will, in male and female physiology. So what is endocannabinoid system? It's not well probably dwelled into in our health related teachings, whether it's in pharmacy or medicine or wherever else it is. Endocannabinoids, these are molecules we make in our body. These are derivatives of so-called arachidonic acid. We are all familiar with arachidonic acid, but that's where our education pretty much stops. And a lot of them interact with so-called cannabinoid receptors. And these molecules, I'm sure, are familiar to you. Something called 2-AG and anandamide are very integral in this endocannabinoid system, and they are ligands to the CB receptors, if you will. And these receptors now also very closely involved in our body functions. The CB1 receptor modulates the neurotransmission in the CNS. And just to give you an idea how important these are, the levels of the CB receptors exceed essentially all other neurotransmitter receptors in the neuron. So it tells you how important these receptors are, just because of the numbers that our body produces. CB2, on the other hand, is mostly involved in the immune cells in the periphery. And exogenous cannabinoids, the so-called THC, CBD, those types of molecules that we are familiar with mostly, all of them act on these two and other receptors as well in the endocannabinoid system. This is just to give you an idea. I don't want to get into the details of this, but how cannabinoid receptors are so closely involved in the signaling of estrogen receptor and androgen receptor, testosterone receptor related intracellular signaling, very closely linked to proliferation, apoptosis, and other aspects that are involved in that. So over the years, we've had some drugs related to cannabinoids. Sativex has been in the market for about 20 years now. It's a THC CBD combination. Nabilon, a THC synthetic derivative for pain, vomiting, things of that nature. And dronabinol, it's a synthetic THC. We use it all the time now for HIV and cancer related. And the very recent introduction, I think about 8 years ago or so, Epidiolex that came for Dravet epilepsy related symptoms in that. We've been, CBD is what we started to look into because we've been working on it for a number of years now with this particular molecule. And it is involved in cell signaling, especially immune regulation on all immune cells like macrophages and other immune cells. You would find CB2 receptor, it's a GPCR, and the ligands bind on these GPCRs. And there are multiple modes of action, hence you see multiple cell signaling systems essentially taking on when these ligands bind on these immune cell receptors that in our immune system. Without going into a lot of history of it, because I want to show you the data, cannabidiol has a variety of activities through the CB2 receptor that I just mentioned to you. And one of the animal models that we have handy is something called MOG-induced EAE model. I'll describe that to you briefly, what that model is and how versatile it can be. CB2 cannabidiol, it basically downregulates the transcription of pro-inflammatory genes. That's how it tends to help with its anti-inflammatory activity. Now, we look back in the literature, most of these experiments, or all of these experiments are done in female mice. There is a very close link to that because it's easy to develop EAE mouse model in female mice. And also a lot of diseases related to immune system, such as arthritis and others that are used to investigate this, are typically considered a female dominant diseases. And hence, just in general research, animal research standpoint, you find evidence in female mice, let alone finding drug efficacy, in males and females. It's mostly tends to be females as well. And so we started to look into this more carefully, now employing both male and female mice in equal. And the way this mouse model develops is that basically you need to induce that. I mentioned this to you, MOG peptide. MOG is basically a myelin peptide that is injected into the mouse. Myelin is in the CNS. It's in the brain and spinal cord. And if you inject it peripherally, our immune system recognizes that an antigen and starts to develop autoimmune like disorder in itself. So you basically boost that with corticosteroid, and when we start to see symptoms showing up during the second week of the second week more or less of after the induction of the disease. And here you see a bunch of clinical scores that are written there. Basically how we see clinically if the mouse is getting worse or not. There's something called this clinical disease or course of the clinical disease. Tail is paralyzed, followed by hind limbs, followed by forelimbs, and then ultimately the mouse cannot move its head also and becomes basically moribund, if you will. So in this particular model, we basically start our treatment at the time when it reaches a particular clinical score, which is 1 in this case, and start to treat with CBD and look at the CNS, histopathology, and also blood for cytokines, basically looking after what happened to the mouse, when the disease is developed versus how is the treatment affecting, and when there is no treatment, what are the effects, and how is it different in males and females. Okay, so with that into the experiment here, I'll now talk about this, something called clinical disability that I was just mentioning to you. You just saw the scores, score 1, score 2, 3. Usually you do not let this, the disease go beyond score 2, 3, because after that it's irreversible. Pretty much the mouse goes to the dead state, if you will. So in this particular case, what you're seeing here are vehicle treated males and females. That is a male, this is a female. And as you see clearly here, with statistical difference, male mice reach the peak of the disease sooner. That's why you see that coming ahead of female profile, if you will. Now, males did experience slightly severe disease condition, and by day 18 or so, the chronic phase sets in, which is around here, and you see that both males and females have now comparable disease profile moving on. This is very important. Now, recognition that in males and females, the disease development itself is different, if you will. This is just to give you an idea. I hope you can see the video that gets communicated hopefully through Zoom. This is how a mouse looks like. This is a mouse that scores number 2, which is in a clinical score 2 there, where the hind legs don't work and the tail is not working at all. Only the front legs and the head. Basically, the mouse can feed itself, but really not comfortable otherwise, if you will. So this is now the disease state established, which show that it is different now. Now, how is the treatment with the CBD or the cannabidiol that operates on the ECS that has effects on the immune system? How is it different in males and females? So yes, what you're seeing on the graph here is the no treatment or vehicle treatment, if you will, in black, and in red is the CBD treatment in females. And you notice that the mice are getting better, and then the disease is getting worse after the 3rd week of treatment, if you will. Now, this in this middle graph, now the same profile of the clinical score, but in male mice, where you see that the clinical no treatment basically is continuing to be sick at around score 2, but with treatment with CBD, it's going down essentially here, more or less below 0.05 until the termination of the experiment, if you will. You see a small spike showing up there. We don't know. We terminated the experiment essentially at day 31 after the establishment of the disease. And here, a direct comparison of these blue and red graphs here, so males and females, directly comparing with the treatment of the drug CBD and how it is different. In females' case, the treatment essentially is not working in chronic treatment, whereas in males, we can see that efficacy is still shown after a month of treatment. So, and these are statistically significant differences that we find. So that's very, very exciting. Now, moving on here now, immune system, how is it getting different in males and females? First thing we looked at was that at the end of the experiment, take the mice, their brain, spinal cord, and blood. All 3 of them were analyzed. This is what you're seeing is the analysis of the spinal cord, and inflammation is localized mostly in the white matter of the spinal cord, and myelin damage is visibly higher in the spinal cord, and in both males and females, and perivascular lymphatic inflammation, which you see here in red arrows there, and also in the lymphatic infiltrates, essentially through those yellow markers there. We can clearly see them in both the vehicle treated and CBD, but in CBD treated, they are much less in comparison to vehicle because of the efficacy that we see. And this in the brain, similarly, you can see both of these types of effects, the perivascular inflammation there, but that's much reduced when CBD treatment is present. Now, here these are now more quantified than just looking at the pictures of this. And I'm going to highlight the message here to take so that we don't spend too much time here. In this particular case, you're seeing spinal cord lesions. So lesions in the spinal cord as measured percent area in the spinal cord section that is basically analyzed. And in males, you see statistically significant reduction in CBD treated males versus in females, while you see some reduction, this is not really statistically significant at all. And in brain lesions, again, we see average difference is much good, and it went down in CBD treated males, but in females, not much difference either. So the treatment really did not make much difference by that day. These are T cells. T cells are not supposed to be present in the brain or spinal cord. They are only peripherally restricted immune cells. However, in an immunocompromised, immune stimulated model like this, you will find CD3 positive T cells. Basically, CD3 is a marker on T cells in the CNS, into the spinal cord and the brain. What you see here is a healthy section in female and male. Literally, there is no stain that you would find in the brains. Now, vehicle treated, not CBD treated, you'll see all those black dots showing up there. Those are all CD3 positive T cells infiltrating the brain because of the brain protein. We gave it as an injection, you may remember, to stimulate the model. And the T cells, peripheral T cells, are thinking that there is a bad myelin. We need to get into the CNS to clean them up. That's how you're seeing a lot of this showing up there. And when treated with CBD, now you don't see that. There are very minor, black spots there. Otherwise, you don't see much of that, essentially telling us that the T cells got cleared from the CNS to a great extent. These are the, these are the quantifications of the same. As you can see here in male mice, very clear reduction in the T cells after CBD treatment, versus in females, not as much clearance in the brain, but in spinal cord, we see some clearance in females and in, as well as in males as well. So this is a clear indication now for us that in males and females, the T cell clearance is different, and that can be explained through the clinical scores that we saw. In males, the disease returned, so hence the T cell infiltration. Now you see that back in the brain again, whereas in females, it's returned. Whereas in males, the disease did not return, it's going down. So you do not see the same course and the T cells in the brain, which is essentially matching the disease course that we observed on the final day of the termination, if you will. Similarly, we can look at astrocytes. As you know, these are star-like cells in the brain. They are very, very important for metabolism, homeostasis, structural and functional integrity of the neurons, and they tend to act like immune cells, if you will, or in a protective nature. And in neuroinflammatory diseases such as the EAE model I just told you, you see all kinds of bad things happening and astrocytes getting activated. Very quickly to show you the data from that, we identified that using a GFAP marker, and in females, there is not much difference, whether CBD treated or healthy, they're all the same. So there is no astrocyte activity whatsoever, whereas in male mice, we see good activity kicking in with CBD as well as with vehicle as well. And in this case, the higher density of astrocytes was observed in the spinal cords of male, which is good. And together with the lesions that I just showed you earlier, it implies that there is a greater damage in the spinal cord, especially in the females than in males, if you will. I'm going to skip the macrophages and microglia just in the interest of time, but both are important immune system, if you will, in the CNS. And these guys tend to be active as well. And to quickly give you an idea on this, CBD treatment had comparable effect in both males and females with respect to microglia and macrophages, as you see from the graphs here. So we didn't find much difference in this case. Now, other important aspect whenever we talk about immune system is our peripheral immune system. So we obviously looked at this cytokines, which are all peripheral in nature, and starting from day one, when we give this MOG antigen, all the way the mouse is going through various changes in its peripheral immune system until we collect the blood here. So that whatever data you're seeing is basically the status of the blood on at the day of termination. And the idea for us is to see what are the pro and anti-inflammatory cytokine changes that have happened, and does it reflect what we are observing. So basically collect the blood, do a multiplex analysis. And I just want to remind you here that in females, you may recall that the disease state is almost as close as possible to the untreated mice. And in males, there is a significant effect of the treatment. So mice are closer to a normal state, if you will. So these are very short list of pro-inflammatory cytokines and their status. You can clearly see that interferon gamma levels have gone up, and this profile actually compares and corroborates some of the earlier data from the literature that when EAE mouse model is treated with the CBD, interferon gamma levels do shoot up, but you don't see that happening in females. It's only in the males that you see. Similarly, IL-1 beta as well as IL-12, both of them only in males, you see the changes happening, not in females whatsoever. And this is a very interesting observation, if you will. And of course, it's on the 31st day when we terminated the experiment, but it does show very clear difference. And also what this suggests is that both IL-1 beta and IL-12 are involved in promoting tissue repair and protection, which makes sense because in males, we saw better treatment effects than in females, if you will. So these are reflecting the clinical observations. With respect to anti-inflammatory cytokines, IL-4 levels don't seem to be much different, but IL-10, that's slightly reduced, as you see. It's not statistically significant, but slightly reduced IL-10 levels, protecting from chronic damage to the system in general. And IL-13 levels are also reduced when treated with CBD, but they are increased compared to the vehicle treatment versus CBD. Versus in females, it's the opposite, if you will. None of these are statistically significant, of course, as we see these are average levels that are different. And the fact that these are so different here raises one question for us for sure. Is there any vehicle effect? Because here you see from the vehicle standpoint of view coming much up there, all the treatment seems to show reduction there. Is there any vehicle effect? That's something that we need to investigate that further. But the take-home message out of this pro and anti-inflammatory cytokine profiles that, as I showed you, is that the response is different in males and females, and it corroborates with the clinical observations and clinical course that we see there. So for the first time, we demonstrate here that cannabinoids such as CBD show different treatment effects in the clinical disease course in males and females. And CBD affects the lesions in the brain and spinal cord differently. And also the microglia, astrocytes, macrophage level, macrophages are basically infiltrated macrophages coming into the brain, and T cells are quite different. And cytokine profiles obviously are different. And of course, they do align with the disease clinical course that we see. So now, if anything, why is it important to study? And I showed you a whole bunch of data and the numbers, whatnot, all of that. And why is it important? And it essentially goes to everything that you probably have heard in this conference so far, and whatever you're going to hear in future. Number one is pathology of the disease. If there is no disease, all of us can pack our bags and go home. So the pathology of the disease point of view, it's different in males and females. If we don't recognize that, then we obviously are not going to recognize that treatment could be different. So it's extremely important to understand the disease. Is it different from males and females? We talk about genomes, proteomics, and all of that. And very rarely, I would say there are efforts happening, but it's important to intensify those efforts equally to understand the differences in males and females and the disease course. And then the flip side there is what we are doing here, all of us, which is understand the treatment effects when we recognize the disease course difference, or the disease treatment effects are different, and how are they different from signaling point of view, basic mechanisms, if you will, so that we can treat the patients in a more meaningful way and get the appropriate patient compliance, and also the treatment, if you will, to be effective in almost all cases for males and females equally. And there is no, I think I would say, universal rule that this works in males versus that drug in females, and everything should be the same way. It's always, in every case, we have to evaluate and clinically justify that. So with that, I'm going to stop. I hope I did not go over time, and happy to address any questions that you may have.
Thank you, Dr. Kotra, for a wonderful research you have elaborated to the audience. And this is the end of this scientific session. Before to end this, I want to share and discuss some words about this topic, not related directly to this topic. As a universal truth, the male-female anatomy physiology will be slightly different always. As we have seen the example in our society, in this earth, that a lady monkey, after an accidental death of the baby, that female monkey carry this body of the baby till the all disappearance of the bones, of the skin. And see, in the Hindi words, we have seen Karuna, Mamta, Vatsalya. This is specifically characteristics found only in the females. It's a God gift and the powers given to the women, not to the men. Karuna, Mamta, Vatsalya. And these are the reasons behind this, that female monkey carry this dead body of the baby. And the male cannot. I think all the women are agree with my points. So there will be difference. God has given the special powers to the women on this earth instead of the men. And thank you for this research you have elaborated to the audience. Now I invite audience to share and explore this topic with any question and discussion.
Hello, sir. This is Sisir. Thank you very much for your beautiful lecture. Sir, who is more immunized, male or female?
Good to hear from you, Cecile. I don't know if you can universally have one answer for that. There are certain situations where, females tend to have more resistance versus males and vice versa. So I think it's context specific. But in general, when it comes to autoimmune disorders, we know that females are more susceptible. But that's not the entire story. There are situations where males are also susceptible.
Yeah. So, Premakant, first of all, congratulations that you are coming up, and I think still that vigor is in you. And I'm so happy that the whole new topic, this idea, I think this idea is very good because it has come, you have shown. But normally, this was perhaps not done because in always take male mice and male rats, when they test the model. So people then prescribe the same medicine develops. But I think this is a very kind of a new direction at all, particularly on this topic. And I think Lord, I'm very, very happy to see that your companies and you are growing. And I remember the days when you joined me in CDRI, how much what we had and how much you have gone. So it gives me a great pleasure. And I hope that you come up in your life still better and better.
Thank you, Dr. Saxena. I can see your
Thank you.
head from the back in the camera. Good to see you and good to hear from you. Thank you very much for the invitation. Also, I appreciate that.
Anybody else? Thank you, Dr. Kotra, for a wonderful talk in this scientific session. Thank you, sir.
Thank you very much. Have a good day. Bye-bye.
Thank you, sir. Now I would like to request Honorable Chairpersons, Dr. A.K. Dwivedi sir and Dr. Arun Kumar sir to present the memento of the speakers of this session. And I would like to invite Professor Dr. Indra Ghosh ma'am, Dr. Shrikant Rath sir, Dr. Alok Saxena sir to join us on the stage. And I would like to request Rajan sir to show the e-memento of Professor B. Jayaram sir and Professor Dr. Lakshmi Kutra sir. Thank you, sir. Thank you, ma'am. Now, I would like to request Professor Dr. Indra Ghosh ma'am and Professor Dr. Ashmita ma'am to please present the memento to the chairpersons of the session. Thank you, sir. Thank you, ma'am. Now, we have reached to the last session of today's evening, which is the online poster presentation session, and for which I would like to invite Professor Dr. Ashmita Samadar ma'am, and who is a well-known face to all of us, and Dr. Vaishali M. Patil ma'am to chair the session. And Vaishali ma'am is the principal and head of pharmacy department of Chaudhry Charan Singh University.
Now, this is the session for young researchers, e-poster sessions. Each e-poster presenters will present 3 minutes only. So senior members, if you would like to have your dinner, you please go. I mean to say, I am sorry because I am telling you go, no, means You please join the dinner. You can or else if you want, you can enjoy the lecture. Yes, and you please enjoy the dinner, and you can come after the dinner as well. Yeah. Yes. This session will be continuing. Yeah.
Thank you, ma'am. It's over to you.
Hello. So let's start with the first candidate, Abhishek Kumar Mishra, who will be speaking on exploring NAV 1.7 blocker in each management, insights from GX201 and sulfamethoxazole interaction. So over to Abhishek. Okay, the rules and regulation are 3 minutes for presentation and followed by 1 to 2 minutes question answer. So the time will be monitored.
Hi, good evening, everyone. Hope are you able to see my screen?
Yes, it is visible. Start.
So I'm Abhishek Misra from the Kaziranga University. Here I wanted to present my poster exploring sodium channel 1.7 blocker in the each management, and mostly the GX201 is a inhibitor. So as we know, the neuropathic pain is a severe and the long-lasting condition caused by nerve damage, often associated with diabetes, chromotherapy, and a spinal cord injury. So many pharmacological studies shows that the inhibitor sodium channel 1.7 can elevate each drug that block sodium channel, reduce histamine dependent itch, which is often triggered by allergic reaction and inflammatory process. In this one, by inhibiting sodium channel, this drug decrease sensory neuron excitability, thereby reducing each intensity. And here GX201, a potent sodium 1.7 blocker, is currently investigated for treating neuropathic pain. In this study, our goal is to determine whether sulfamethoxazole also bind to sodium channel 1.7 receptor and whether it shares the same binding site as GX201. If theoretically inhibit itching, our studies seek to highlight that from each inhibit sodium 1.7 alone may not provide complete relief from each, as other mechanisms such as histamine receptor activation also play a significant role. And in this one, our objective of the study is a molecular docking of GX201 and sulfamethoxazole with sodium channel 1.7. Second is the identification of binding site of GX201 and sulfamethoxazole. Third is binding pocket and binding site analysis. Revisiting the role of sodium 1.7 each management based on our findings, whatever is there. So, and in this one, I choose the 4 methodology. First one is a protein structure acquisition and optimization. So in that one, I got the data from protein data bank, and here is the link is given. Here's the detail is given over there. And third one is the acquisition of second one is acquisition and optimization of GX201 structure. A structure I determined from chemical structure GX20 was obtained from PubChem, and for 3D structure optimization, various molecule modeling and computational tools used to employ these things. And is Avogadro, which free and open source molecular building and visualization tool I used to do the acquisition and optimization of GX201.
You have only 1 minute. Wrap up, please.
So In this one, the result which I got, GX201, if you see here. So in the result, GX201 and sulfamethasazole binding to the same each site and on the NAV 1.7 and receptor, both they exhibit different bindings and energy with GX2, showing binding energy 9.2 calorie per mole and the sulfamethasazole showing the binding site of 7.31 kilocalorie per mole. And my future treatment is to target both sodium channel and histamine pathway, potentially using combination therapy for more comprehensive itch relief. And the conclusion is both GX2 and the sulfamethasone bind to the same site and site on the sodium 1.7 receptor, but they exhibit different binding energy with GX201 showing a binding energy 9.2. So this is the different bindings and both have a different energy for that. And this is possibly due to more hydrogen bond and better fit within the hydrophobic binding pocket compared to sulfomethoxazole. Thank you for your time and giving me opportunity to present my poster over here.
Okay, Abhishek. Next one is Akshay Kumar. You please brief about the methodology and conclusions and the main observations.
Akshay, you are requested to upload the presentation. Akshay, please upload the presentation. Next, Aman Mugdal. Aman Mugdal. Akshay Kumar is not here. Aman Mugdal is not here. But actually, acha. Bangmoi Das. Bangmoi Das. Bangmoi Das. Debashish Tripathi. Acha, Bang will be present. Okay. Bangmoi Dash. Upload your presentation. Bangmoi Dash, upload your presentation.
Good evening, everyone. Am I audible?
Yes, you are audible.
Okay.
Start presenting. Your PPT is not visible.
Is it visible now, ma'am?
No, it is not. You start your presentation. Start your presentation. You start speaking. If it is not visible, also start speaking.
Good evening, everyone. Myself, Manmay Das, Assistant Professor of Biochemistry. My topic is about engineer antibiotic strategy to combat multi-drug resistant bacteria. So first of all, we should know regarding the antibiotic. Antibiotic are the compound which are released from a microorganism to kill the another microorganism. The problem is associated with that, the antibiotic resistance, which are associated with explaining the antibiotic inactivation with enzyme and alteration in the target protein and membrane impermeability and the biofilm. So first of all, we should know regarding the biofilm. Biofilms are the bacteria community adhere to a place and form a slimy layer for which the antibiotics are unable to enter to the bacteria surface and kill the bacteria. So we should know what are the major problems associated with multi-drug resistant bacteria, threat to therapy, prolonged illness, high medical cost, easy target to the immunocompromised condition, and decrease the effectiveness of drugs and high mortality rate. We should know, according to Clinical Laboratory Standard Institute, antibiotic resistance, based on antibiotic resistance, bacteria are classified into three classifications: pan-drug resistance, multi-drug resistance, and extreme drug resistant bacteria. So according to World Health Organization, bacteria are classified into 12 categories which are associated with antibiotic resistance. So my topic is related to formulation of engineered antibiotics such as vancomycin. So first of all, vancomycin is a glycopeptide antibiotic which is used to kill methicillin-resistant Staphylococcus bacteria. But nowadays, antibiotic resistance is a major problem which is faced by current researchers, pharmacists, and biochemists, and health-related workers. So we should know the proper management, how to enhance the antibacterial activity of vancomycin. In an article, Zhao et al., a scientist worked on vancomycin and combined it with silicon oxide and cyclate as a nanoprobe to enhance the bactericidal property of vancomycin and kill the bacteria along with eradicate the biofilm, which is the major part of this research. Next, another topic associated with modification vancomycin with nanomaterial against both gram-positive and gram-negative bacteria. The adaptation of glycopeptide antibiotic on the nanomaterial and enhance the bactericidal property of vancomycin and combined with silica-bound iron oxide nanoparticles. Next is tetracycline, which is another antibiotic associated with various particles. Research combined with modifying the carboxamide group of tetracycline and combining it with cellocardium, which is a chemical compound, and somehow it is similar to tetracycline, but addition of some methyl group and other group, which is enhance the bactericidal property and antibacterial activity of tetracycline. And another, some of the other activity which are based on the modification of tetracycline, which are based on synthetic polymers such as polynitrile fibric 1-ethyl-3-dimethylaminopropyl is used to enhance the bactericidal property of tetracycline, which is useful to eradicate biofilm and enhance the antibiotic lifespan of tetracycline. Next is based on tetracycline and ciprofloxacin modification.
You have only 1 minute to wrap up.
These are the major methods to enhance the antibiotic lifespan and the properties of magic bullet. My topic is associated with an engineered antibiotic modification, which are the major topic for researcher to enhance the antibiotic lifespan and to eradicate the biofilm. Thank you, ma'am.
Thank you. Next, Debashish Tripathi. Debashish Tripathi. Debashish Tripathi. Start presenting.
Yeah.
Start presenting.
So, am I audible, madam?
Yes, you are audible. But you're not. Your PPT not visible.
There is some technical issues there.
You start speaking. No problem.
Okay. So, my topic focuses on the bacterial potential of bacterial potential of plant-derived bioactive compounds in food preservation. With the growing concern over synthetic preservatives, plant-derived compound offer a natural and sustainable alternatives. So here food safety is a critical challenge, and synthetic preservatives raise health and environmental concerns. So we can here discuss how bioactive compounds such as phenolics, alkaloids, terpenoids, and essential oils exhibit antimicrobial properties through cell membrane disruption, enzyme inhibition, quorum sensing, interference, biofilm breakdown. So prior to several scientific validation for using plant compounds in food safety, addressing concerns related to antibiotic resistance and chemical preservatives. So it is synergy we can find here as a synergistic effect between plant compound and conventional method, which impact the food quality, which ultimately increases the stability, increase the bioavailability and regulatory approaches to commercialization. Also, we are focusing here the main quorum sensing disruption, which is a promising strategy. This is a mechanism bacteria use to regulate gene expression, virulence, and biofilm formation through chemical signaling molecules called autoinducers. Disrupting this communication can weaken the bacterial pathogenicity, making them more susceptible to antibiotics and immune responses. Strategy like we use quorum sensing inhibitor, which block the signal reception, and also quorum quenching enzyme, which degrade signaling molecule. Also, we are here quorum sensing, which decrease the biofilm formation, which is a major cause of AMR. So here bioactive compounds like allicin from garlic, carbocol from oregano is also already being explored. This research paves the way for safer and more sustainable food preservation strategy. Thank you, madam.
Okay, thank you. Gayatri Khosla, upload your presentation. Gayatri Khosla, upload your presentation. Gayatri Khosla, upload your presentation. Next, Mayank Kumar, get ready. Gayatri Khosla. Okay. Mayank Kumar, get ready. Start presenting fast. Gayatri, you are not audible. You have only 2 minutes. Gayatri, you have 2 minutes and 1 minute more. Start speaking.
Hello, ma'am, my
Yes.
voice is audible?
Yes, start speaking.
Okay, ma'am. The title of my study is the quality by design based extraction of Palmagon from Palmago indica roots and its antioxidant activity. Here I have used a Palmago indica, which is a perennial herb or small herb that thrives in warm tropical climates, and it is grown as a medicinal and ornamental plant throughout the temperate and tropic zones. And chemically, if we talk about, this is a 5-hydroxy 2-methyl-1,4-naphthoquinone. It is a natural bioactive organic compound which is taken from the Palmago indica roots. It is a simple hydroxynaphthoquinone with commercial significance due to its vast spectrum of pharmacological activities like diabetes, anti-fertility, diabetic uses. And in this present study, we are using the utilizing the research surface methodology to optimize extraction parameters of Palmagon from Palmago indica root, and quantification analysis done using the HPTLC. The objective of the study is the optimization of extraction parameters for extracting the maximum amount of bioactive constituent from the plant in a single run using RSM method, and analyzing the antioxidant DPPH activity. First of all, the extraction was done by the 4 methods: oxide extraction using the oxide apparatus, then reflex extraction using the reflex, then UAE, that is ultrasound assisted extraction, and lastly extraction by maceration method. And they were optimized by the quality by design, specifically BBD. Quantification analysis of Palmagon in different extracts was done using HPTLC. Then further antioxidant potential of the extract was assessed with DPPH method. And in this, the result was find out that methanol is exceptionally effective extractive solvent, and the ultrasound assisted extraction approach yields a maximum amount of Palmagon. The ideal extraction temperature for pigment was determined to be 68.489 degrees Celsius, and the solvent to drug ratio to be 31.249 ml per gram. The extraction duration to be 35.682 minutes using the BBD, and the yield of the Promagin under these circumstances was 1.981 mg per gram, almost identical to the expected value of 1.893 mg.
You have only 1 minute to wrap up.
Okay, ma'am. With this, 79.39% percentage inhibition was found out using the antioxidant activity, and as a result, we can say that methanol was found to be the most effective and extracting solvent using the UAE extraction method. And here in the graphs, I can quantification was done using the HPTLC, and we can compare with the graphs that with the standard using the Promagin and the methanolic extract. The conclusion of the studies is that ultrasound assisted extraction method stood out to be the best among all the thermal and non-thermal modes of extraction, and methanol was proved to be the most efficient extracting solvent. And furthermore, plasmonic extraction was significantly affected by all the three different variables. The present work highlights the use of QBD, a multivariate statistical technique in the extraction field of therapeutically potent phytoconstant, which makes the optimization method less laborious and time-saving than the traditional optimization method. Thank you, ma'am.
Okay, thank you. I will request all the participants who are present online, don't decline the panel that has been provided from GTHTM. Don't decline the panel. You have to accept that so that your presentation can be seen. Okay. You upload the presentation there through panel list. Okay. Okay. So we have Neha Mathur. Dr. Neha Mathur. Dr. Neha Mathur, please upload the presentation. Dr. Neha Mathur, please upload the presentation. In between Mayank Kumar. Okay. Muhammad Tafsil. Pallavi Suyal. Pallavi Suyal. Don't decline. You are being provided the link or the panel link. Don't decline the link. Accept the link so that you can present.
मैम मैं तो नाम तो आ गया मैम, मैं लिंक कैसे हूँ इसमें?
हाँ
नीचे से वो आ रहा है लोअर हैंड QA मोर करके.
There is a button that is the
लोअर हैंड को है?
share. You have to share the screen. We have
PPT शो ही करना नहीं आ रहा मुझे तो.
panelist.
Okay, you start speaking, no problem. You start your presentation. You start speaking, Pallavi. Pallavi, are you there? You start speaking.
We had provided you the control, and please share your screen through the green color button that is on the control panel.
Good evening all of you.
Both of them are there. Who is Pallavi? Okay. Start speaking, start speaking.
Yes, please share your screen.
Good evening all of you. I am Pallavi Suyal. My title is Unveiling the Therapeutic Potential of Fluliconazole, a systematic review on preclinical and clinical safety and profile, future prospects. Introduction. Fluliconazole is a new antifungal agent from a azole family intended for topical use in treating infections caused by dermatophytes. It is mainly utilized for Tinea pedis, Tinea cruris, and Tinea corporis. The antifungal efficacy of fluliconazole might stem from its notable in vitro activity and favorable pharmacokinetic properties. within the skin. Luliconazole cream is a proven and safe topical solution for dermatophytes infections. Its low water solubility poses challenges in developing alternatives formulations. Objectives identify challenges and limitations, highlights drug resistance concern, gaps in current research, discuss emerging nanoparticles based on formulations. Mode of action: luliconazole antifungal that exerts its effect by